galactose.
Yeah.
Ma'am, can you hear me? Yeah. So
Yeah,
it is
I can hear.
comprised of repeating units of mannose and galactose. So my focus here is on the galactose residues which are present on Gorgum, like how I mentioned, because this galactose will have affinity to the asialoglycoprotein receptor of liver, which is overexpressed by the hepatocarcinoma cells.
What is a common name for Gorgum? This plant? This plant, do you know? What is
the plant name is Siamopsis. The plant name is Siamopsis tetragonaloba.
I'm just asking you do you know the name which we call it normally, like this thing, like the Malayalam name or something like that? That's what I asked you.
Vernacular names.
No, ma'am.
Vernacular name. Yeah, vernacular name of Gorgum.
Okay, fine. Okay, sorry, so thank you. Anyway, good
yes,
presentation.
Thank you.
Continue. Hope so you continue well and proceed further in your research.
Yes.
Wishes success.
Thank you.
Okay, thank you, Tina.
Yes.
Thank you.
Okay, ma'am, can we move to the next? Okay.
We can move on.
Yeah, okay. Thank you, Dr. Sharil and Dr. Vinod Kumar. So we will go to the next participant.
And the next participant, OPSC20. OPSC20, Akhil K. Mohan.
Yes, ma'am.
Yes, Akhil, you are audible. You can now start sharing your screen.
Okay, ma'am.
Yes, it is visible. You can start the slideshow.
I hope it is visible now.
Yes, it is visible, Akhil. You can, the time starts now for you.
Good afternoon, all. I am Akhil K. Mohan, PhD student working under the guidance of Dr. G.S. Vinod Kumar, Rajiv Gandhi Center for Biotechnology, Tirunelveli. I would like to share a few findings of my study entitled Cytotoxic Peptide and Targeted PLA Nanoparticles for Breast Cancer. As we all know, cancer is formed due to the failures of mechanisms that control growth and proliferation of cells, and is a leading cause of death. There are reports that 1 in 8 deaths in the world is due to cancer, and recent reports tells that by 2030, global cancer burden will shoot up to greater than 23 million new cases and greater than 13 million cancer deaths. And I'm directly coming into peptides, the application of peptides in drug delivery application. It opens many doors in the field of targeted drug delivery with their high specificity, selectivity, small dimensions, ease of modification, and high biocompatibility. Among all the targeting agents, peptide presents promising opportunities in designing novel therapeutics due to their small size and the ease of large-scale production. Added advantage of using peptides include lower immunogenicity than monoclonal antibodies or proteins, and no accumulation in kidney, liver, or any other organ. Now, PEP5, it is a hydrophobic peptide. When introduced into cancer cell lines, it can arrest the division of tumor cells. And there are reports that it can reduce tumor volume in vivo by almost half. And actually, PEP5 is an intracellular peptide, which is generated by the degradation of cyclin D2 during G1S transition of the cell cycle. And expression of cyclin D2 is low in most of the tested breast cancer cell lines. So cyclin D2 may act as a tumor suppressor. And when PEP5 is reintroduced into such cell lines that express a low level of cyclin D2, they arrest their cell division. Therefore, PEP5 presents a relevant therapeutic potential by preventing the carcinogenesis of cancers with low cyclin D2 expression. We synthesize this peptide by solid phase peptide synthesis using FMO chemistry to target malignant breast cancer cells. Then coming to another peptide, TKD. Normally, human tumor cells express a heat shock protein 70 in their plasma membrane, which we will call as MEM-HSP70. It is a potential target of tumor diagnostics and therapeutic procedures. because they are present in human tumor cells in their membrane and absent in adjacent tissues or other healthy cells. Actually, TKD is a 14-mer peptide sequence that derived from the oligomerization domain of MEM-HSP70. So this TKD showed specific binding with MEM-HSP70 and rapid internalization in cancer cells. We synthesize TKD peptide also by FMO chemistry to target breast cancer cells. It has been proved that biopolymeric nanoparticles enwrapped with drugs are effective and extensively useful for therapeutic purposes. Biopolymers like polylactic acid are frequently used for constructing therapeutic agents. They are well known for their properties like biodegradability, biocompatibility, enhanced protection of the drug from degradation, and the possibility of sustained release. These unique attributes makes them attractive biopolymers for targeted drug delivery systems. In the present study, we have synthesized a drug delivery system that can deliver a hydrophobic anti-cancer agents to breast cancer cell, irrespective of the growth factor expression status of these cells. So in this work, we synthesized PEP5 enwrapped PLA nanoparticles, and these nanoparticles were modified with the targeting ligand TKD. Initially, so this is the methodology we followed. Initially, we synthesized both the peptides using standard FMO chemistry and are characterized using mass spectrometry using Malditov mass spectrometer. Then peptides was enwrapped into PLA nanoparticles by a modified double emulsion solvent evaporation method. Then the size and morphology of the nanoparticles were characterized by dynamic light scattering and TEM analysis. Then in vitro evaluation of the particles, including cytotoxicity analysis, cell uptake study, clonogenic assay, and live dead assay were done on two different cell lines, MCF7 and MDMB231 cells. Here, MDMB231 is a triple negative breast cancer cell, and MCF7 is an ER positive breast cancer cell line. Coming into results, so we synthesized both the peptide TKD and PEP5 using FMO chemistry, and molecular weight of the peptide was investigated by mass spectrometry using Malditov mass spectrometer and is found to be 1667.38 and 1056.620, which agrees with the calculated value of the peptide 1600. We obtained a value of 1673.78 and 1056 and 6.62, which agrees with the calculated value of 1667.38 and 1056.620 respectively. Then we synthesized peptide enwrapped PLA nanoparticle by a modified water in oil water double emulsion solvent evaporation method. Then particle size, these particles are characterized and their particle size is measured through DLS and TEM analysis. So here particle without PLA nanoparticle without peptide, we got a size of 229.2 nanometer, and for peptide enwrapped particle we got a size of 301.6. So this increase in size confirms the enwrappment of peptide into the particle. TEM analysis further confirms the morphology and size of the particle that we analyzed by DLS. Then TKD was also conjugated to the surface of the nanoparticle by a carbonimide chemistry, and we measured the theta potential or surface charge of the nanoparticle before and after TKD conjugation to the nanoparticle. PEP5 enwrapped PLA nanoparticle showed a surface charge of minus 15.14, whereas after TKD conjugation, it decreased. The particle charge shifted to minus 11.60. So this shift in surface charge of the nanoparticle indicates the effective conjugation of TKD with the nanoparticle. And this double solvent evaporation method gave a good yield of 76%, and it gave an entrapment efficiency of 75.89%. And we done an in vitro peptide release study, and the particle showed a burst release mechanism during the initial 12 hours. It is evident from the graph, and then release ceases. Over the experimental condition, the entrapped particle showed a release of 79.15%. Then we done the all in vitro analysis. Initially, we checked the cytotoxicity analysis of PEP5 entrapped nanoparticle on MCF7 and MDMB231 cell lines. We compared the efficacy of PEP5 on peptide only and PEP5 entrapped PLA nanoparticle and TKD conjugated PEP5 PLA. Both the cells exposed to each of these formulation at a concentration of 250, 300, and 350 micromolar. And cells were exposed to this much concentration for 48 hours. And in all the tested formulations, cytotoxicity increased in a dose-dependent manner in both the cell lines. And this results indicates that if you see the TKD conjugated particles show a high cytotoxicity, so which satisfies the therapeutic potential of TKD as a targeting ligand. And by contrast, PLA nanoparticles without peptide showed no cytotoxicity in both the cell lines. So which shows the biocompatibility of the nanoparticle in using encapsulation of the peptide. Then we cell uptake analysis of the particles. So here we use MDMB231 cell lines that have a green inherent green. We use an MDMB231 cells that is transfected with a green fluorescent protein, so that these cells will express these cells will fluoresce green color in their membrane. And we stain the nucleus with haste, and rhodamin D entrapped nanoparticle was used to study the cell uptake. Then from the image, it is clear that TKD conjugated nanoparticle is internalizing into the cells at an incubation time of 4 hours. Then we also done a clonogenic assay using the same formulation like free peptide, peptide entrapped PLA nanoparticle, and TKD conjugated PLA PEP5 nanoparticle. And these results indicate that PEP5 and all these formulations, they significantly inhibited the clonogenic potential of these cells. But in control, there are colonies formed. So we didn't treat control with any of these agents. So this shows PEP5 can inhibit the colony formation ability of tumor cells. Then we performed a live dead assay again to check the ability of peptide to induce cell death in cancer cells. So here also we check the activity of peptide in two cell lines, MCF7 and MDMB231 cells. So where green color represents live cell and red color represents dead cells. So from the image, it is clear that TKD conjugated PEP5 entrapped nanoparticle shows more death. So which suggests the use of TKD as a targeting which suggests the use of TKD as a very good targeting agent and PEP5 for killing tumor cells. So conclusion, here we developed TKD conjugated PLA nanoparticle entrapped with PEP5 for breast cancer targeting, and the efficacy of the nanoparticles in exerting anti-cancer activity was proved through biological evaluation on two different cell lines. And with the ability to induce tumor cell death, PEP5 claim an attractive position in the fight against cancer. Thank you.
Thank you, Akhil, for your presentation.
Thank you.
Now we will move on to the QA session. Dr. Vinod Kumar and Dr. Sharil, please.
It is a wonderful presentation, wonderful piece of work.
Thank you.
So actually, there is no much questions to say. It's a very good piece of work. Yeah, congratulations. Okay. So are you trying for in vivo studies?
Yes, sir. Of course, the work is going on.
Okay, fine. So all the best. Yeah.
Good work, Akhil. That's what I would also like to say, because you have gone conclusively in all directions, and I guess some more work which would like rather than the in vitro. The in vivo studies would conclusively help you to prove your observations. That's what I would
Okay.
also like to say. And
Okay.
and I guess I would, this is a small suggestions. Of course, different people have different ways. I would like you to be a little more confident in your voice. That would, you have done a very good work. Okay.
Okay.
So just when next time when you present, just try to be a little more confident. That would help you a lot in your further progress. Anyway, good work, Akhil.
Okay. Thank
So,
you.
thank you.
Thank you.
That's it.
Thank you, Dr. Vinod Kumar and Dr. Sharil. So there is no questions in the chat box also. So we will go to the next participant.
The next participant, OPSC 21. OPSC 21, Arya TP. Arya,
Hello,
are
ma'am.
you present? Yes. You are audible, Arya.
Okay, ma'am. Shall I
Please
start?
present. Please share your screen.
Okay, ma'am. Ma'am.
Yes. It's not visible, Arya.
Ma'am.
Have you shared? Yeah, it's coming. Yeah.
Okay, ma'am.
If you're presenting through mobile, you have to zoom your slides.
Okay, ma'am.
Okay.
Shall I start?
Okay, now, okay, your time starts now.
Very good afternoon to everyone.
Arya, you're not audible. Your voice is breaking.
I am Arya.
I can't hear Arya. Is it the same for everyone?
Yeah, it's
Of
same only.
Department of the Asha Gangadharan. Ma'am.
Your sound is breaking, Arya.
Oh, God.
Arya, your voice is breaking in between.
Shall I start?
Okay.
Yes, now it's clear. Start presenting.
Okay, ma'am. Very good afternoon to everyone. I am Arya, student of Department of Biochemistry, Marathnicious College, Kodamangalam. I done this work under the guidance of Dr. Asha Gangadharan, Assistant Professor, Department of Biochemistry, Marathnicious College, Kodamangalam. Today I am going to make a presentation on the topic green synthesis and characterization of zinc oxide nanoparticles using aqueous extract of hemigraphis colorata and its in vitro antibacterial, antioxidant and cytotoxic activities. Let me begin the presentation with a brief introduction. Nanoparticles are the manipulation of matter through certain process to create specific properties which is used for particular application. Zinc oxide nanoparticles are considered to have many characteristic features for biomedical application. Out of physical, chemical, and green synthesis becomes an emerging field in nanotechnology as it is an environment-friendly, cost-effective, biocompatible, and safe properties. However, there is a need for scientific validation, standardization, and safety evaluation before recommending them as drugs. Hemigraphis colorata is a medicinal plant which is used for wound healing in traditional systems of medicine. Aim of our study is green synthesis and characterization of zinc oxide nanoparticles using Hemigraphis colorata and analyze its in vitro antioxidant, antibacterial, and cytotoxic activities. There are three main objectives present. The first one is to synthesize the zinc oxide nanoparticles using aqueous extract of Hemigraphis colorata, and the second one is to study the in vitro antibacterial, antioxidant, and cytotoxic activities of the zinc oxide nanoparticles. And third one is to characterize the zinc oxide nanoparticles using UV spectroscopy, FTIR, XRD, and TEM analysis. Here, the experimenter, here next we move into the materials and methods. Here, the experimenter plant named the Hemigraphis colorata, and it's belong to the family of Acanthaceae. In Kerala, it is named as the Murikuti. For the preparation of the plant extraction, for the preparation of the plant extract, chocolate extraction method is used. Here, the present study, water extract is used, and the extraction yield was calculated. The co-precipitation method using zinc sulfate and zinc nitrate is here used as a precursor for the synthesis of zinc oxide nanoparticles using aqueous extract of Hemigraphis colorata. Here, the evaluation of the antimicrobial activity of zinc oxide nanoparticles are done by antibiotic sensitivity test and the antibacterial assay by agar-well diffusion method. Here, the antibiotic sensitivity test is done by using the test organisms like E. coli, Klebsiella, Pseudomonas, Staphylococcus aureus, and the antibiotics used are penicillin, gentamicin, chloramphenicol, and ampicillin. Here we use in antibacterial assay by agar-well diffusion method. Here, 50 ml of zinc oxide nanoparticles in DMSO with the concentration 1 mg per ml is tested against the E. coli, Klebsiella, Pseudomonas, Staphylococcus aureus. Then in vitro antioxidant activity of the zinc oxide nanoparticles were calculated by using DPPH radical scavenging activity and FRAP assay. Then the in vitro cytotoxic activity was done by MTT-assay. Here the cells used are CH2K1 cells maintained in DMEM F12 medium, and the samples collected were plant extract and the zinc oxide nanoparticles which is synthesized from the aqueous extract of Hemigraphis colorata using zinc sulfate as precursor. Then the viability of the cells were calculated by the equation mean OD of the sample minus mean OD of the blank divided by mean OD of the control minus mean OD of the blank into 100. Then the characterization of the zinc oxide nanoparticles were done by using 4 methods like UV visible spectrophotometry, Fourier transform infrared spectroscopy, X-ray diffraction, and transmission electron microscopy. Here the result of the antibiotic sensitivity test. This result shows that the gram-negative organisms like Klebsiella, E. coli, and Pseudomonas are multi-drug resistant because they are resistant to all the 4 antibiotics present, and the gram-positive organism like Staphylococcus aureus are resistant to chloramphenicol, and they are intermediately sensitive to penicillin, and they are sensitive to ampicillin and gentamicin. Then here is the figure shows the result of the antibiotic sensitivity test. Here, the result of antibacterial activity of the nanoparticles, which shows that the zinc oxide nanoparticles which is synthesized from zinc sulfate as precursor with aqueous extract of Hemigraphis colorata, which shows the inhibition to Staphylococcus with 60 millimeter, and the zinc oxide nanoparticles synthesized to Staphylococcus aureus, and the zinc oxide nanoparticles synthesized from zinc nitrate as precursor with aqueous extract of Hemigraphis colorata shows inhibition to Staphylococcus aureus with 10 millimeter only, and the then in the case of Klebsiella, the zinc oxide nanoparticles which is synthesized using zinc sulfate as precursor shows inhibition to Klebsiella with 9 millimeter only. Then in vitro cytotoxic effect by MTT assay shows that the samples with the HC1 is the aqueous extract of the Hemigraphis colorata, and HC2 is the zinc oxide nanoparticles synthesized using aqueous extract of Hemigraphis colorata from the zinc sulfate as precursor. Both these values indicates that they have low cytotoxic values, and they are non-toxic to normal cells, and it is beneficial for the drug delivery and wound healing, etc. Then here is the characterization of the zinc oxide nanoparticles which is synthesized from the aqueous extract of Hemigraphis colorata using zinc sulfate as precursor. Here the formation of zinc oxide nanoparticles was confirmed by the UV spectroscopy and the FTIR analysis. Here is the figure indicates the transmission electron microscope images. Here it indicates the zinc oxide nanoparticles which exist in the heterogeneous nano agglomerates. Then the XRD from XRD, it is calculated that the particle size was calculated as 9.18 nanometer, corresponding to 100 nonpolar plane. The sharp diffraction peaks were observed at 2 theta values of 59.507 degree, 36.454 degree, 34.613 degree, 33.291 degree, and 28.660 degree. Then the 100% intensity was showed at the degree of 33.291 degree, which correspond to zinc hydroxide. Then the wide appearance of a peak indicates the conformation of nanoparticles. The presence of zinc oxide nanoparticles was confirmed by matching with the diffraction peaks. The Joint Committee on Powder Diffraction Standards, JCPDS, card number 0361451 for zinc oxide nanoparticles. Other extra peaks may be due to the phytochemicals absorbed, which can be further confirmed. Then the summary of the study is zinc oxide nanoparticles was synthesized using aqueous extractor hemigraph colorimeter from zinc sulfate and zinc nitrate as precursors. The antibacterial study showed that the zinc oxide nanoparticle was effective against Staphylococcus aureus. The zinc oxide nanoparticle synthesized from zinc sulfate precursor possess antioxidant activity. The cytotoxic effect evaluated by MTT-assay revealed that the synthesized zinc oxide nanoparticle was non-toxic to normal cells. IC50 of the zinc oxide nanoparticle was found to be 54.51 μg per ml. The characterization of the zinc oxide nanoparticles by UVB spectroscopy, FTIR, XRD, TEM result confirmed that the zinc oxide nanoparticles produced by green synthesis exist as heterogeneous agglomerates, and the particle size was calculated as 9.18 nanometer in 100 nonpolar plane, 100 nonpolar planes. Then the conclusion of the study was the green synthesis of the zinc oxide nanoparticles using hemigraph colorimeter provides an effective route for eco-friendly method. The zinc oxide nanoparticle material proved to possess significant morphological and structural properties. Also, it has exhibited potential antibacterial and antioxidant activity with a low cytotoxicity. Hence, it can be concluded that the synthesized zinc oxide nanoparticle is a suitable drug candidate for various biomedical applications like drug delivery, wound healing, dermatological applications as dressing material, skin care, cosmetics products in future. That's all. Thank you.
Thank you, Arya, for your presentation. The floor is now open for QA. Over to you, Dr. Sharil and Dr. Vinod Kumar.
So Arya, it's a good piece of work and it's a good presentation. So
Thank you, sir.
can you show me the particle size once of particle size? Have you calculated the particle size analysis of this nanoparticle?
Yes, sir. Yes, sir. Sir,
Yes.
the
Okay, you
particle
have
size was
calculated based.
calculated.
Yeah, you have calculated based on the XRD. Okay, fine. So that was my question: whether you have calculated based on the lattice size or your particle size analysis analyzer. Okay. So another thing I want to mention this is antioxidant activity you have checked. So for that you don't have a duplicate results, right? So how we can conclude whether the results are will be consistent for your nanoparticles? So you have only a single data for all these assays. So so that you need to address. So whether it is a PG or a PG work or a PhD work.
It's a PG work, sir.
Okay, but still, I think at least you need to have a duplicate sample so that you can say this is a what it is a significant result. So then only you can say whether it is consistent at happening or not, right? And you can at least just
Yes,
for
sir.
your information, at least Arya, just see to this chemical formulas where you need to have subscripts and superscripts. Just take care of these FeSO4.
Okay, sir. Okay,
Yeah,
sir.
yeah,
Okay, sir.
yeah,
Definitely.
yeah. So you don't present somewhere in a wrong way, right? So these are the things basically coming into
Okay,
things.
sir.
Okay.
Thank you.
So, but it's excellent piece of work. You have done a good TEM image. What is the shape of this zinc oxide nanoparticle from the TEM image?
Heterogeneous it's exists in the heterogeneous nano agglomerates.
Okay, but you cannot predict the shape of the thing as such, right?
Yes, sir. Yes. Yes.
Okay, fine, fine. Okay, okay. Thank you, Arya.
Thank you,
Thank you.
sir. Thank you,
Yeah.
sir.
Okay, Arya, a good piece of work. In fact, for a PG work, it is well and good. But as you told, to make your work more satisfactory, you could include little more triplicate values as well as statistical evaluation of your work would be sufficient enough. And
Definitely, ma'am.
it's good. And I would also like you to, if you are interested, probably you could also extend your work probably into the practical applications of it in animal model and all like that. That would be more efficient because do you know what is the common name for this plant?
Hemigraphis colorate is the scientific name, and
Yeah.
Murikuti. Murikuti in
Okay.
Kerala, it's known in the name of Murikuti.
So what practical application can you possibly use for this? Any idea?
Yes,
I know that. I'm
definitely,
asking you.
ma'am. Ah,
What
okay,
is
ma'am.
I want your answer.
Uh,
That's it.
Okay,
Hmm.
ma'am.
Uh,
Hemigraphis colorata is traditionally used as a medicinal plant, and it is excellent wound healing property. It have.
so what is your next practical application of this? That's my question.
Next,
What can you do?
yes, ma'am. Next, I could find the anti-cancer properties. It's just a preliminary test I was done only. I could next prefer the anti-cancer property of the
Okay, that
plant.
is there. Also, might be you can also go into like certain grafts and all that wound healing, no? Where the wound healing is not easily possible. Okay, you can also try to develop grafts where these particles have been incorporated. Okay, I'm just telling about the practical extent of it. Okay,
Okay,
so just
ma'am.
think
Thank you.
over it. Just think over it. That's
Okay.
it.
Okay,
So
ma'am.
anyway, thank you. You did a good
Thank you,
work.
ma'am. Thank you, ma'am.
Thank you, Dr. Sharel and Dr. Vinod Kumar. So we will go to the last participant.
Okay, I think the presenters are over. I'll just call out the
Absentees.
Absentees. Is Kirtana S, OPSC 18 present? Kirtana S, OPSC 18 present here? Okay, I think she's not there. So I'll mark her absent. Okay. Next, is Ashwini Sabu present? Ashwini Sabu?
Yes.
Ma'am, I'm here.
Yes, Ashwini. Yes, yes.
Yes, ma'am.
Okay. Asha Miss, can you please stop sharing your...
Screen.
Okay. Okay, Ashwini is ready now. So, chairs, please note, Ashwini, code number OPSC6 is going to present now. Ashwini Sabu, OPSC6 is going to present now. Ashwini, start sharing your screen. You want us to share?
Yeah, yes, ma'am.
Okay, Asha ma'am. Okay, Ashwini, you are presenting. I think you are sharing the screen.
Yes, ma'am.
Okay, you can share. Please zoom your slides. Okay.
Yes, ma'am. Okay, wait. Ma'am, now it's okay?
Yes, now it is visible and it's good. Okay, now Ashwini, you can start presenting. Your time starts now.
First of all, Good afternoon, everybody. I am Ashwini Sabu. I'm talking about toxicity studies and regulations of biopesticide or pesticides under the guidance of Dr. Jisha MS. Let's move to the introduction. A pesticide is any substance used to kill, repel, or control certain forms of plants or animal life that are considered to be pests. Sometimes pesticide affecting or non-targeting organisms like fish, bees, butterflies, birds, and other beneficial organisms in soil, underwater bodies, and including human. Kerala has banned pesticides are monocrotophos, carbofuran, atrazine, and few others in 2011. Pesticides are classified different types: rodenticides, herbicides, nematicides, molluscides, fungicides, insecticides. Then go to biopesticides. Biopesticides are the byproduct of insects, nematodes, microorganisms, plants, as well as chemicals. Biopesticide classified microbial biopesticide, plant incorporated pesticides, and biochemical pesticides. Then aims and objectives. To study the toxicity of biopesticide in soil and earthworm, to evaluate the biopesticide toxicity in fish, Orichromys nicoticus. Then move to the materials and methods. Toxicity studies are conducting for the evaluation of environmental impacts caused by the pesticides or biopesticide application. Toxicity testing is well established science for the comparing hazards of chemicals to a particular test organism. Then first of all, the earthworm toxicity studies. Earthworm toxicity studies mainly two methods are used. First one, paper contact toxicity test. Then the procedure of artificial soil test. Earthworm incubated in 750 gram of artificial soil for 24 hours. Then expose different concentration of pesticides. Then container covered with a polypropylene sheet for avoid evaporation. Earthworm incubated for 40 days. Then observe behavioral changes and mortality. Then filter bubble test procedure. Take flat bottom glass vials. Their sides are lined with the filter paper. Test substance dissolved in water. Then 1 ml of solution taken in each vial. The earthworm kept on moisture filter paper for 3 hours. Earthworm placed in a test glass vial. Then each glass vial sealed with a plastic film for avoid evaporation. Then test duration 4 days. Behavioral changes and mortality are noted. Then second one, the soil studies for the mineralogical characters. Kethel method used for the nitrogen analysis. Then Breyer's method number 1 is test for the phosphorus. Flame photometer test for the potassium. Third for deheterogeneous activity. The first Kethel method. 1 gram of soil sample taken in a Kethel flask. Add 0.5 gram of copper sulfate, 50 gram of K2SO4 and 30 ml of H2SO4. Then add small amount of paraffin to reduce frothing. Then allow it to cool. Then 50 ml of water and transfer the distill flask. 20 to 25 ml of is added in the conical flask. Add 2 to 3 drops of methyl red indicator. Then add water and tap water runs through the condenser. Then add 30 ml of NaOH in the distilled flask and heat gently to distill the ammonia for 30 to 40 minutes. Then titrate excess acid in the distillate with 0.1 ml of NaOH. Then calculate the nitrogen level in the soil sample. Then Breyer's method number 1. Add 50 ml of Breyer's extract to the 5 gram of soil sample in conical flask and shake for few seconds and filter. Then prepare stock solution. 0.21 gram of KH2PO4 is dissolved in 1 liter of distilled water. 10 ml of stock solution dilute with 0.5 liter of distilled water. Then take 0, 1, 2, 4, 6 and 10 ml of solution separated into 25 ml flask. Then add 5 ml of extraction solution, 5 ml of molibrate reagent and dilute with 2 ml of distilled water. Add 1 ml of dilute SnCl2 solution and take again and dilute to 25 ml. After 10 minutes, detect the blue color of the solution on the spectrophotometer and wavelength. Potassium test. Potassium test. Add in soil sample. Then shake and filter. Then determine the potassium. Then soil dehydrogenase test. Soil sample taken in a test tube. Add 2.5 ml of phosphate, 0.2 gram of CaCO3 and 1 ml of substrate. Thus, test tubes are incubated at 25 degrees Celsius for 72 hours. Then sample centrifuged at 3000 rpm for 10 minutes. Supernatant liquid was removed. Add 5 ml of methanol and shaken for few minutes. Again 5 ml of methanol added and repeat the operation. Again the tubes were centrifuged. Supernatant liquid was poured in a clean tube. Then absorbance of solution was of concentration was calculated using calibration curve. And fish toxicity will affect fish, just fish slugging and swimming ability, reduce the ability to feed, change color. The selection of species depending on the regulatory requirements and environmental factors. Then red tilapia Orichromys nicoticus kept in a water for 1 month. The test substance added in water provide 20 to 60 hours photo period daily. Then water replaced daily for maintain the oxygen level in water and observe mortality and behavioral changes. Then the purpose of pesticide and pest control law and regulation to provide proper, safe and efficient use of pesticides in food production, protect the environment from harmful pesticides, to assure the agriculture and pest control workers have safe working, to assure the applicator, consumers and other users that pesticides they use are properly labeled, encourage the development and the implementation of integrated pest management system. Then moving to the conclusion and future perspectives, efforts and research should also focus on how to integrate biopesticide into integrated pest management strategy effectively. There is a need to sensitize and expose uniformed farmers, especially in developing countries, about the significance of biopesticides as a sustainable solution that could mitigate most of the adverse effect experienced with some synthetic pesticides. Regulation that promote the registration of biopesticides can also enhance commercialization and market availability. Then thank you for all listening my presentation.
Thank you, Ashwini, for your presentation. Now the floor is open for question answer.
Thank you, Ashwini. So it's nice presentation. It was an extensive review. So we have covered almost all the topics and all related things. So
Thank you, sir.
hope you will be able to do some wet lab data also in future. Yeah. All
Okay,
the
thank
best,
you, sir.
Ashwini. Yeah.
Okay, thank you.
Ashwini, good work, but little more. Try to improve your little more in the way you prepare the presentations. That's it. Okay. So thank you.
Thank you, ma'am.
Thank you, Dr. Vinod Kumar and Dr. Sharil. Edna, any more absentees to call again?
I'll just call them, the last
Yeah,
call.
okay. Okay. Okay.
OPSC 11, Fatima Dilkush. Okay, she's absent. And OPSC 18, S. Kirtana. She's also absent. Okay, so the presentations are over for the student category.
Okay.
Smita ma'am, please take over.
Okay, Edna. Thank you. With this, we are winding up contest paper on student category. Thank you, Dr. Sharil and Dr. Vinod Kumar, for your valuable suggestions to the participants. Thank you, all the participants. You all have done wonderful presentations. Now we are going to presentation only category for researcher, faculty or student. And chairs for the session, Dr. Sumi, Associate Professor, Department of Microbiology, Sri Shankara College, Kaladi, and Dr. Sudha K, Assistant Professor, St. Peter's College, Kolancheri. Hopefully by 3 p.m. Now it is over now, 3 p.m. over. We will start this session also. I think Dr. Sudha ma'am and Dr. Sumi, they are here. Shabana ma'am, Dr. Sumi and Dr. Sudha.
Yeah, I am
Yes.
here. I am Dr.
Yes,
Sudha.
they are there.
They are there now. Okay.
Yes, ma'am. Yes, ma'am.
Hello, ma'am.
Yeah.
Hello.
You can start with.
Sumi
Shall I call
ma'am,
the first
Yeah,
participant?
Sharin. Yeah,
Sumi
Sharin.
ma'am,
you can
your
start.
voice, can you?
Audible or not? Whether we are
Yes, it is very feeble. Sumi ma'am, your voice is very feeble.
Okay.
How about mine? Am I audible?
Yes, Sudha ma'am is
Yeah,
audible.
Sudha ma'am.
Yeah, anyway. Sumi ma'am, you can start anyway.
Sumi ma'am, I think you can use earphones
Okay.
because your voice is feeble.
Now,
Good
how is it
afternoon,
now?
Tony and Sudha.
Yeah,
Good
it's fine.
afternoon, Sudha, and welcome you to this Bio-Inventiyon 2020.
Yeah, thank you so much, Jisha ma'am. Thank you for inviting us as our chairs. We have 17 presentations to go. I think we can go ahead. Sumi ma'am, you can go ahead.
We can go ahead. Just for inviting us for this.
Okay, Etna, you can start. You can call upon.
Sharin.
Okay, ma'am.
Sharon will
Yeah,
be
yeah.
handling.
Yeah. Sharin,
Okay.
you can call Sharin.
Our first participant is OPF1, Dr. K. Shivasubramani. Sir, are you there? OPF1, Dr. K. Shivasubramani. I think he's not there in the room.
He's not there, Sharin. Then you can move to the next.
Okay, the next participant, OPF2, Nandita CK.
Hello.
Nandita, are you there?
Hello, ma'am.
Yes, Nandita.
Can you hear?
Yes, you're audible. Can you share your slide?
Yeah.
Sharon, it will be good if you announce the instructions.
Hello.
Okay.
Hello. Is it visible?
Yeah, yes.
It is.
Visible.
Yes, it is.
Okay.
Nandita,
Okay.
can you hold on for a minute?
Okay. Yeah. Sure.
Okay. There are a few instructions for the participants. You are requested to present your presentation within 8 minutes. as this is not a contest and we're running out of time, so it's better we can present it soon. And Nandita, you can start with your presentation.
Okay. Good afternoon, everyone. I'm Nandita CK, working at Nano Drug Delivery Systems Laboratory at Rajiv Gandhi Center for Biotechnology. I'm presenting a part of my work titled Antimicrobial Peptide Incorporated Electrospun Nanofiber Scaffold for Wound Healing Application. Wounds are a common type of injury that can occur in our day-to-day life, but in some cases, the wounds fail to heal or healing is prolonged. Such hard to heal or difficult to heal wounds are called chronic wounds. Unlike normal wounds, chronic wounds fail to proceed through an orderly and timely reparative process, namely hemostasis, inflammation, proliferation, and remodeling stages. One of the major examples of chronic wound is diabetic foot ulcer, which occurs in up to 50% of all individuals with diabetes. The prevalence of chronic wound in India is approximately 5 per thousand of the population. One of the common complications associated with chronic wounds is wound infection. Most infected wounds are caused by bacterial colonization. Staphylococcus aureus and Pseudomonas aeruginosa are the most frequent pathogens isolated from chronic wounds. Wound infection also triggers the body's immune response, causing inflammation, tissue damage, and thus slowing the healing process. Also, it can cause significant pain, discomfort, and psychological detriment for the patient. Systemic complications include cellulitis, osteomyelitis, septicemia. The most common treatment is the use of topical antibiotics, but increasing bacterial resistance to topical antibiotics has become a major issue. Also, traditional dressing materials like cotton, gauze, and other dermal grafts does not effectively. Hello? Hello?
Yeah, yes.
Hello? Is it audible?
Yes, Nandita, it is so.
Oh, okay. Okay.
Continue.
Other dermal grafts does not effectively help in proper healing of chronic wounds and address other major issues for a complete skin regeneration. The solution to this problem is the use of regenerative medicine technology, that is the use of three-dimensional scaffold that can mimic the natural extracellular matrix, also incorporating bioactive materials like peptides, proteins, small molecules to the scaffold for enhanced wound healing activity. Antimicrobial peptides are currently being widely used as a bioactive agent for various applications. Antimicrobial peptides are naturally occurring endogenous peptides, which are either expressed or induced to provide a fast and effective means of defense against pathogens. They are normally oligopeptides composed of varying lengths and are present in linear or cyclic arrangement. They generally constitute of L-amino acids and also are observed by the formation of secondary structures like alpha helices, beta sheets, etc. These molecules are cationic, hydrophobic, or amphipathic in nature. They exhibit diverse biological activities against gram-positive and negative bacteria, viruses, fungi, protozoa, and even tumors. The antimicrobial peptides also help in wound healing application due to their various activities like immunomodulation, where the AMPs help to regulate the levels of pro-inflammatory and anti-inflammatory cytokines. Cell proliferation, angiogenesis, cell migration are other activities of the antimicrobial peptide, which helps in complete wound healing. This figure shows that the antimicrobial activity of AMPs. The positive charge particles interact with the bacterial membrane, forming secondary structures and disrupt the bacterial membrane by forming pores. For the present work, we have selected LLKKK18 peptide. This peptide was synthesized using solid phase synthesis and characterized by MALDI. The peptide is an 80 amino acid sequence. Various studies have shown that LLKK18 inhibits the growth of E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, Candida albicans, etc. Also, it is shown to reduce oxidative stress, inflammation, and stimulate angiogenesis. These features of the peptide makes it suitable as a suitable wound healing agent. But the topical application of peptide as such to this wound site is not a suitable option because the peptides can be degraded easily. For the effective delivery of the peptide, we have synthesized electrospun nanofiber scaffold as a carrier. The major features of electrospun nanofiber scaffolds are they mimic the fibrous nature of the natural extracellular matrix. They promote improved cell adhesion, proliferation, and migration. Now, they also provide great tensile strength and also promote increased gaseous exchange and water absorption. And it's also a suitable drug delivery agent. To obtain nanofibrous scaffold that would match the required properties of wound healing materials, selection of the appropriate polymer is very important. So for this study, we have developed an electrospun PHBV PLA chitosan nanofibrous scaffold as a medium for delivering LLKKK18 peptide. The PHBV is a biodegradable and biocompatible polyester formed by bacterial synthesis and is a promising scaffolding material for tissue engineering application. PLLA is synthesized from the polymerization of L form of lactic acid of PLA. It is also biodegradable and characterized by high tensile strength. Chitosan is a well-known potent wound healing biomaterial and is widely used to enhance skin wound healing process. Chitosan also confers mechanical strength, anti-inflammatory properties, excellent biocompatibility, and hydrophilic nature of the fiber. The utilization of PHBV along with PLA and chitosan improves the biocompatibility, metability of the nanofibrous scaffold. So this polymer blend is well suited for electrospinning and as a suitable carrier system for peptide delivery. So we synthesize the nanofibers using electrospinning technique. The PHP, PLLA, chitosan blended solution was used in 2:1:1 ratio, and the peptide was mixed with the polymer solution to form fine nanofibers. The characterization of the nanofiber was done using Fourier transform infrared spectroscopy and scanning electron microscopy. The FTIR shows the functional groups of PHP, PLLA, and chitosan. The spectrum of the nanofiber shows the combination of individual polymer spectrum. The scanning electron microscopy shows fine nanofibrous scaffold, and the average size of the single nanofiber was found to be approximately 300 nanometers. We also did in vitro study of the antimicrobial peptide incorporated nanofiber scaffold in head cat cell line, which is the human keratinocyte cell line. MTT assay shows that the peptide and the peptide carrying scaffold has greater cell viability percentage when compared to the control. We also did scratch wound healing assay in head cat cells. As we can see, the peptide carrying nanofiber shows much greater wound closure than the control. The in vitro test result shows the potential of the developed peptide carrying scaffold as a suitable wound dressing. In summary, the antimicrobial peptide LLKKK18 carrying PHBV, PLA, chitosan nanofibers was synthesized using electrospinning technique. The developed scaffold helps in cell proliferation, migration, angiogenesis, and also antimicrobial activity, which makes it suitable wound healing agent for chronic wounds. We are doing in vivo study also. I acknowledge my guide, Dr. G.S. Vinod Kumar, Director RGCP, and thank you all for listening.
Thank you, Nandita, for your presentation. Over to you, Dr. Sudha and Dr. Sumi, for any valuable suggestions.
The presentation was very nice.
Hello. Thank you, ma'am.
Very audible. And actually, I have a doubt because you explained that the formation of this nanofiber scaffold. So how do you mean the way of application and the further here said in the summary part that the application part. So whether you have done any work related to that application part or something like that.
Actually, we have synthesized this scaffold material as a dressing for wound healing application. We have done only the in vitro works, but the in vivo works we are carrying out in animal diabetic mice model.
And how this the antimicrobial peptide is being incorporated?
Yes, the antimicrobial peptide was blended with the electrospinning solution and then electrospun to form the nanofibers.
Okay, so how is the stability? Whether you have done any work to test the stability of this peptide, this scaffold and peptide? inhibitor peptide in this scaffold?
No, ma'am, we are checking this. Actually more works are going on this in this.
Then
This is
only that
also
wound healing part will be okay.
yes, actually I have done only the preliminary test for this.
So that was only my query.
Good work, Nandita. I too have a question in relation to Sumi only. Like the concentration you have taken is 50 milligram per ml methanol extract.
Yes. Okay. It was dissolved. The peptide was dissolved in methanol for electrospinning purpose only.
Okay, so it's a quantified. That quantity of the microbial peptide is quantified.
Yes, yes.
Good. I wonder how that will look like. It will look like a gauze. It's very.
Which, ma'am? This fiber.
Yes.
This it will look like a mat, like a plain white mat. It will look like.
So
it's like gauze only. It's like gauze only.
On the wound, you can keep it like a gauze.
Yes, we can keep it like that.
Gauze and okay. I have one more question to you. Like, what is the role of this antimicrobial peptides? Like, how is it different from antibiotics and bacteriocides? Why didn't it choose that specifically?
Actually, antimicrobial peptides are shown to be more potent than antibiotics because in antibiotics there is a chance for resistance, but in this antimicrobial peptides, the chance for resistance is, though there is a chance, but the chance is very less. It disrupts the bacteria, so that thereby killing the bacteria.
What is actually the role of this antimicrobial peptides in bacteria?
This in bacteria, they interact with the membrane of the bacteria and form spores. So the bacterial membrane is disrupted, and also they show various intracellular activities. Also, it's mainly for the destruction.
Autolysis.
Yes.
Okay. Anyway, it's a very good presentation. I think we can go for some audience questions also if they have any doubt.
Okay.
Yeah, there is a question in the chat box. Can antibody microbial peptides inhibit biofilm formation?
Yes, antimicrobial peptides are known to inhibit biofilm formation. Various antimicrobial peptides are there. The activity of antibiofilm activity of this peptide we have not checked.
Okay.
Okay, I think we can wind up for this presentation.
Okay.
Thank you,
Dr.
Okay, thank you. Thank you.
Thank you, Dr. Sumi and Dr. Sudha. Now we will move to the next participant.
Okay, ma'am. So our next participant is OPF3 Murshida P. OPF3 Murshida
ma'am. Yes, ma'am.
Can you please share your slide?
Okay.
I would like to repeat the same instructions again. Please wind up your presentation within the 8th minute and you can conclude in the remaining 1 minute.
Hello, 1 minute.
Murshida, are you able to share your screen?
Yes.
Murshida, we are waiting for you.
Hello.
Murshida.
Is it seen?
No, not yet. The screen is not visible. Do you require some more time to present? We can move on with the next.
Yes, I want some time.
Okay, if you're not able to share your screen, you can send us your PPT. We can share the
Okay.
screen to you later.
Okay.
Okay, Murshida. Dear respected chairpersons, please note OPF3 Murshida will be presenting her presentation later. We're moving on to the next presenter, OPF4, Dr. Anne Mary Jacob. OPF4, Dr. Anne Mary Jacob. Dr. Anne, are you there? I think even she is not there in the.
Not there, Shirin. Anne is also not there.
Chairpersons, please note OPF4 is also not present. We move on to the next, OPF5, Nida MU.
She is also not there, Shirin.
Okay, ma'am. Okay, then we'll move on to the next participant, OPF6, Anushri TE.
Yeah, she is there. Anushri T, please respond.
Anushri, are you there? Anushri, are you there?
Yeah, her mic is on. I think some connectivity problem or something like that. Anushri, are you audible?
Even she is not responding.
Yeah. Then Sharon, move to next.
Ma'am, I think we should call the next participant later. That is OPF7. She won't be available now in this time. Dear respected chairpersons, please note, we'll be calling the next participant OPF8, Dr. Indu C. Nair. Ma'am, are you there?
Yes. Am I audible?
Yes, ma'am.
Yes,
You are
ma'am. Yes.
audible.
Okay. Thank you. Can I
Ma'am, can you
start?
please share your screen?
Okay.
Ma'am, are you able to share your screen? Yes, ma'am. Now it's visible. Ma'am, can you change it to slideshow mode?
നീ എവിടെ നിന്നോട് ചോദിക്കുന്നത്? എനിക്ക് ഓടിയായിരുന്നു.
മിസ്സിനോട് പറയാം. അപ്പൊ മിസ്സിന് അറിയണ്ടോ സൂമിന്റെ എങ്ങനെയാണെന്നെ?
എടാ എനിക്ക് ഓഡിയോ കിട്ടുന്നില്ല. എന്താ?
നീ പറയുന്നുണ്ട്, അവര് കേൾക്കുന്നില്ല.
Participants, please mute your mics.
Hello, am I audible now?
Yes, ma'am, you're audible.
Can I start?
Yes, ma'am, you may start.
Good afternoon, one and all. Good afternoon, respected chairs.
Mute ആണോ unmute ആണോ?
First, can you hear me? Am I audible?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Yes, in the teacher. I can hear you very well.
First, I thank Dr. M.S.
ഓൾക്ക്
Jisha,
presentation
Professor
കിട്ടാൻ
and Director, Institute of Plant Science, Mahatma Gandhi University, Dr. Asha and Dr. Shahana Moore for giving me an opportunity to present a topic here. Today, I would like to present a general topic, that is
mute.
role of aspartic proteases, the catalytic role of aspartic proteases in food industry. As we all know, enzymes are wonderful catalytic machineries. They drive all the biochemical reactions in the living world. And again, they have many interesting properties to which we are familiar with. They're highly specific, proteinaceous in nature. And the last two properties are important in industrial biotechnology and microbiology. That is, as they are proteins, they can be engineered and they are green. Green means they not at all harm the environment. They are completely biodegradable. So they can be considered as industrial biocatalysts, which are very safe. From time immemorial, enzymes have been a part of food preparation. In age-old processes, enzymes were not directly applied. In the sense, they are not isolated and purified, but microbial cultures, plant or animal extracts, etc. were used.
Dr. Indu, your mic is mute. Please unmute. Dr. Indu, please unmute your mic.
Indu, it's not audible.
Yeah.
Yes,
okay.
ma'am. Now you're audible.
Yeah. Now audible. You can continue.
There are hundreds of enzymes isolated from different sources, which are successfully used as industrial catalysts. And coming to proteases, they also find their own role in industrial food preparation. Broadly, proteases are classified into endopeptidases and exopeptidases. And in endopeptidases, we get a group aspartic proteases, on which today we concentrate. Aspartic proteases, they are also important in baking, fruit juice clarification, meat tenderization, etc. But they are most popular in cheese making. And since they have an acidic pH is required for hydrolysis, they are known as acid proteases, and they have two important aspartic residues in their catalysis, So they are known as aspartic proteases. Coming to the role in cheese production, these are the standard steps in cheese production. And as the fifth step, we get rennet addition. And rennet is a group of enzymes. This is an inevitable step in cheese production. And rennet is a group of enzymes in which the main component is an aspartic protease. Now coming to the role of rennet in cheese making. The milk protein, milk is a dispersion of fat droplets in aqueous solution or in an aqueous medium, and also casein protein is there. The casein proteins are repelled due to steric hindrance. So a liquid consistency of milk is appeared. But when rennet is added, rennet hydrolyzes casein protein to produce pyrocasein and micropeptides. In presence of calcium, the pyrocasein and micropeptides aggregate together to form a gel, and a gel-like consistency is appeared, and this is the second stage of cheese production. So coming to the sources of rennet, chymosin, this is the traditional and most popular source, and this is also the best suited and safe one. And this is obtained from the calf stomach. The fourth cavity of neonate calf produce chymosin. The chief cells are responsible for the production of chymosin from calf stomach, and their main property is the limited hydrolysis of casein. And this can produce highly palatable cheese, which has huge demand all over the world. But the chymosin from calf stomach can meet the increasing requirements, so substitutes are to be there. And these substitutes are also traditionally used. There are plant rennets and microbial rennets. In plant rennets, mainly cardunculus is used. Different species are there. Some species of ficus is used. And microbial rennets are also there, mainly aspergillus and mucor species. And these microbial rennets are mostly now used for industrial rennet production. There are slight differences between the plant rennets and animal rennets. Plant rennets generally offers extensive hydrolysis. Because of this excessive hydrolysis, micropeptides are produced in larger amounts, which provides a bitter taste to the cheese. Because of this, the palatability is reduced and also acceptability is reduced. But microbial rennets, some are very much similar to chymosin. Because of this, today rennet production mainly concentrate on microbial fermentation. And also another popular method is recombinant chymosin production. Here the chymosin gene from calf is isolated and expressed in different systems. This is a standardized process, and this also offers high industrial production. Cluveromyces lactis, Aspergillus niger, E. coli, etc. are well standardized expression systems. And plant rennets are also subjected to recombinant and DNA technology experiments. Cardosin gene is expressed in Saccharomyces cerevisiae and Pichia pastoris, and aspartic protease from grasshopper was expressed in Pichia pastoris, and they are produced in plant cell suspension cultures. And another approach is protein engineering, and here mainly plant rennets are subjected to protein engineering, and the catalytic efficiency of them should be reduced. That is the main aim of protein engineering of plant aspartic proteases. Here cardosin A and B are subjected to protein engineering in modern studies, and in a report in 2020, this cardosin from Cyanara cardunculus is used for altering the properties where cardosin B was successful, while cardosin A was not experimentally successful. And these are the main points I highlighted. I selected this topic because of the high demand of the enzyme and the need for quality of food production, and also it promotes a green technology. Thank you all. Thank you.
Thank you, Dr. Indu. Now the floor is open for QA session. Over to you, Dr. Sumi and Dr. Sudha.
Indu, it was a general talk about this aspartic proteases. And I want to know whether what you have told about that genetically engineered microorganism, whether it is being used in industrial purpose nowadays.
Yes, yes, that is used for industrial purposes. Not, they are well expressed in chymosin. Gene is well expressed in the microorganisms.
So whether it is commercially used for cheese production.
Yes. Commercial products are also there. One is Chymostar, and that is produced by DuPont Biosciences.
So that's all, because it was a general, it was nice hearing you. Thank you.
Thank you.
Over to Dr. Sudha.
This was so simple and nice presentation.
Thank you, ma'am. Thank you.
That renin and rennet, the main difference is ren is the original which we collect from the abomasum of the calf, and the rennet is the alternate for that. Is it so? I'm not aware of it. That's why I'm asking.
Rennet is a collective name for all these microbial rennet. And then chymosin is especially the one specific member of this rennet.
Okay.
Renin or chymosin. Renin and chymosin.
Renin may be another name for chymosin. Okay.
Yes.
In continuation to Sumi ma'am's question, like whether it got permission to add in the food products, like whether it is regarded since it is genetically engineered.
Not genetically, it is produced through Saccharomyces cerevisiae or E. coli, etc. But they are not modified. The chymosin gene as such is produced through Saccharomyces cerevisiae. Expression system is only Saccharomyces or Cluveromyces.
Okay, no more questions. It's a wonderful presentation. I think some of the participants can ask, audience can ask questions regarding this.
Any question
I think
from participants?
there are no questions from the participants.
As there is no question in the chat box, can we move to the next participant, ma'am?
Thank you. Thank you.
Yeah, as there is no question in the chat box, we'll move to the next participant.
Move on to the
next
participant?
Next participant. Okay. We'll move to the next. Sharon? Sharon, are you there? Sharon? Sharon, are you there? Next, we will move to the next participant. That is OPF9, Alex John. OPF9, Alex John. Alex John, are you there? Alex? Alex, are you ready?
Hello?
Yeah, Alex.
Yes, madam.
Yeah. Share your screen. You can share the screen now.
Hello.
Yeah, you can share the screen.
Yes,
Share
nice
your
one.
screen here.
Madam,
Yeah.
it's okay.
Yeah, it's okay. You can start.
Good evening, everybody. Myself, Alex John. My topic is development of native bacteria antagonists for the management of Pythium rhizome root in ginger. As we know, ginger is herbaceous rice crop used for many medicines. In Kerala, production is more than 33,000 tons and area is 6,000 tons, more than 6,000 tons per hectare. In ginger cultivation, many diseases are there. In this, most destructive disease is rhizome rot, and the coastal organism is Pythium, that is Pythium muriatilum and Pythium affidermatum. Why we select this study? Because intensified agriculture production system farmers mostly depend on agrochemical for plant disease management. This goes to serious environment and health hazards. So the development of alternative strategies should be there. Then we go for native strains of biocontrol agents are more effective and have potential to control the plant disease than in introduced one. Objectives were isolation and characterization of bacterial strain from rhizosphere soil of ginger, and in vitro and in vivo screening of bacteria isolates against Pythium muriatilum. Soil samples collected from ginger rhizosphere from Wayanad, Kozhikode and Pathanamthitta district. Total 24 rhizosphere soils were collected. In this, 8 soil samples from forest area of Muthanya, Wayanad and Rani, Pathanamthitta. These are the photos. Soil sample analysis, total microflora quantitatively estimated by serial dilution plate techniques, 276 fungus and 166 actinomyces and 312 bacteria were isolated. Coming to a pathogenicity test for Pythium muriatilum, proved by coach specialist after 25 days of inoculation with the Pythium muriatilum, infection of leaves and delays almost 100%. Thus, proved the pathogenicity test. In vivo preliminary screening of isolated 312 rhizobacteria against rhizome not pathogen. We only took the bacteria culture only. By dual culture method with the double agar techniques, isolated 51 rhizobacteria shows zone of inhibition. These 51 bacteria isolated tested antagonism individually along with the reference culture of Kerala Agriculture University PGPR2. Infusion zone chart is there. In this, 8 bacteria isolates shown more than 60% inhibition. This 8 bacteria isolates in this BA51 and BA276 shows more than 78% inhibition. These are the inhibition antagonism pictures. Coming to the biochemical identification of promiscuity bacteria isolates, BA51 identified as, rotionally identified as Bacillus, BA147 and BA270 also Bacillus, and BA97, BA121 and BA154 is Pseudomonas species. Went through the mechanism: salicylic acid, pseudophore production, HCN production, IAA production, ammonia, nitrogen, volatile organic compound, and phosphorus solubilization. After the mechanism, went for molecular identification with 16 SR DNA techniques in Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. From the molecular identification, BA51 as Bacillus subtilis, BA97 as Pseudomonas, and BA121 as Pseudomonas, BA145 as Bacillus subtilis, and BA276 as Bacillus amyloliquefaciens. We only selected 2 bacteria strains for further studies. That is Bacillus subtilis strain BA51, Bacillus amyloliquefaciens strain BA276. Others were human pathogen or less inhibition zone. These 2 isolates for gone for in vivo studies with substrate as talcum powder. Inverse studies, pot culture experience was done with variety Mahima, that is IASR Codicode variety, design CRD, treatment stall, replication 10, and location ICR Pishujianaganam, Pathanamthitta. BA 51 as antagonist first, and BA 276 as antagonist 2. T1 is an absolute control, T2 is control. T3, T4, T5, T6 are with antagonist first and second. Seed treatment, drenching and spraying 5 times with pathogen and without pathogen. T7, T8 is Kerala Agriculture University Pseudomonas strain. T9, T10, T11, T12 are antagonists first and second with seed treatment only 4 times with and without pathogen. In this presentation, I took only 3 parameters: spotting percentage and disease incidence and yield. Spotting percentage, almost all treatments are same except T1 and T2. In the disease incidence, at 180 days after planting, T8 shows low in disease incidence. After that, T2, T4, and T6. In yield, gram per plant, pot culture, T8 is shown highest yield. T4 and T6 are the second and third. These are pot culture experience pictures. Then, based on the pot culture study, gone for field experience. In this variety, Mahima IASR variety, same as pot culture, design RBD, treatment 8, replication 5, location same, Pathanamthitta. And antagonist is 51, BA 51, and antagonist second is BA 276. The T1 and T1 is absolute control, T2 is control, T3, T4, T5, T6 are with the antagonists first and second with seed treatment, drenching with the drenching and spraying 5 times with and without pathogen. T7 and T8 are KU Pseudomonas culture. Here also 3 parameters take: spotting percentage, disease incidence, and yield 10 per hectare. In spotting percentage, almost all are same except T1 and T2. 180 days after planting, disease incidence is low in T8. After that, T4 and T6 respectively. Yield also the same as the disease incidence. That is, the highest yield was in T8 treatment 21.38, and in T4 19 ton per hectare, and T6 is also 19 tons per hectare. Field experience photos pictures are here. Conclusion: potential antagonist bacteria BA51 and BA270 were isolated from rhizosphere of Singer. Application of potential antagonist BA51 and BA270 improve the plant growth characteristics, disease suppression, and help in better establishment of plants. Antagonist BA51 and BA270 is in par with Kerala Agriculture University isolates and best alternative for biocontrol agent for Pythium rhizome root in ginger. Thank you, madam. Thank you.
Thank you, Alex John, for your presentation. Now the floor is open for question and answer session. Over to you, ma'am. Dr. Sumi, Dr. Sudha.
Next, hello.
Hello.
You choose 3 parameters, no?
Yes, ma'am. Spotting percentage, disease incidence, and yield for this presentation only.
Okay. So, what was the significance of your study? Means whether the sprouting percentage or what was the significance?
Good significant difference in all other treatments than this BA51 and BA278. They shows the spotting percentage good, disease incidence good, and the yield also. Shows high the CD values also here, madam.
So,
significant difference is high.
Yeah. So it was the disease that does its antagonistic activity. You mean by yield means the fruiting yield.
Yes, madam. Isomild.
Okay, okay. So did you try a consortia of these two microorganisms?
No, I have, madam.
So because these two are working very well, so you could have also checked in that way also.
Okay,
If it is a consortia, it may be working much more better than a single organism. Because it is
madam, please.
Pardon? Tell me.
BA51 is Bacillus subtilis and BA276 Bacillus amyloliquefaciens.
So both are Bacillus species, no?
Yes, ma'am. Yes, ma'am.
Okay. So whether means these two preparation you have whether it is a commercial, you have made a commercial product or not?
No, madam. I just finished the field studies, madam.
Okay, so it was done using the application. The mode of application was talcum powder only, no?
Pardon, madam?
The mode of application. How did
talcum
you
powder.
Talcum powder. Okay, so means how many times the application did you do? This talcum powder.
Talcum powder in treatment. In treatments, 5 applications. Seed treatment is first, then 25 days intervals, 5 applications.
Okay, so it was it gave the result according to the Kerala Agriculture University preparation.
Madam.
Okay, nice presentation. So
thank you. Thank you.
You have included this parameter. So nice presentation.
Thank you.
Over to
Alex John, this presentation.
Madam.
And the extensive research you have done.
Thank you. Thank you.
My question is about the Pythium rot you said, no? Is it a common disease for this senior? How prevalent it is in?
There are many. There are many. Can you see the slide, ma'am?
Yes, I have seen that dried leaves and all. Yes, that one.
There are many. 1 minute. There are many diseases affecting in ginger cultivation: rhizome rot, basal rot, wilt, thread blight, leaf blight, and all. Rhizome rot is the most destructive one. 50% destruction is happening due to this. The cultural organism is basically Pythium. That is Pythium myrtille and Pythium affiniromata is the most destructive ones.
Okay. Pythium. So Pythium will be spreading through the air. Where is it affected? Like a rhizome
by soil-borne and seed-borne, madam.
Okay. So in vivo studies means what exactly it is? It's not clear for me.
In vivo study, madam?
In vivo studies, you said in vivo experiments, no? What does it really mean?
I am not included in this presentation. Okay. One towel method is there for the for this disease incidence. Roll towel method. That was also done in my in my study, but in this presentation, I am not included because of the time limitation.
Okay. So you are in field experiments. You are spraying it as well as you are incorporating it with the seedling, and two types of mode of application is there.
Seed treatment before sowing, then spraying and drenching.
And with the control, you are control, you are establishing that these two organisms have major impact on subsiding the effect of Pythium.
Yes, madam. Yes, madam. Yes, madam.
Okay. So, why didn't you conduct any sterile type of experiments, like in-house experiments, to prove the exact effect of these organisms? In the other case, we can't say that since it's not sterile, many other organisms are there.
Also there. There are many other diseases also there. Insects are also there. Mainly this stem borer.
So, photobiotic experiments are there with sterile substrate, you can prove that these two organisms are very good. Since you have done extensively on this organism characterizing etc. for production and all the other productions, no, you should have included that also.
In pot culture study, I did the soil sterilization with formaldehyde. After that, the pot culture study started.
Okay. Okay, one more suggestion I have is like you used a rhizosphere organism, no?
No,
And
madam.
you should have used the microorganism, antagonistic microorganism isolated from the leaf surface, like a plane organisms also. Since you are applying it in that mode, there may be antagonistic organism you can select or detect from the leaf blade also, no? Is there any possibility like that? It's a suggestion actually.
No. Okay, madam. Okay.
Okay, I think that I'm done with. If any questions from the participant sides, you can take it.
Thank you, Dr. Sumi and Dr. Sudha. There is no question in the chat box. So we will move to the next participant.
Thank you, madam. Thank you.
Thank you.
Okay, ma'am. Our next participant is OPF 10, Ragida VM. OPF 10, Ragida VM. Ragida, are you there? Ragida, are you there?
Yeah, not here.
No, ma'am, she's there here with the screen name HP.
Oh, okay.
Ragida, can you increase the volume? We can't hear you. Your mic is unmute, but we can't hear you.
Ma'am, are you able to hear me now?
Yeah. Now you're audible. Can you please share your screen? Yes, just change it to the slideshow mode. Yeah, fine. You can start your presentation, Ragida. Ragida, we can't hear you.
Are you able to hear me now?
Yeah, now you're audible.
Okay, so I am Ragida, Junior Research Fellow in JNTBPRA, Palo. I would like to thank the organizers of Bio-Inventiyon for giving me a chance to present my research work here. Today I am going to present a small piece of my research work titled as Screening of Solvents for Studying Antidiarrheal Effect of Plant Extracts or Compounds in Mixing Chamber. So solvent can be defined as any substance which can dissolve other substance known as solutes based on their polarity. So there are certain criteria for a solvent which can be called as an ideal solvent, which includes its ability to dissolve solutes, its poor ability to dissolve impurities, it should be non-flammable, and it should be non-toxic. But except water, other solvents do not follow the features of an ideal solvent. Based on the polarity, solvents can be divided into non-polar and polar solvents. So non-polar and organic solvents, but actually produce toxic effect on living system. So if we select a solvent to study in a biological system, we should be very careful to select that solvent, because otherwise it will produce its effect on experimental procedures, which will interfere our studies. So an ideal solvent which can be used to use in a biological system, it should have minimum effect on our experimental model. So these are commonly used solvents which are used in biological system, among which ethanol and dimethyl sulfoxide, that is DMSO, is very commonly used in literature also. So what we have seen here is that ethanol produces its maximum toxicity at 1%, and dimethyl sulfoxide produce its maximum toxicity at 1.5% on vertebral embryo cells. But literature reports also suggest that even at 0.1% concentration, these solvents produce toxicity on live cells. So for giving a brief background, our primary target or our research work target on developing an antidiarrheal formulation based on botanicals. So we study the antidiarrheal effect of plant extracts. So diarrhea, we are primarily target diarrheal treatment. So diarrhea is 3 or more loose stools per day. During secretory diarrhea, it is actually the secretion of anions from intestinal tissue, mainly chloride and bicarbonate. So in our cells, the secretion of these anions are regulated by second messengers like cyclic AMP, cyclic GMP, and calcium. During diarrheal infection, the toxins produced from enteric pathogen are actually increasing these secretagogues, and which will further enhance the anion secretion from intestinal cells. Following these anions, water will move out of the cell and produce diarrhea. So in order to study the antidiarrheal effect of plant extract, first we establish a diarrheal model in Using chamber. For that, we use the phoscolin. It is a diterpenoid which is produced from Coleus phoscoli, and which will actually enhance the secretagogue, that is cyclic AMP in the cell, and thereby we can produce an in vitro diarrheal model in Using chamber. So then we studied the active ingredients from plants to inhibit anion secretion and to improve barrier function. So one of the limiting factors to use plant extract is their solubility and bioavailability, which limits its usage as a drug molecule or study their effect. The solvent we use will also influence the solubility and bioavailability of plant extract. So our objective is identify the most suitable solvent which will produce minimum effect on anion secretion and barrier function. Barrier function means we can measure conductance or the barrier membrane integrity in our instrument, that is called Using chamber. So I will explain it in later slides. So we have to select a solvent which has minimum effect on basal anion current and conductance. And also we are establishing a diarrheal model using phoscolin. So our solvent should not interfere with phoscolin stimulated anion current. That is our objective. So methodology, all studies were done using cis-albino male mice of 8 weeks old. So based on literature, we selected a series of solvents and we studied their effect on basal anion current and conductance. Conductance means its barrier function. So what we found out that DMS on ethanol were superior to all other solvent studies. So we selected those solvents for further study. Then we studied, we select an arbitrary concentration, that is 10 microliter per 5 ml concentration of DMSO and ethanol. We select this concentration because literature says that even 0.1% concentration is toxic. So we just take a little higher concentration based on literature, and it is 0.2%. Then we study its effect on basal anion current and conductance. And also we studied its effect on phoscolin-induced anion secretion. So after that, we have to find out what is the minimum and maximum concentration of these solvents to produce its effect on anion current and conductance. So we did saturation kinetic study by adding increasing concentration of these solvents into the system. So this is the instrument that we mentioned, that is Using chamber. It is actually developed by Hans Sussing to study the transport across Frogs membrane. And now it is a sophisticated instrument used by physiologists to study the ions, nutrients, and drugs across various epithelial tissues. So what we have done is that we will take out the intestine from mice and we will mount it into Lucy's chamber, and then that tissues were bathed in Ringer solution, which has pH 7.4 and osmolarity 292.5, which is similar to our intracellular osmolarity, and which will be bubbled continuously with carbogen. It is a combination of 95% oxygen and 5% carbon dioxide, and all the system will be maintained at 37 degrees Celsius. Thus, we create a living environment for the tissues during the experimental time. Then we the tissues were voltage clamped to 0 for studying the transepithelial current, which is basically the anion current. And we studied the conductance or membrane integrity by using Ohm's law by this instrument. So we found out that at 10 microliter
Ragida, we can't hear you.
Hello, are you able to hear me now?
Yeah, yes. You can
For
continue.
DMS,
Ragida, please check your audio. We can't hear you.
are you able to hear me now? Hello.
Yes, you're now audible.
Okay. So, but ethanol did not increase short circuit current compared to control group. So, for basal anion current studies, we can select ethanol as a suitable solvent. So, we did the experiment using the same concentration, that is 10 microliter per 5 ml in presence of phoscolin. And we found out that DMSO actually decreased the phoscolin-induced anion secretion. So, we cannot use DMSO in our system. So, but ethanol did not significantly change the phoscolin-induced anion secretion compared to control. Thus, ethanol can be further used in presence of phoscolin also. So, we did saturation kinetic studies and we find out that Km, which is the minimum concentration to produce half maximum velocity, and for DMSO it is 0.9 microliter and for ethanol it is 1.9. So it is visible that even at higher concentration, almost 2 times concentration than that of DMSO, ethanol produces only 52.2% increase in short circuit current. We did the same study to find out the effect on conductance, and we find out that DMSO further increased conductance compared to control group, even though ethanol increased conductance, but it is not significantly higher than that of control group. Then we did the study, saturation kinetic studies for conductance and find out that 0.7 microliter was for DMSO, but higher concentration can be used for ethanol for 2.3 microliter. So the minimum concentration that we can use for DMSO is only 0.2 microliter, that is 0.004%. But for ethanol, we can use up to 0.4 microliter. Conclusion is that even under basal current and also in the presence of phoscolin, ethanol is a better solvent than that of DMSO. And the maximum short circuit current was induced by 1.8 microliter by DMSO and for 3.8 microliter for ethanol. The maximum effect on conductance was observed with 1.4 for DMSO and 4.6 for DMSO. The minimum concentration of ethanol with least effect on ISC and G was observed as 0.4 microliter. So our study confirmed that ethanol is superior to DMSO in vitro Within chamber studies measuring anion current and conductance. I acknowledge Dr. Vidya Sagar, Dr. Anastas, Dr. Shivraj, and Dr. N.S. Pradeep for their valuable guidance and support, and Ms. Savita S.K., Ms. Harikrishnan, and Dr. Lakshmi K. Edison, lab member, and also funding source SRS Nutrition, KCST, JNT, BGRI, KCST for providing fellowship, and Srijitra Edan Institute of Medical Science, Pujapura. Thank you.
Thank you, Ragita. Participants, you can type your questions in the chat box, if any, regarding the presentations. Over to you, Dr. Sudha ma'am and Dr. Sumi.
Hi, ma'am.
Hi, Ragita. It's a very good presentation.
Thank
Dr.
you,
Sumi, you can start with.
No, first. You can also start first. No problem.
It was a very nice presentation, like that. That Asing's chamber and all is a very interesting thing. What I understood is you are optimizing that solvents for plant extracts for diarrheal disease.
Yeah, in Within chamber.
Okay, using chamber. It would be good if you could explain that using chambers functioning one more time. The slide.
That slide. Sure, ma'am.
And you explain what kind of tissue you are taking, whether it's intestinal tissue.
Yeah, we are taking intestinal tissues from mice. And are you able to hear the screen, ma'am?
Yes.
So this is Within chamber. It is a physiological instrument. So these are 2 chambers which will be filled with a Ringer solution of having pH 7.4 and osmolarity 292.5. It is similar to our biological system. We can mount the intestinal tissue in this slide after opening the intestine. Then we can separate its mucosal side and serosal side.
Okay. So only the piece of the intestinal tissue you are taking out from the mice, or
Yeah, ma'am.
it's a dead tissue or a live tissue? That's my question.
It is actually live tissue we are maintaining in living condition. That
By
is why
using
we are
that isotonic solution, you are keeping
yeah.
it in icon, but it is detached from the mice.
We are detaching. It is in vitro study. We will open the intestine and we can take the study in whole intestine because we have 8 chambers in this system. So from jejunum to ileum, we can use all the tissues.
Okay. So even if it is dead, the membrane potential and everything will be maintained in this.
Actually, it is not. We cannot say that it is in dead condition because up to 2 hours of study we can maintain in live condition. After that, we can understand whether it is live or dead by measuring the conductance. So conductance, it is actually membrane integrity. So in dead cells, the membrane integrity will be lost and the conductance will increase. Okay. Whether our study is in living system or not by measuring the conductance.
Okay. Your focus is not clear for me. Like what kind of diarrheal disease you are studying on? Like how did you induce the diarrhea in mice, and what is it, bacterial diarrheal disease?
Are you saying about in mice?
Yes. The focus of your work is not clear. Like whether you are studying particularly for extracting plant for treating diarrheal disease, then like how you induce the diarrhea in it. Like whether you are focusing on cholera protein, like that kind of thing.
Okay, ma'am. Diarrhea actually induced in our body because of increased secretagogue in the intracellular compartment. That is like cyclic AMP, cyclic GMP, and calcium. So diarrhea can be induced by pathogens like Vibrio cholera, Shigella, and rotavirus, etc. But each organism produce different intracellular messengers. For example, Vibrio cholera.
Not 12% single paper.
So in your studies,
Hello.
so in your studies,
ma'am, in
how
our
did you induce
study,
the diarrhea?
So for Vibrio cholerae, cyclic AMP will be increased.
Ma'am, 1 minute.
Calcium will increase.
Ma'am, 1 minute. Participants, please mute your audio. Okay, ma'am, continue.
Vibrio cholerae.
Cholera toxin.
Cyclic AMP.
Cyclic AMP pump disruption in
cyclic GMP and rotavirus induced calcium. So the difference is in the secretagogue. So currently we are studying using cyclic AMP, that is Vibrio cholera producing diarrhea.
Okay. Then I wonder what will be the control normal? Like again, you are not audible properly.
Are you able to hear me now?
Now it's clear, but still with some disturbances.
Okay, ma'am. Actually, in Usin chamber, we will take intestine from healthy mice. Okay, then we will mount the tissue in the Usin chamber. After stabilizing the tissue, because we are taking the intestine through a painful condition. So the tissues will be stressed some condition and it will viable in new environment. So that we will give some time for the tissues, and after 30 to 45 minutes, the short circuit current and conductance will be stabilized in the chamber. After that, we will induce diarrhea using forskolin, 10 micromolar forskolin, which will increase cyclic AMP. Then there will be a sudden increase in anion current, and we will use our plant extract after stabilizing the forskolin induced current. Then we can know that whether that extract is able to decrease that increased anion current because of forskolin. Then we will select that extract. Then we will do studies using loop assay. For that, we will take the intestine of the mice and we will put that extract in the presence of forskolin and using healthy mice as control and also forskolin treated mice alone. Then we study the secretory.
You're not audible now.
I can hear her, but with some background noise.
Your voice is breaking.
Hello? Yeah, earphone issue, I think. Some disturbances are coming. Yeah, we are hearing you.
Okay, so after that, we will look into Western studies.
Now not audible.
Hear me now, ma'am.
Hello.
Hello.
Yeah, we can't hear you. Still disturbances are there.
Hello.
Yes, now we can hear you.
Okay, so we will study the secretory channels like CFTR and anoctomy. Through that channels, actually chloride and bicarbonate is secreted into the intestine. Then we
Okay,
will
now
select
it
that
makes sense to me, like it's clear.
Yeah.
And I have one more question, like you are planning to use plant extracts, no?
Yeah.
So plant content, that phytochemicals in the plant is different. So
Yeah.
what is the purpose of optimizing a particular solvent? Like, because for each plant there will be one suitable solvent system based on the phytochemical content.
Okay, fine. So we are preferred to water soluble. We are actually preferred to water insoluble compounds only in the case if we didn't get any water soluble extract. Then only we go for other non-polar components. So in our study, we confirmed that ethanol is most suitable. And for DMSO, we can't even use 0.2 microliter concentration. So what we are decided to move further is that we only use ethanol, and what are the compounds that dissolve in ethanol, we will further select, because bioavailability is an issue. Even though we completed our study in DMS and other non-polar solvents, we cannot use that in our biological system as a drug molecule. So we only concentrate more concentrated on water soluble extract, and just little move towards little non-polar compounds.
Okay, interesting work, Rakita. All the best
Thank you,
for
ma'am.
your studies.
Thank you, ma'am.
Go ahead.
Rakita, I have one based on Sudha's inquiry only. Means, there was no focus in your study, means presentation, because you didn't give importance to the plant extract. That one thing. Again, the importance was more towards the solvent preparation. So we were wondering whether you have, means you started with screening of anti-diarrheal plant extract, but not an extract plant was named in your presentation. So that you have to, because when you explained, we got the point. That's the thing.
Okay, ma'am.
Okay.
Actually, I can mention the plant, the name of the plant, because we are actually patenting, filing a patent based on our work. That is the reason. So I just present a peripheral study here.
Yeah. But you could have take all the concentration or like that. Now what we got was that you are using only the solvents. That ethanol, and there is no plant extract in your study. That you didn't mention anywhere. So that was the one confusion. Then you were telling about water soluble compounds, but you are taken ethanol, organic solvent. You were telling that you are for your studies for the water soluble compounds and all, but it is an aqueous extract, no? So,
Yes,
but
sir.
your study was mainly about the organic solvent. So mostly the organic solvent, that part only will be coming to your extract when you are treating. That's the one thing.
So our primary target was finding a suitable water soluble extract, but we are not sure we are able to find water soluble extract for both diarrhea and conductance for barrier function also. So currently available product in the market only target for diarrhea, not for barrier function. That is gut permeability. That is the reason for autoimmune disease like diabetes, thyroid, and so on. So we have to find out drug for diarrhea, and the other aspect is for conductance also. So for that we may have to use other non-polar compounds. That is why we are using ethanol in that case also.
It is like mostly the physiological work you have explained. So the presentation was good and new. This type of the physiological work was new for us. Okay, then all the best.
Thank you, ma'am.
All the best, Ragita, for your patent and all. Thank you.
Thank you, ma'am. Thank you very much. Shall I leave?
No. 1 second, Ragita. There is a question in the chat box. How do you add the solvent into that chamber? That is the question from Visha Harnibi.
Okay, we can directly add the both of these chambers using a
one more question. One more question is there. Why did you choose ethanol and DMSO as specific? In specific, why did?
Okay, we select that solvent specific because we did the same study, that is the preliminary study on anion current and conductance using different solvents. Compared to other solvents, these two solvents are better. That is why we further selected these solvents and studied and optimized the concentration of that solvent.
One more question, Ragita, whether you studied the toxicity study of using this extract for the normal tissue and the normal industry.
Using extract.
So the is toxic. Yeah.
Okay, actually we can study the toxicity of the extract in this instrument because if the extract is toxic to the cells, then the conductance will increase. So we can use this instrument for
We are not audible. We are not audible.
We can't hear you.
We'll move to the next one. Presentation.
Okay, ma'am. Thank you. Now we will move to the next participant. Sharon?
Yes, ma'am. So our next participant is OPF 11, Mir Khalid. OPF 11, are you there? Mir Khalid. He's there in the room. Mir Khalid, can you please unmute your mic?
Mir Khalid, are you there?
Yeah, he's there.
Hello, am I audible?
Okay,
yes.
Sorry, ma'am. Because of slow internet connectivity here in Jammu and Kashmir, so I was not able to get connected.
Okay, it's okay. You can share your screen.
Yeah. Okay. Ma'am, I'm here. I want to give you oral presentation of my work.
Yeah. Yes, we are waiting for it.
Am I visible as well as audible, ma'am?
Yes, ma'am. Yes, sir.
Okay. Thank you so much. Good evening to all the organizers and the presenters of Bio-Inventiyon 2020 on recent advances in biosciences.
So please share your screen, your PPT, please.
Ma'am, I have not presented PPT. I want to deliver it in oral form. Is it possible?
Chairpersons,
it is okay. Yeah, it is okay, Khalid Mir. Like if you have any questions or if we are not catching up, we'll interrupt you and ask. That's only the problem.
I thought because of 2G internet connectivity, I was not sure whether I will able to share my PPT or not. That's why I prepared it. I thought I will deliver it orally.
No issues. You can carry on. Start it.
Okay. Thank you, ma'am. Thank you.
Okay, so you can start your PPT presentation.
Okay, I'm highly thankful to the organizing team for providing me such a great platform to present my work over here. Ma'am, the topic of my presentation is a quantitative assessment of municipal solid waste management and public consciousness in Bemina, Srinagar of Jammu and Kashmir, as Srinagar is the summer capital of Jammu and Kashmir. So as we know that increasing population, industrialization, urbanization, and improved standard of living has resulted in increased solid waste generation. So management of these huge quantities of municipal solid waste has become a serious concern for authorities and public at large. Present study was conducted in Bimna area. It is highly developing area, or we can say that highly urbanizing area presently in Srinagar. So, of the city, which is located some 4 kilometers from the main Lal Chowk. So this area contains almost 60 colonies. So 8 colonies that were my study, that I included in as my study site, included Hamdaniya colony, Nundrish colony, Iqbalabad, Usmanabad, Tawhidabad, Alamdar colony, SDA colony, and Mominabad. So these were the colonies selected with almost 200 respondents from 200 households, having a sample size of 25 in each colony. So aim of my work study was aim of the study conducted was to determine the municipal solid waste generation from households in highly developing Bimna area locality of Srinagar city. Also to understand the public perception or general knowledge and their attitude towards the management process of these solid waste coming out directly from the homes. As we know that management is the collection, transportation, processing, and recycling or disposal of the waste generated. So the methodology which I followed, that was the survey was conducted in the months from August to October 2020 during pandemic lockdown. A random sampling population of 200 households was selected. A total of 200 survey questionnaires was the main thing which was done. A total of around 200 survey questionnaires were randomly distributed among 25 households in each of the 8 different colonies which were selected as the study site for the collection of data. The respondents were asked questions pertaining to their usual behavior and their knowledge attitude towards the solid waste management. Questionnaire was designed which consisted of many sections like natural environment, municipal solid waste management, concerned about solid waste management, willingness to participate and pay, and environmental health, solid waste management attitude scale as well. So purpose was to reveal the residents' preferences and willingness to pay for an improved waste collection service, to check and provide baseline assessment of current public attitudes and barriers to waste management, and to suggest areas for improvement. Survey was completed through face-to-face doorstep interviews and limited to 1 per household. There were 2 stages of selection for participants. Household was first selected and member of the household to be respondent was then selected. Potential participant was approached, given an information sheet plus verbal explanation of the study, followed by request for participation. Questionnaire took almost 10 to 20 minutes to complete, and the participants were all conserving adults. Questionnaire were derived from literature review in accordance with the study of the aims. So questionnaire also sought details under the 4 main headings as household details, which include gender, age, education, number of people residing in the family, extra. And second was household management and disposal, to check the current practices. Third was collection services and related satisfaction. And the fourth was perception regarding solid waste management, which was the main focus area. So in this section, 8 questions were asked to check the respondents' knowledge on composting technique, awareness of problems, impacts of waste on health and environment, and major source of information related to solid waste management. So this was a methodology which was followed, and the results which came as there were results compiled on different things, like results about disposal behavior of the respondent was showed that 70% had nearby containers and 22% had nearby their homes. So 8% threw their wastes in the open spaces. Dustbin availability was available to almost 82% people, and 18% were without dustbins. So distance, or if we talk about the distance or travel to deliver the waste, which was something 22% people had to travel a distance of up to 50 meters to throw their waste. 42% has to travel 50 to 100 meters. 22% more than 120 meters to 200 meters, and 14% people were about 200 meters. So percentage of females to males as disposable, if we talk about disposable members, females were very high. 82% of the females were the disposable members, and 18% were the males as disposable members. Segregation of waste was not strictly followed, as only 9% of the people were able to segregate their waste. Rest 91% didn't prefer segregation. So garbage collection services were available to 98% people, and rest 2% had no such services. If we talk about the type of the collection services in a week, regularly 40% people were given the regular service of collection of waste. Twice in a week 12%, once in a week 10%, and in the alternate days 38% people were provided the services. So monthly payment of the waste collection services was 40% people paid below rupees 50, 45% paid rupees 100, 14% paid above rupees 100, and 1% who didn't pay. Percentage of respondents willing to pay was 96%, and 4% were those who were not willing to pay the further services. When we talk about the satisfaction level towards the service providers, mainly the agency which is working here is Srinagar Municipal Corporation in Srinagar, 70% of the people were satisfied by the services, and remaining 30% were not. Reasons of unsatisfaction were behavior unacceptance, collection problems, disposable problems, charging extra money, or unusual time collection, we can say that. So media awareness was available to 95% of the people. Awareness about composting was to 45% people were familiar with this, and 55% were not. Knowledge about wet and dry waste and organic and inorganic waste was available with 55%. 45% were unfamiliar with this thing. And problems caused by improper solid waste management, 93% solid waste around people said that it is causing damage to health because waste gets accumulated in the open spaces, and flies, odor, rats were around, and 77% people were unaware about complications which were caused by this waste accumulation. So conclusion remarks, we can say that despite having municipal services, many people dump waste in open spaces, which can cause serious health and environmental problems. Also, solid waste was carried out through unskilled and professional personnel, which is very important because as it is a matter of health and environmental degradation, so skilled personnel are needed. Shift services and night services were completely absent. There were no shift services or night services. 80% of the waste generation was taken to the Achan dumping site, which is a only dumping site in the Srinagar city. 80% of the waste of the city gets dumped there, and 20% remains unattended. So it is also a matter of concern. Recycling, reuse, and reduce concept was missing completely. Awareness about segregation was lacking among the people. So people needs to be educated about the segregation of the waste. So also residents were concerned about the health and environment, but they need true counseling and guidance. So recommendations, if we think that open dumping of the waste should not be allowed, one thing, and the placement of receptacles should be correct. Also 4R formula, that is reduce, reuse, and resource management or recovery principle should be followed. So government and the concerned authorities will have to pay proper attention towards this thing. So with this, I conclude my work, and I also again thank the concerned authorities and the organizers for providing me the platform to present my work over here. Thank you. Thank you so much.
Thank you, Mir Khalid. Over to you, Dr. Sumi and Dr. Sudha, for any questions and for giving valuable suggestions.
Mir, can you please introduce yourself, because we didn't get your institute from where you are representing and all, because it was not a PPT presentation, so we are not getting it.
Ma'am, it was an oral presentation. I'm Khalid Mir. I'm presently I'm pursuing my PhD in the botany from Shri Venkateshwara University, Gajrola, Uttar Pradesh. I'm from Srinagar, ma'am.
Okay, so whether this was a part of your PhD work or just
no, ma'am.
What
it was
the
not
purpose of your work?
Basically, ma'am, my sister is there. She's a master student in environment. So we collectively did a work. She was interested in this work, so I was there to help her, and I was there with her all the time. So I thought that this work should be presented. Right now I'm preparing paper on this also.
So the thing is that if it was a contesting paper, you cannot contest because it was not a PPT presentation. So because it involves lots of statistics.
Yes,
so
yes,
it was better to have that type of presentation because we are hearing 90%, 96%, 4%, but we are not compiling. Our brain is not compiling all this data. So
yes,
you could have done that way. Otherwise, it is like a survey. So,
ma'am, it took me almost 3 months to complete this work, and unfortunately, because of 2G services, I'm feeling we are having a lot of problems over here because of this internet connectivity. Last time I was able to present my paper, that was a very difficult to present my paper at that time. Also, they also said that are you not having any connectivity or something like that? So I said no.
you are doing that repeating, but there was no break in the connectivity now. So you could have done in a
Then I am
PPT
highly
mode.
lucky.
Because
I feel
nowadays acceptableness is not so good for the oral presentation because this is also that is online presentation also. We are not physically seeing you. So the thing is that for this type of presentation, PowerPoint presentation is very inevitable. So that's survey. Even the questionnaires has to be shown. Even the graphs have to be shown. So that's very important. So what was what was the conclusion or the concluding remarks of your work? What did you found? Other than the usefulness of this work.
Ma'am, concluding remarks. I think that every concerned authority or the government or the municipal authority will have to pay attention towards different services like night services, like shift services. They were completely missing. Shift services were completely missing, as well as night sweeping was completely missing. So
did you intimated these things, your work results to these authorities or anything like that? Did you do that?
I hope that my paper will get soon published. Soon will get published about this thing. So then I can convey.
Otherwise, it should not be there on paper. It should be published. It should be intimated to the authorities also. Then only the good thing of your work will come out.
When I will.
You.
Okay, thank you so much.
Thank you.
Thank you. Yeah. Thank you.
Yeah, most of my questions are answered. In fact, like it is, I got answer for all of that, except for it is government municipal service, no?
Yes, ma'am. Okay. Srinagar Municipal Corporation is the authority.
Yeah, then services are similar for all the families.
Ma'am, services are similar, but the personnel which are involved, they are attending some areas and they are missing some other areas. Okay. They are not punctual.
Let me interrupt and ask you, like, I wonder, why did you ask about that willingness to pay and all? Like, if it is a government pay, it will be similar pay for all.
Yes, I can say that, ma'am. Basically, the thing is, the personnel which are working, they are getting salaries. So if they come to your homes and they collect your waste and they take it away from your home, so there is something. I think it becomes the duty of a person to pay something for them also.
Okay, so I too join. Anyway, it's a good presentation. Your effort is appreciable. It's very good. Like I joined Sumi ma'am. That this kind of presentations require a PPT. Without that, we can't correctly comprehend it. What I understood is like the municipal system is not properly working, so you are concerned about it, and you want scientifically prove that, and have to contribute to that.
Yeah,
contribute for rectifying the problem. Is it?
Yes. Basically, this area is having a very heavy load of population moving. There is a lot of immigration from the villages. So if people are moving in such an large numbers, so there should be modification of the services. There should be more personnel. There should be duty bound also.
Yeah, so we appreciate your intention actually. And other queries are about like your selection of 200 household and how did you select it? That kind of thing you have to.
No, that was random. That was random.
That you have to prove statistically. Then only it will be valid.
Okay. Okay. No problem at all, ma'am.
Okay. Anyway, all the best for your work.
Thank you. Thank you so much, ma'am. I'm thankful to you. Thankful to the organizers for allowing me to provide. I was not sure that I will be able to present my work or not, but thanks a lot. This time, internet connectivity was, I think, far good from last.
Anyway, thank you, Mayor. Thank you.
Thank you so much. Thank you so much.
Thank you, Dr. Sumi and Dr. Sudha. Now we will move to the next participant.
Okay, our next participant is Dr. Ganga G. OPF 12, Ganga G. OPF 12, Dr. Ganga G. I think she's not there in the room.
Yeah, she's not there, I think.
Excuse me, Sharing, is their name Harita inside the room? They already
Harita.
sent the PPT. Yeah. They sent the PPT also. Sure.
Ma'am, Harita has informed that she will not be presenting.
Okay, then that's the care of stuff.
Okay, okay.
Gangaji is not there.
OPF 12, right?
OPF 12.
Persons, please note OPF 12 is absent. Our next participant is OPF 13, Nitya M. Nitya,
I am Nitya. Yeah, I am Nitya.
Okay, we can hear you.
I am ready to present. I am ready to present.
Okay, please share
let me share the slide.
Hello, Nitya. Nitya, can you hear me?
Yeah.
Nitya, you can start.
Here. Yeah, ma'am, I have shared my slide. Is the slide visible for you?
Yes, it's visible. You can start your presentation.
Okay, thank you, ma'am. Thank you, ma'am. A very warm good evening to one and all. So I'm M. Nitya, Assistant Professor in the Department of Biotechnology. So now going to work that is on evaluation of streptomyces against Pythium species, the causal organism of rhizome disease in turmeric, to improve the curcumin content in finger rhizomes. So let me move on to my introduction. So currently there is an increasing public concern regarding the continued use of agrochemicals to control the phytopathogenic fungi. So several efforts have been made controlling these plant pathogens among with the biological control using the microorganisms. Turmeric belonging to the family Zinziberaceae. It is one of the most ancient and important spice crop grown in India. Rhizome rot disease of turmeric caused by Pythium. It is a major constraint for the production of healthy rhizome. Sometimes it will be causing the total crop to be failure. Rhizome rot disease of turmeric caused by the fungus Pythium species causing the total failure of the crop is a major limitation for the production of healthy rhizome. So my present study aims to isolate, evaluate, and characterize the streptomyces species collected from different agroclimatic zones of Tamil Nadu, and it have been evaluated for the potential antagonistic activity against Pythium species. There is a causative agent of rhizome rot disease of turmeric plantation crops, and my objectives of the study is to isolate the streptomyces species from various turmeric plantation soils of Tamil Nadu, and to categorize those isolated species level, and the screening of the potential strains of streptomyces species for the antagonistic activity against rhizome rot disease of turmeric plants causing a fungal pathogen, Pythium afanidermatum. And the next one is to develop a simple, reliable, and reproducible protocol for mass multiplication of Streptomyces species and to prepare carrier-based bioformulations. And the methodology what I have adapted is for determining the edaphic parameters like soil pH, electrical conductivity, total organic carbon. I have used pH meter, AC meter, as well as Valke and black. And for estimating the total nitrogen and phosphorus, potassium, calcium, sodium, and magnesium, I have followed AOAC method. And regarding the environmental parameters, I have determined the rainfall, temperature, relative humidity, and sunshine. So this I have gathered from the respective farm areas where I have worked. Then for isolation and identification of chitin degrading bacteria, I have followed the Mondale and Rees method, as well as for screening purpose, I have used Sanmukhya et al. Method. And for enumerating as well as biochemical characterization of chitin degrading bacteria, I have followed the method of Cappuccino and Sherman method. And the result of this study showed that, so I have chosen 5 plantation district, that is Dharmapuri, Coimbatore, Erode, Karur, and Salem. And the pH of the soil was very high, of around 7.8 in Erode. And regarding the electrical conductivity, it was found to be 0.37 in Dharmapuri. And regarding OC, it is found to be 3.7 in Erode. And the nitrogen content was very high, of about 39 in Dharmapuri, and 16.5 of phosphorus in Dharmapuri. And similarly, the potassium content was found to be high in Coimbatore, of about 67.3, and calcium, magnesium, and sodium was found to be 228.2, which is in Erode. And regarding magnesium and sodium, it was found to be 123.8 in Coimbatore, and sodium it was found to be 34.8 in Erode. And regarding the chitin degrading bacteria, that is number and designation of chitin degrading bacteria, isolated from various plantation district of turmeric. In Coimbatore, I have selected 3 taluks, Palladam, Pollachi, as well as Avinasi. And in Karur, I have selected 3 taluks, that is Paramati, Pudithalai, and Krishna Rayapuram. And in Erode, I have selected Kodumudi, Govichettipalayam, as well as Andiyur. And most probably the number of isolate I have obtained from Coimbatore is around 15, 20, and 16 of the corresponding taluks. And in Karur, I have found 28, 25, and 20 correspondingly to the taluks what I have mentioned here. And in case of Erode, Kodumudi, I have isolated 31, and in Govichettipalayam, I have isolated 25, and in Andiyur, it was found to be 24. And the next one, Sam, I have selected 3 taluks, and the number of isolates that I have obtained was found to be 15, 20, and 14, correspondingly to Metur, Atur, as well as Sankiti. And regarding Dharmapuri, I have selected Pendagaram, Palakodu, as well as Pochampalli. So the total number of isolates that I have obtained was found to be 28, 31, as well as 20. And this is the image how I have isolated the chitin degrading bacteria. So the clean primary stage and based zone of clearance, I selected the bacteria. And secondary screening, I have isolated before
Nitya ma'am, we can't hear you. Your voice is breaking. Ma'am, are you there? Nitya ma'am, are you there? I think she's lost her connection.
Yeah.
characteristic features of chitin degrading bacteria that have been isolated from Tamil Nadu.
Ma'am,
I
can you
have gone
please
for.
hold on? We can't see your screen.
Yeah, ma'am. Let me wait. Let me wait for a few seconds.
Okay.
Okay. Ma'am, is it visible now? Yeah.
Yes, yes.
So I have undergone biochemical tests like hydrogen sulfide production, nitrate reduction, starch hydrolysis, gelatin hydrolysis, L-arabinose, D-xylose, as well as mannitol and casein and lipase hydrolysis. So almost for many of the biochemical tests, it will be producing a strong reaction, and to some extent, it will be producing the medium, and few of them have the response of negative. And these are the characteristic features of chitin degrading bacteria that have been isolated from Tamil Nadu turmeric plantations Rhizosphere SIAS. So I have performed the following biochemical tests: gram staining, motility test, indole test, methyl red test, Voges Prescott test, catalase, oxidase, as well as citrate and urease. And regarding this, to some extent, most probably for indole, methyl red, Voges Prescott, I have got the negative reactions, and the strong reactions was obtained for that is motility test, as well as for catalase, oxidase, and citrate test. And I have identified this organism as streptomyces, and in species level, I have not determined. And regarding, so once I have got those organisms, I have gone for standardizing the media for growing that organisms, especially of streptomyces. So for standardizing the media, I have used Dubey and Maheshwari, and I have also studied the biotic sources as well as the effect of abiotic sources. And also I have gone for antibiotic resistance capacity of streptomyces. So this is the result of streptomyces strains growing in liquid media containing different carbon sources. So almost when I have used glucose as a carbon source, all the organisms have shown a major growth rate. And in case of nitrogen sources, almost in peptone as well as yeast extract, I have seen that streptomyces have well growth. And in case of amino acids, almost trypsin, aspartic acid, arginine, as well as glutamine, it shows a major result for the growth of streptomyces. And in case of vitamins, vitamin like thiamine, pyridoxine, as well as folic acid and biotin have shown a major growth of streptomyces. And this is a minimum inhibitory concentration of antibiotic resistant profile when I have used streptomyces strains. And regarding the summary, the maximum amount of organic carbon and nitrogen content were noted in Erode of about 3.7%, and in Coimbatore it was noted of about 3.8. And moreover, the soil sample collected from Coimbatore region showed the highest density of chitin degrading bacteria. And EP3, SP5, KP6, CP4, and DP6 representing each agroclimatic zones that I have chosen for the study. And all the strains were identified as streptomyces. Okay, let me conclude with this. Thank you.
Thank you, Ms. Nitya, for your presentation. Now the floor is open for question and answer session. Participants, you can type your questions in the chat box. Over to Dr. Sumi and Dr. Sudha.
Cynthia, it was a speedy presentation you did. So many data were shared. And the thing is that, why did you choose this plant, Rhizosphere soil of this plant particularly?
Ma'am, since when we are saying, ma'am, I am in Erode, so the turmeric was found to be a major cultivation here. So I have interested to select those plants.
What is its relation with chitin degradation?
Because almost all of the rhizomes will be infected by pythium. So when we are saying about the nature of fungus, the membrane will be having a chitinase. Okay. So in order to degrade those chitin and to degrade the cell wall of the fungus, I have chosen especially of the chitin degrading bacteria.
So did you do the experiment using this pathogen, Pythium bacteria and this Pythium pathogen?
No, ma'am. No, ma'am. No, ma'am. So far, just I have isolated the organism and it is under pipeline.
So your objective was that. That's why I asked. Okay. Okay. Then. Over to Sudha.
Okay, ma'am.
Nitya, very
Yeah,
extensive
ma'am.
presentation.
Yeah,
Before
ma'am.
we could hear you are rushing through the slides. That's good presentation anyway. I have one question, like why did you use colloidal chitin medium for that? And you are confusing with the bacteria and streptomyces. Streptomyces belong to actinomycetes. For isolation of actinomycetes, why did you use colloidal chitin agar? What is its ingredient? Usually
Ma'am,
let me tell you one more thing. Like why I asked the question is, for actinomycetes, usually a separate medium is there.
Yeah.
Kuster's agar, like Kuster's agar. So why did you use this particular medium? And
Yeah,
what is the
ma'am.
pH of this medium?
Ma'am, previously, what's my main objective was my work was full focus on chitin degrading media because since this fungus contains chitinase, I have gone for using this chitin degrading media for cultivating the isolated bacterium from different rhizosphere soil. Okay, so simultaneously or parallelly, I have gone for that media. And present, I am also trying for the media that is a specific media for streptomyces.
So you're optimizing media for this streptomyces. Now
yeah, ma'am.
Okay.
Yeah, ma'am.
What is the pH of that media?
Available, no? For the streptomyces and all, actinomyces, casein, starch agar, like that. Many
casein, starch agar, casein containing or starch containing low pH media is there. It's called Kuster's media. Specific medium is there.
Ma'am, I don't have idea regarding that media.
Yes. Actinomycetes will form some very small powdery colonies in that. And you can choose it. And for identification, we mainly use morphological characteristics rather than all the biochemical tests. For actinomycetes, it's better to use go for staining technique, and morphologically you can identify it. Okay. And another doubt I had is
like, let me note, ma'am. Okay.
Okay. While you are preparing that antibiotic resistance one chart you have shown, and you have skipped through it, but it shows antibiotic resistance. Whether streptomyces shows that much resistance, multi-drug resistance?
Yeah. Yeah, ma'am. Yeah, ma'am.
It is showing multi-drug
I have done a triplicate. It had shown that much of zone.
Okay. So how can you use it for soil reclamation or for you are planning to use it as a antagonistic agent like a biopesticide?
Biofertilizer. Fertilizer. Yeah, ma'am. Biopesticides.
Against the Pythium? No. Against the Pythium? Against the fungus Pythium? Okay. So how can you add, if it is with that kind of resistance, how can you add it to soil?
Ma'am, I have chosen to use talc powder to mix it up and to use it as a biofertilizer.
No, my concern is about if we are spraying or adding abundance of antimicrobial resistance organism to the soil, it will raise to another problem. No? That was the question. And usually streptomyces is slow growing, and as a antagonistic agent or biopesticide, we will be usually using bacteria. And maybe this is a new study. You need to study a lot on this topic, but it looks good. It's good presentation.
Okay.
Whether it's a part of your PhD work? I felt some lack of clarity somewhere. That's it. Other than that, it's a good presentation. I think you need to study more.
Yeah, ma'am. Ma'am, lack of time, I have skipped many slides. That's the main reason.
Yeah, I understand. Okay. If participants have
Okay,
any
ma'am.
questions, you
Okay,
can
ma'am.
answer that. Thank you.
Thank you, Dr. Sumi and Dr. Sudha. There is a question from Dr. Gaurav Kumar. How can you link chitin degrading bacteria role in the turmeric? Miss Nitya, there is a question in the chat box.
Yep. Okay, ma'am. So after performing many biochemical tests, we have confirmed that as a chitin degrading bacteria. So after isolating that, we have gone for biochemical test and we have confirmed this as streptomyces. So now we are going to use the streptomyces as an antagonist against the against this pythium. And after preparation of this streptomyces, we can the plantation, turmeric plantation crops.
Okay, thank you. Thank you, Miss Nitya.
Okay, thank you, ma'am. Thank you, ma'am.
There is no more questions in the chat box, I think. Now we will we will go to the next participant.
Okay, ma'am.
Thank you.
The next participant is OPF 14, Dr. Maya Rani. OPF 14, Dr.
Yes.
Maya Rani.
Yes. Am I audible?
Yes, ma'am,
Okay.
you're audible. Can you please share your screen?
Okay, I'll try. Is it visible?
Yes, ma'am, it's visible, but not your PPT.
Oh, I see. Excuse me. I am sending my PPT to your email. If possible, kindly help me. Hello.
Okay, yes, ma'am. Shabana, ma'am, are you there?
It's not working. I don't know what happened with that.
Okay, ma'am, can you hold on for some while? We'll call on the next participant. Till then, you can send the PPT to our Gmail
I am
account.
sending. I'm already sent. I am already sent.
Okay. Shabana, ma'am, are you there?
Yeah, I'm here. Did she send her this her PPT?
Yes, she has sent.
Hello.
Yes, ma'am, she has sent.
Please tell her what is the title of that PPT?
Maya, ma'am, can you
Deleterious
please
effect.
tell us the title
Hello?
of your PPT?
Yes. Deleterious effect of the butylated hydroxytoluene on the kidney of Swiss albino mice. Just now I am sent.
Yeah, it's here. I am presenting.
Okay, then Maya, ma'am,
Hello?
can you please stop sharing your screen?
Okay. Okay.
Okay.
Yes, it's visible now, Gigi. Maya, ma'am, is it visible to you? Dr. Maya Rani, are you there? Ma'am, are you there?
Yes, I am. Hello?
Ma'am, is the PPT visible to you?
Yes.
Okay, ma'am, you can start your presentation.
Okay. Good morning, one and all present here. I am Maya Rani from Patna Women's College. Today I'm going to present my topic is deleterious effect of the common food antioxidant butylated hydroxytoluene on the kidney of mice. As we know, all foods are subject to microbial attack. Fats and oily food become rancid, particularly when exposed to the human. Preservative and antioxidants are nowadays frequently used to prolong the shelf life of food. Preservative inhibit the microbial growth so that the natural flavor, taste, and the food value of the foodstuffs are maintained. Antioxidants are added to prevent rancidity, browning, and the development of the black spot due to the oxidative changes. Together, they are referred to as food additives. Food additives play a vital role in the today's bountiful and nutritious food supply. They allow our growing urban population to enjoy a variety of the safe, wholesome, and the tasty food around the year, and thus they make possible an array of inconvenient of the daily shopping. The food additive defined as a substance or a mixture of a substance other than the base food stuff present in a food as a result of any aspect of the production, processing, storage, or packaging. Though these chemicals carry out a variety of useful functions, their excessive and the regular use has been noticed to be detrimental to the human health. The present study was conducted to elucidate the relationship between the level of toxicity of an antioxidant butylated hydroxytoluene and the induced renal toxicity. Butylated hydroxytoluene has been widely used as an synthetic antioxidant to prolong the shelf life of food. It stops the chemical breakdown of the food that happen in the presence of the oxygen. It is extensively used in oil, chewing gum, dried potato flakes, and nuts. Unsaturated fatty acid in oil and lipids are particularly susceptible to the auto-oxidation. These antioxidants function by the interrupting the free radical chain mechanism involved in BST can also improve the stability of pharmaceutical, fat-soluble vitamin, cosmetic. The service life of the rubbers, elastomers, and the plastic is increased by the addition of BST, and from such use, BST is often present as an indirect food additives. Now, method and methodology. Next slide. Next slide, please. Next slide, please.
Shabana
Ma'am.
ma'am, are you there?
Shabana ma'am? Okay.
Okay, no problem. The material and methodology. In the present study, the male Swiss albino mice with the average body weight of the 30 to 32 gram were selected for the experimental study. The animal were housed at the controlled environmental condition to 22-24 degrees Celsius, relatively humidity 50 to 60%, and the 12-hour dark light cycle. Animals were housed in a polypropylene cases and allowed to free access of the food and water. The mice were divided into the two groups. There was a control group and the treated group. The control group were not treated and therefore supposed to have the normal basic structure of kidney. The second group was treated with the BST 225 mg per kg body weight and 300 mg per kg body weight orally for 60 days. After the experimental period was over, the animal sacrificed from each group for the histological study. The selected organ was dissected and fixed to 10% neutral form of fixative and cured for hematoxin and eosin examined under the microscope. The procedure were following those dirty and valid. All the experimental data, statistical analysis, these are the parameters of the study. The present study was the Swiss albino mice. The different segment of the kidney were observed and analyzed, like a diameter of the chromosome, diameter of the Bowman's capsule and epithelial cells, diameter of the proximal convoluted tubule, diameter of the distal convoluted tubule, and the diameter of the collecting duct. Next, statistical analysis. Okay. The diameter of the different subdivision of the kidney were taken by the standard method. All the experimental data were expressed as the mean plus minus standard deviation. The changes in the diameter of the different part of the kidney in the treated group were compared to the control and therefore significant analyzed by the t-test. Difference were considered significant if the p is less than 0.5%. There are now results. Next slide, result and discussion. Table 1 shows the diameter of the glomerulus of the kidney of the mice after the oral treatment of BST. Table 2, here is the diameter of the Bowman's capsule of the kidney of mice after the oral administration of the BST. Next. Table 3, table 4 shows the diameter of the distal convoluted tubule of the kidney of the mice after the oral administration of the BST. Next. Diameter of the collecting duct in the kidney, in the of the kidney of the mice after the oral administration of the BST. Next. Next. These are the photographic plates of the normal kidney. Here the normal architecture of the glomerulus, Bowman's capsule, proximal convoluted tubule, and the distal convoluted tubule. Next. Next. Normal previous one, please. Previous one, please. Yeah. Normal control mice. These are the photographic plate of the normal control mice. The proximal convoluted tubules appeared normal. The star-shaped lumen of it can be easily seen. The distal convoluted tubule are visible distinctly. The nuclei of the renal tubules seem normal in structure and shape and number. Next. This is the photographic plate of the treated mice with the 225 mg per kg body weight butylated hydroxytoluene for the 60 days. It showed the clumped glomerulus and the necrosis of the epithelium of the Bowman's capsule. Necrosis of the tubular epithelium is also visible. Irregular spacing are also visible there. In the second which is also treated with the 225 mg per kg body weight butylated hydroxytoluene for the 60 day. It shows the degeneration of the tubular epithelial cells, agglutination of the epithelial cell nuclei, renal tubular cells are observed. Next. Next. These photographic plates shows the treated, next previous one please. Yeah. In this photographic plate, we can see the decreased number of epithelial cell nuclei in the proximal convoluted tubule and the distal convoluted tubule, as well as the cytoplasm of the proximal convoluted tubule and the distal convoluted tubule to be degenerated. Loss of brush border and the proximal convoluted tubule clearly visible. In the second photographic plate, it is also seen the parietal layer of the Bowman's capsule degenerated. Glomerulus appeared clumped, tubular lumina contained the sloughed epithelial cells. Tubular epithelial cells appear to be degenerated, irregular spacing, and the decreased number of epithelial cell nuclei seen. Next. There was a significant change observed in the diameter of the glomerulus with both doses. That is the 225 mg per kg body weight and 300 mg per kg body weight BST orally per day after the 60 days. In histopathological examination, clumped glomerulus was found. The glomerulus is target of the toxic effect of the number of drugs and the chemical agents that may cause the changes in the glomerular morphology. The epithelium of the glomerulus undergoes irreversible damage. The capacity of these cell types to repair damaged tissue is restricted as compared to the other cell types such as the PCT. Although the glomerular and Bowman's capsules are together referred to as the renal corpuscles, but they differ greatly in the extent of damage as well as the repair. In the present study, the Bowman's capsule so on the treatment with the 225 mg per kg body weight BST orally per day, there was a significant change observed in the diameter of the Bowman's capsule after 60 days, but no significant statistical study was observed at the rate of 300 mg per kg body weight BST. In histological study, there was a degeneration of epithelium cell of the Bowman's capsule was observed. Next. Because of the large volume of the fluid enters and reabsorbed by the proximal convoluted tubule. Ma'am, previous one, please. I am right now in, you are on the collecting that, I am in the proximal convoluted tubule, just previous one. Yeah. Distal, no, this previous one. Yeah. Yes, this one. because of the large volume of the fluid entering and being reabsorbed by the proximal convoluted tubules, its many and varied transport mechanism, and its mitochondrial and cytoplasmic enzyme activities associated high metabolic rate, the proximal convoluted tubule is a target for a wide variety of the toxicants. In the present study, there was no significant change observed in the diameter of the proximal convoluted tubule after the treatment of the 225 mg per kg body weight BST after 60 days, but the diameter of the proximal convoluted tubule showed significant increase after the 60 days treatment of the 300 mg per kg body weight BST treatment at the P is less than the 0.5. In histopathological study, decreased number of epithelial cell nuclei and the loss of brush border and degenerated cytoplasm of the proximal convoluted tubules were observed. Tubular lamina contain the sloughed epithelial cells. The increase in the diameter of proximal convoluted tubule may be attributed
Dr. Maya, are you there?
as it is known. DCT, please. Yeah. As it is known, the intraluminal concentration of the toxicants are likely to be higher in the distal region of the renal tubule, providing a great concentration gradient to drive the passive diffusion from the lumen of cell. The distal tubule is characterized by the relatively tight epithelium with a high electric resistance, a feature that may be impede diffusion to solute. In butylated hydroxytoluene treated group, the diameter of the distal convoluted tube showed a significant increase for both the doses 225 mg per kg body weight and the 300 mg per kg body weight after the 60 days treatment. In the light microscopic study, the sloughed cytoplasm and the cellular debris were common within the tubular lumina. There was no significant in the next collecting duct. Next slide, please. Yeah, there was no significant changes observed in the diameter of the collecting duct after the treatment with the 225 mg per kg body weight for the 60 days. There was significant increase. There was significant increase in the diameter of collecting duct after the treatment with the 300 mg per kg body weight after the 60 days. In histological study, there was a tubular cytolysis was observed. The collecting duct appeared to be relatively insensitive of the most of the nephrotoxicants. Damage due to the tetracyclines may be occurred in this segment. Next. In the present study, glomerulus and the distal convoluted tubule shows the maximum changes among the various subdivisions of the kidneys. The present study show that continuous use of butylated hydroxytoluene and its use in excess quantity, it's harmful to the human health. Therefore, the quantity of these food additives has to be carefully monitored in the food products. Thank you.
Thank you, Dr. Maya Rani, for your presentation. Now the room is open for QA session. Participants, you can type your questions in the chat box. Over to Dr. Sumi and Dr. Sudha. Hello.
Okay, Dr. Maya.
Yes.
Your presentation were mostly about the tissues. So how did you take that specimen and what was the staining method you used for this study?
My selected animal was the Swiss albino mice. We have an animal house in our college. We procured the animal from that only, okay, from the animal house. The selected animal model was the Swiss albino mice. So we rear it, okay. And next is, what the next question is? How I processed, okay. After the treatment, Hello?
You are not audible.
Hello? Am I audible?
Tell me. Yeah. Now it's okay.
I'm not audible?
Yeah,
Oh,
now it's okay.
okay. So my select. Okay.
No, not audible, Dr. Maya. You are not audible now.
Hello.
You are
Hello.
not audible now. Now it's okay. You can tell.
Okay. The my selected animal was the Swiss albino mice. Okay.
Yeah.
And tissue was actually after the treatment we sacrificed the animal and all the vital organs we removed and fixative put it into the fixative and the processing was done like a we microtomy. Basically we do the microtomy.
Yeah. Then staining, what type of staining was done?
A hemotoxylin and the eosin.
Okay. So
Double staining method. We are using the double staining method, hemotoxylin as well as we are making the paraffin wax blocks and then trimmed and
okay.
staining
That's
is
a normal
basically the
thing.
double staining methods. Okay.
Yeah.
Yeah.
So
This is the light microscopic study. Actually, I am also done the electron microscopic study.
So this is part of your PhD work, or no? You are having
Yes.
PhD, no?
Yeah.
You are having PhD.
Yeah, I am
no?
having PhD, but I am having PhD, but this was my part of my PhD work.
Okay. Earlier. Okay. So, in which type of food you find this type of preservative normally?
Basically, fats, oil, you can see that every food product have a food additives number like a E21, E20. You all know that. Okay, whenever you check on their, like you buy some biscuits and you get there is a E21 is there. So suppose E21 is the butylated hydroxytoluene. Basically, it is used in the fats and oils like a mayonnaise.
Yeah. Is there any that permissible limit for that one? And whether it
Yes.
is correlating with your work?
Limit is a 0.125 no, my work is basically toxicity, induced toxicity. So my dose is much more than the permissible limit.
That's why I was wondering, because your dose is more and the permissible limit is different.
Yeah.
Again, did you do any antioxidant studies?
Permissible limit is 0.125%.
Antioxidant studies.
Pardon?
Any antioxidant studies?
Antioxidant?
Yeah.
No.
Because in the introduction you were telling about this antioxidant activity or something like that.
No, I can antioxidant. This is used as an antioxidant. Butylated toluene is antioxidant. So it has a beneficial effects also. This is a controversial. This is a kind of the controversial topic. Some people say that it has a very beneficial effect. By the time they use, you know what? It has also a detrimental effect. My work is that support the detrimental effects.
Okay, that was my query. Okay, you can move on. Yeah.
Thank you very much. Thank you, ma'am.
Dr. Maya Rani. It's a very good presentation.
Yes, ma'am. Thank you, ma'am.
Like it is a grass compound. Butylated hydroxytoluene is a BHT is a grass compound. So the minimum permissible limit is 125 milligram per
kg body weight for the human being.
Okay. And it is antioxidant also, like second continuation of Sumi ma'am's question. It is an antioxidant. So she was asking
it is.
Why did you check the antioxidant potential? Like,
pardon, ma'am?
The question is like
pardon,
permissible one, whether you are checking the kidney damage is there when we are using excessive amount of BHT.
No. At the permissible, it is not kidney damage. This is basically for the academic study. Okay. So suppose when you are doing the toxicological study, this is basically a core toxicological study. Okay. So we are doing we done the toxicological study. So we first perform the LD50. And then after that, we select the different doses.
Dr. Mayarani, why I asked is my concern, I was worried by seeing the slides. The kidney is severely damaged. Kidney is severely damaged in the slides.
Yeah.
And
Yes,
then how
yes,
such a
yes.
chemical got permission as a GRAS compound?
No, many of the countries, foreign countries, like the European countries banned it, but it is not banned in India. That's why I'm selecting the topic.
Okay, so it's a relevant study. Anyway,
Yes.
questions are answered. You can move on to the questions of participants if they have.
Sure, ma'am.
Thank you, Dr. Sumi and Dr. Sudha. Dr. Mayarani, there is a question in the chat box from Dr. Gaurav Kumar. You can read that also.
Yes.
Some antioxidant will be used in the limited amount in a variety of the food products to enhance the shelf life of the products, that is 10 to 50 ppm. What was the dose authors used in your study?
Yes.
Then if it is not equal to the food, how can you justify your work? It is the question from Dr. Gaurav Kumar.
Hello, am I audible?
Yeah.
Yeah, actually, this is basically pure academic purpose. This study is for the academic purpose to know what kind of the damage occurred at the higher doses. You know what? If I'm doing that, this is the permissible limit. The lots of work is already done. So there's no need to doing the such and repeated work, no? Hello?
Hello?
Am I?
Yeah. Dr. Gaurav?
Am I audible?
Yeah, you are audible.
Okay.
Dr. Gaurav, you are satisfied with the answer? Okay. Now there is no more questions in the chat box. Now we will go to the next participant.
Okay, ma'am.
Thank you, Dr. Mayarani. Thank you, Dr. Sumi.
Thank you very much. Thank
you. Now we will go to the next participant.
Our next participant, OPF 15, Suresh Ambati. OPF 15,
Yes.
Suresh Ambati.
Yes, good afternoon, ma'am. Am I audible?
Yes, sir, you are audible. Can you please share your screen?
Sure, ma'am. 1 second. Is the screen visible, ma'am?
It's still loading. Yes, now it's visible. Can you please put it in a slideshow mode?
Okay, I just changed the screen into what you call PDF, ma'am, so that it doesn't get disturbed. Means it doesn't move. So, any problem with it? So, you want me to share only in the PPT mode or what?
No, it's okay, but you will have to still move, right? While presenting?
Yeah, and I should move myself, no? Means I'll be moving it.
Okay. If you're comfortable with it, you can continue.
1 second. Yeah, now that it's moving. Can you see? Can you guys see it?
Okay. You can start your presentation.
Yeah. Good evening, one and all. Once again, it's a very wonderful webinar. What I have been seeing past 2 days. I think I'm the last but one of before, but the presentation which were made earlier before were related to some kind of genetic and microbial thing, and mine is a kind of solid waste management, a kind of environmental related thing. So my paper is a scientific approach on solid waste management practices, a case study. This is a case study what I did in the Andhra Pradesh, urban local body, or you can generally call it as a municipality. And see, I have selected these are my contents, abstract, object, everything. See, why solid waste management is more important, or why it's gaining a kind of more thing towards the general means. The reason behind it is because due to a lot of urbanization, population explosion, technology, everything, the human mankind is making a lot of generation of waste in the form of packaging, product making, anything. So it is creating such a big problem because we guys as individuals, we are just generating it, but we are unable to control that particular waste in a proper way. So in near future, if the same trend goes on, we'll be in such a state where our garbage pile will be next to our house only. A condition may come in the near future, our garbage will be in our own backyard. Nowadays, we are just throwing it outside with the municipality guys or the the panchayat guys, they are picking up. In future, there won't be no place available where we can at least even dumping or processing. That is the main thing. Solid waste management has been a prime more importance for guys nowadays. See, my objectives are adopt methodology as a tool for making decisions on the investment of infrastructure facilities, make it an efficient and effective and efficient mechanism for managing and processing of the garbage. Estimation of shortest path from the source point means that is collection, where the garbage has been generated, to the processing facilities. Optimization of resources, because in a country like India, we need to go for maximum optimization, because as we guys know, there are many financial constraints, legal constraints, political constraints will be there. So we need to optimize maximum resources, and for more effective implementation of the composting methods as well as various solid management activities. Evaluation of nutrients in the compost. See, as we know, there has been a rapid increase in the production, consumption, and quantity of waste generated in the urban society, as well as due to which the municipal solid waste management has become a continually growing problem at global, regional, and local levels, as we guys know. This necessitates management of solid waste generation, storage, collection, transfer, transport, processing, and disposal in an environmentally sound manner in accordance with the best principles of public health, economics, engineering, conservation, aesthetics, and environmental considerations. Thus, a solid waste management includes all administrative, financial, legal, planning, and engineering functions. By continuous research and adopting latest and best practices, we can achieve our goal. I am concentrating on a Tenali municipality. It is a special grade municipality in the Guntur district of Andhra Pradesh state, which is generating an average of 65 metric tons of garbage daily. See, any waste which is generated in the municipal limits is called municipal solid waste. See, this is a general scenario. The world cities are generating around 2.1 billion tons of urban solid waste every day. And it is estimated that around 136,000 metric tons of municipal solid waste is generated daily in our country, in India. And per capita generation is in major cities ranges from 0.4 kg to 0.6. And it is estimated that at the urban local body, at the municipality, at the corporations, they are spending around 500 to 15,000 per ton on the solid waste, various stages, solid waste collection, transportation, treatment, and disposal. This is the amount of the money which has been invested by the municipal authorities or the corporation people, anybody. So the proper disposal of urban waste is not only necessary for the preservation and improvement of our health, but it is the immense potential of resource recovery. Hello? Okay, guys, am I audible? Hello, ma'am, am I audible?
Yes, sir, you are audible.
Thank you, because I got a disturbance. I was just checking. Okay, thank you. In this particular figure, you can clearly see by 2047, the approximate around 260 million tons, more than 5 times the present level, the waste will generate, the solid waste will generated, and you can see the significant impacts in the near future, ma'am. Okay, see, due to this, in 1999, the government of India has, under the umbrella of the EPA Act, Environmental Protection Act 1986, it has framed certain rules. It's called MSWM Rules 2000. That's short form, or Municipal Solid Waste Management and Handling Rules, under the Environmental Protection Act, to regulate management and handling of municipal solid waste, which are amended from time to time. Responsible, in which they clearly mention the various responsibility, various stakeholders, various authorities, especially the role of municipal authority is the main thing, because every municipal authority shall be responsible for the implementation of the provisions of these rules, because these guys will be taking care of the process, collection, transportation, storage, as well as processing. So the main role of implementation of MSW Rules 2000 will be the local authorities. It might be a Gram Panchayat or the municipality or the corporations. So the municipal authorities should make an application of Form 1, because there are almost various sections in this municipal solid waste management rules, under which the municipal authorities has to fill up the following data and submit to the concerned state and state pollution control boards. So these state pollution control boards, they in turn correlate and coordinate with the central pollution control board and make any necessary changes. So the response state government is to shall monitor the compliance of standards regarding the groundwater, ambient air, leachate quality, and the compost quality, including incineration standards as prescribed under the relevant schedules, ma'am. The state board shall also issue authorization upon receipt of Form 1, because as a municipal authority, if they want to they want to install any kind of incineration facility or any facility, they have to fill in the Form 1, and they have to mention all the details. What is the generation, how much waste is generated, how they are going to process it, what are the method of processing. So these are the various scientific things will be incorporated in the Form 1, by which the state board we will be taking accepting the application and giving the permission, ma'am. The central pollution control shall also coordinate with the state pollution control board for implementation and review of the standards and guidelines. See, the main functional elements of solid waste management are waste generation, where the general waste is generated. So if we control the generation of waste at the source itself, our problem will be controlled to the maximum extent. So then, okay, once the waste is generated, next step is how to handle that source. So after the waste handling can be done by at the source. After that, collection. After that, processing. The processing of waste can be done in two ways. Just like dry waste will be collected and will be sent for the recycling, and the wet waste, generally kind of what you call a market waste, or which is a biodegradable waste, can be done by vermicomposting or bacterial composting, something. And inert waste should be properly collected and not supposed to be mixed with the regular municipal solid waste. And hospital waste, hazardous waste, they should be separately collected and they should be separately sent for the concerned processing facilities. And the transfer and transport, finally the disposal of the waste. See, this is my study area, Andhra Pradesh state in Tenali. And see, this is a description of the study area, ma'am. Because Tenali, the city, it's around the latitude and longitude has been given 16 degrees and 80 degrees north. It's a southern delta region of Krishna River in Guntur district, Andhra Pradesh. It's the municipal headquarters of the Tenali Mandal, Tenali Revenue Division. Because this Tenali is I think everybody in India, they know about the Tenali Ramakrishna. He is one of the poet who resided in the Sri Krishna Devaraya regime in the kingdom. He is one of the Ashtadigajas. The city is renowned for the art, cultural, drama, and hence it is often referred to as the nickname as Andhra Paris. The Tenali Ramakrishna, one of the 8 poets and the jester of the king Sri Krishna Devaraya, hails from Tenali. The municipality was formed in 1909 and was upgraded to the special grade municipality in 1965. It spreads around 16.56 square kilometers and comprises of Tenali, Penapadu, and Chinnarauru. The total length of the roads are maintained municipality 170 kilometers, ma'am. See, this is the general information about the ULB. The total number of wards in the ULB at the municipality are 40 wards. In total population, male and female are around 8,147 in the male population, 83,510 in the female population. So, as per 2011 census, there are 1,64,937. The total number of the sanitation divisions are 9 divisions. So the total number of households which are generating a kind of garbage are 43,000 households are there. The garbage generation approximately 65 metric tons. So the vehicles which are used for transport, for collecting, transporting the waste are around 27 autos, 13 tractor trailers and push carts, and they have one compost yard with an area of 6.67 acres, ma'am. See, these are the various solid waste management activities taken to the municipality. As we guys know, as per the Municipal Solid Waste Management Rules 2000, the waste has to be segregated at the source itself. Where the waste is generated, there itself the waste has to be collected in a segregated manner. Just like from households, residential households, it has taken a dry waste and wet waste. You can see the 2-bin system is introduced in the particular municipality. Now all over India it has been introduced, but the system is followed in a very particular way in this particular municipality, where the municipal worker, she is collecting the wet waste and dry waste in 2 different containers, ma'am. See, here we can see the collected residential waste or the household waste is again transferred into the push carts, because where the vehicles can't, there will be narrow lines where the autos or the tractor trailers, they can't go or they can't move into the narrow lanes. So this municipality has adopted a kind of push cart methodology where the particular municipal worker, she'll be taking the push cart to the interior lanes, collecting the waste, and the segregated waste is again separate. You can see in the picture separately collected and in the 2 different containers. You can see in the background there is a lady who is having a notebook. She is called Nagaradipika, a kind of social worker. She'll be motivating the general public, because as we guys know, why we need to give, why is any special reason. So this municipality has developed a kind of special technical scientific method where motivation, IEC activities also play a major role in the proper management of solid waste management, ma'am. See, these are transportation vehicles where from the push carts, it has been transported. You can see 2 different colors, red and green. Green for the biodegradable waste container and red is for the kind of dry waste containers. See, now these are dry waste processing facilities where the guys, the workers who collect the waste, the dry waste is properly collected. You can see in the 2 pictures, the paper, the cardboard paper has been bundled here, and the plastic, the plastic water bottles, everything are separated. So these guys do the separation techniques, and And this is about the dry waste. In this dry waste, this municipality has taken a innovative step, ma'am. I need to acknowledge, because see, for municipal workers, they'll be taking all the pen, collecting the waste. So by the end of the day, they are giving it to the concerned recycler. So he'll be paying the money in terms of finance. See how these guys are implementing the thing is, so they have made an agreement that the worker should bring the waste in a separated manner and transport the waste in separated to the processing facility, where in the final stage the waste is weighed, and for each quantity of the each type of waste they have been given a particular pricing, ma'am. Just like the plastic bottles are there at the rate of 3 rupees per kg, paper is there 6 rupees per kg, cardboard is there 5 rupees per kg. They have a standard pricing chart where if a worker brings a so and so amount of the waste in a proper collected way, so for example, a worker is getting a waste and the amount comes to around 100 rupees. So 50 rupees is given to the worker and 50 rupees is deposited in the municipal treasury, because the reason behind is because even the worker morale is increased and they are getting kind of additional income. So the worker started working in a more sincere way. Because because if we get some kind of benefit, that's a kind of normal thing. If anybody gets a kind of benefit, so they do the work in a more sincere, because they are a kind of class of workers, even they need the money. So this is a very innovative practice I have seen in this municipality. So in the wet waste processing, they are doing vermicomposting, ma'am. As we all know, the biodegradable waste is transported to the concerned composting yard or the composting facility, where the biodegradable waste is treated with it is windows composted, and it is the material is taken, and it is vermicomposting beds has been created, and for 30 to 45 days a proper watering is done, and the first harvest will be coming around 45 days to 60 days. After that they will be removing the top layer of the soil, because once the earthworms started eating the compost, they will be leaving away the vermin, the what you call the material which comes out of this vermin. So that has been slowly collected, and in the next picture you can clearly see this is the market waste that has been collected, and a bed is formed, and the end product you can see clearly, the final product, the black color. You see, this is the thing in the picture. You can clearly see the collected vermin, ma'am, the organic compost, the vermicompost. And this vermicompost is bagged, and it is also sold to the local farm. Hello, ma'am.
Can you please wind up? Your time is up. Can you please wind up your presentation? Your time is up.
Okay. So this is my, I took a thing of collecting the sample and done some kind of analysis, ma'am. So these are my analysis results. And the results says that as per the NUIRI, a small thing, in the compost, we should have a micro and macronutrients, the nitrogen, phosphorus, potassium, which are under the limits only. So as after Kenken, it was found that sample analysis much found to be satisfactory, because before we used to do just like normal degradation. See, if we leave the wet biodegradable waste normally also, it gets decomposed. But if you do in a proper scientific manner, the nutrients has been retained. So which in turn giving a very good quality to the composting. So the farmers started buying the compost from the municipality. So before that municipality, they used to give away the compost freely, just like a kind of advertising we are doing. So in the first year, they gave away the compost freely. Now even they are charging 30 rupees per kg. In the past, after implementation of this, ma'am, I can clearly say they said the income generation has been on 1.5 crore from dry as well as wet waste from past 5 to 8 years. They have been generating an income. This is the income, ma'am, 1.5 crores. And the conclusion is, if any scientific method for the core vision of the waste management in India is to process the waste in a more scientific and technological manner, so which gives increased value of extraction and recycling. That is the main thing behind the solid waste management. There is a need of cultivative community awareness, because without a community awareness and general public awareness, this field or this sector can't be a successful thing when compared to other things, because a public and various stakeholders are various stakeholders are involved in various stages, ma'am. So what I conclude is a continuous awareness in the form of IEC activities, a continuous scientific or any kind of technological development, any kind of methodology or model should be evolving over the time, which makes the solid waste management a kind of more presenting way, at the same time income generating. That is the main thing. And with this, I conclude my thing, ma'am. Yes, ma'am, I have concluded.
Thank you, sir. Thank you, Mr. Suresh Sampati.
Thank you, ma'am.
Now the room is open for QA session.
Hello.
Yes, ma'am.
Is there shall I ask a question, sir?
Yes, ma'am. Go ahead, ma'am.
Actually, I am working in the KVK system, that is a Kerala Agriculture University. So
Yes,
instead of this centralized waste management system, can you popularize a decentralized mode of waste management system, sir? Just like the same methodology, this is vermicomposting or microbial composting, you can popularize the technology that is in the decentralized mode, so that every household they can compost their waste in their place. That is
Yes,
there is
yes, ma'am. Even
that
this
is
municipality, just like I said, IEC activities, information, education, and communication. These guys are even the municipal people, authorities. Even they are motivating the what you call these residents welfare associations, apartment people will be there, ma'am, because that's a kind of a block. So what these guys are doing, a apartment with at least a 20 to 30 households, they are requesting the householders to maintain a what you call the biocomposting, ma'am. Just like bring all your biodegradable waste, put in a container, and we are going to give kind of incentives.
okay. I think it is for in terms of economics, everything it is very good. We can
Yes,
compose the things at the source itself. There is no
yes.
transportation.
And
Okay, okay, sir.
the thing is, like India, it's, ma'am, as you know, it's not that easy, because now the people are getting a kind of more awareness. These in the cities they started to, just like in Bangalore. I have seen some in Bangalore and some in Mysore. I have seen, ma'am, the apartment people, they itself, they start just with the coordination with the municipal authorities or the corporation people. They're giving a kind of incentives, just like if you guys do one, we are going to give a kind of tax and tax exemption for 5 to 1%, something like that. Because people in India, we just look for a thing which what we get. All the thing that always we think what we are getting in turn. So that
Along with this composting, they can also do their farming in their balcony and
yes,
utilize this compost effectively. Because
just like
we have conducted so many demonstrations in our municipality. So I am just
thank you,
okay,
thank you,
sir.
ma'am.
Okay. Thank you, sir.
Any other question?
Presentation was very elaborate and good.
Thank you,
And
ma'am.
you were speaking about two types of processing, like vermicomposting and bacterial composting. So
Yes,
as
ma'am.
ma'am was telling, that is to treat the waste where it is been generated. It is a new initiative,
Yes,
and
ma'am.
mostly in Kerala also many NGOs are working for that, means for the flats and all. They are processing the waste there itself, and they are using for this as a biofertilizer and all.
Yes,
So
ma'am.
the presentation was good and elaborate.
Thank you.
It was means like Swachh Bharat Abhiyan. It was like a part of, we are hearing a part of Swachh Bharat Abhiyan to make India
Thank you.
clean. So very good.
And one thing, ma'am, this sector, just like as you guys were saying about the microbial, because I have seen the plant genetics, microbial genetics. Now, this is a kind of our science, because I'm a science student, to be frank. Mine is MSc Environmental Science, and as a science student, we these means the doctor, everybody, we'll be doing our lab, ma'am. But this sector, it's a public-oriented thing. We should literally wash the brains of the public and motivate them in such a way that tomorrow, if we don't follow the proper rules and regulations, we end up in putting our own garbage in our own room. That will, the stage will come where they will start charging it, ma'am, to be frank, in the near future.
Yeah. Many initiatives are on, but how efficient it is working is
Yes, ma'am.
controversial. That's the thing. So the people who is generating the waste has to be first educated. Then only the from the grassroots level this can be solved. This problem can be solved.
Yes, ma'am. And I got a query in the chat, ma'am. Mr. Khalid Mir, he was asking, I have noticed that blue, red, and yellow, black color dustbins are a success in government. Yes, sir. Because the thing is, as long as there are continuous IEC activities and continuous motivation and information, this will be a successful. Once the what you call awareness is dropped, the people start doing everything. That is the major difference between a developed. I'm not going to any kind of different way. Other countries in India, sir, it's in the hands of the public. Once we start doing a kind of activity, stick to it. That's the main thing. Okay, sir. Why?
Suresh Sambadi, your presentation was very nice.
Thank you,
Very
ma'am.
informative too. Like I too want to add Sumi ma'am about the Alapura model, internationally acclaimed thing.
Yes, ma'am.
It's like a general pattern of solid waste management, and you can concentrate more on plastic waste degradation and the
Yes.
newer trends like soldiers fly, some kind of feed
Okay.
production.
Okay,
And
ma'am.
a lot of areas are there to explore with waste
Yes, yes,
management.
ma'am.
And I too agree that this is going to be a big issue to be rectified in the near future like that.
Thank you,
I
ma'am.
wish all the success to you. It's a good
Thank you, ma'am.
presentation. Thank you.
Thank you. Thank you, one and all.
Mr. Suresh Ambatti, there is another question in the chat box
Yes.
by Visha Harini. Sir, have you personally visited the sewage treatment place?
Yeah, I worked with, because that's what I worked in the as a part of my project. I went over there. I have seen everything. That's what I am very confident what these guys were doing it.
Okay, sir.
Thank you.
I think there are no more questions in the chat box. Now we will go to the next participant.
Okay, ma'am. Our next participant is OPF 16, Srikala CJ.
Yes,
OPF
Sharon,
16.
Sharon, we are
Yes, ma'am.
presenting for her. We are presenting
Okay,
for
okay, ma'am.
Srikala.
Okay, ma'am.
Okay, Mr. Suresh, if you are stop screen sharing, I can. Yeah, thank you.
Srikala, ma'am, are you there?
Hello. Hello.
Hello.
Hi.
Hello.
Srikala, ma'am, are
Yes,
you
yes,
there?
I am here. Yes, yes, I am here.
Ma'am, is it visible to you?
Yes, visible.
Okay, ma'am, you can start your presentation.
Good evening, everybody. I am Srikala, research scholar of SD College, Alappuzha, under the guidance of Dr. Sidili. Today's my topic is population dynamics of plant growth promoting fluorescence pseudomonas from rice bowl of Kerala, India. Plant growth promoting rhizobacteria. These are the bacteria that enhances plant growth and yield by various plant growth promoting substances as well as biofertilizers. Selected my study area is Kutanad. Kutanad is a unique agroclimatic region in India. It's one of a important feature of the Kutanad is the paddy cultivation is done in below sea level. It is one of the important Ramsar site. India's cultivation is promoted through the use of chemical fertilizer and pesticide, and this toxic chemical has polluted the environment and magnified through the biological system. And this ecologically sensitive area need protection. It is also important of microbial biodiversity. Next objective of my study first is the isolation and identification of Pseudomonas strains from acidic soil of Kutanad. Next objective is to check the antagonistic effect and pesticide resistance of Pseudomonas strains against fungal pathogens. To estimate absorbance of citrophore production of pesticide-resistant strains in iron-deficient succinate medium. To compare plant growth promoting efficiency of strains. Methodology. First methodology is the isolation. Isolation done by according to Cappuccino and Sherman. Identification is based on 60S the rDNS gene sequencing and in vitro antagonism done according to Utkade and Rahe. Seed bacterization studies done according to method of Dr. C. Dilip. The identified after morphological biochemical evaluation and the selected strains are subjected to their antagonistic studies, and then these are selected strains are carried out this secondary metabolite production, that is siderophore production. The fluorescent pseudomonas were examined for the siderophore production in 4 iron deficient synthetic media. The 4 iron deficient synthetic media chosen, however, they are Barbaya and Rao, Mayer and Abdullah, Filson and Linus, and Sher and Becker. The siderophore production was noted at 2 temperatures, 25 degrees Celsius and 35 degrees Celsius, and pH 7.2. Thus, the siderophore produced this bacteria are subjected to the seed bacterization. Rice is selected for these studies. Mainly UMA variety MO16 is used for our work. Rice were surface sterilized with the 0.5% solution of the sodium hypochlorite for 3 to 4 minutes. Then it washed it repeatedly and dried in sterile air for 1 hour. Strains were Kingsby broth in a shaking incubator at 28 plus or minus 1 degree Celsius for 24 hours. The seeds were then treated with 1% CMC without bacteria strains serve as the control. The bacteria seeds were then used for field experiment to check the plant growth promotion. The results. First step is the soil sampling. After soil sampling, the colony forming characters are not soil sampling. The soil characters are noted, and then isolation. Isolation is by pore plate method. Out of 97 strains from acidic soil of the Kuttanad, only 21 strains were selected on the basis of their morphology, physiological, and biochemical characters. Out of 21 strains, 6 strains were show significant antagonism and plant growth promotion. This table shows the result of the soil sampling. Randomly selected 10 study sites are selected for the soil sampling. This soil sampling result shows that the pH, the soil pH is very acidic in nature. The colony forming unit of rhizosphere and rhizoplane bacteria before flood shows before flood and after flood. The before flood shows that there is a decrease in the colony forming unit. After flood, there is an increase of the colony forming unit. The colony forming unit are observed in Na agar medium and nutrient agar medium and KB medium. The KB medium shows the luxuriant growth than Na medium. Then this bacteria, identified bacteria, are subject to their molecular identification by 60S rDNA sequencing. The table shows the result of the antagonism. The antagonism against the fungus, Aspergillus fumigatus, Aspergillus flavus, Aspergillus tubensis, Fusarium oxysporium, Rhizotonia solani, and Aspergillus niger. The strain RP12 and MIP show significant antagonism than other strains. The standard strain FPO4 also compare with these strains. The FPO4 is also isolated from earlier from Kuttanad. These are the PCR profile of the RP12, BPS, SCP, H1, RRP1, and MIP. Selected antagonistic strains were used for their siderophore production in synthetic iron deficient synthetic succinate medium. Preliminary examination of the siderophore production reveals that 6 Pseudomonas isolates were show fluorescence under UV light and capable of producing siderophore. The secondary examination of the detailed study reveals that the strain RP12 and the MIP in 4 iron deficient synthetic medium shows the absorption peak near 404 plus or minus 5 nanometer. It indicates that they produce hydroxamide type of siderophore. The siderophore produces the most efficient antagonistic strain, MIP and RP12, were then they're selected as for field experiment. This the figure show the field studies. These are the plant growth promoting parameters. The treatment, the strain RP12 plus rice Uma variety is selected for field study. Show the highest germination percentage, shoot length, root length, fresh weight, and dry weight than control and FP of our reference strain. These are show the plant growth promotion under field conditions, the yield of the strains. Summary. Major objective were to explore the PGPR wealth of the acidic paddy field of the Kutanad in 2 crop seasons. Soil sampling was carried out systematically from randomly selected areas of Kutanad. The pH of the soil sample indicate that they are very acidic in nature. The isolates are broad spectrum of antagonistic plant growth promoting property that can be developed as a pest control, biocontrol agent, and also biofertilizer for sustainable environment. Conclusion. The present study concluded that Pseudomonas isolates, RP12 and MIP can be used as a biocontrol agent, plant growth promoter for sustainable agriculture. It is the need of our to develop and discover various ecological niches for beneficial microorganisms. A proper scientific use of chemicals lead to an era of agriculture prosperity. These are my references. Thank you.
Thank you, Srikala. Over to Dr. Sumi and Dr. Sudha for any questions and for giving valuable suggestions.
Okay, madam.
Srikala, it sounds to be a complete work. And only 2 questions I want to know, because you studied about this biocontrol agent. So did you use any this rice pathogen or pathogen related to disease causing pathogen related to rice to study the biocontrol capacity of the your isolated Pseudomonas fluorescence? And second thing was whether you compared. So because I know Kerala Agriculture University, they are giving, commercially they are producing this Pseudomonas fluorescens and providing to the farmers as a biocontrol agent. So whether you compared your Pseudomonas, the potency of your Pseudomonas fluorescens with that of KUs or any such reputed agencies. So that
yes, madam. My strain is also antagonistic to Rhizoctonia solani. Is procured from Himtech, Hyderabad.
Okay. So you haven't included that part in this study and in this paper, no? So, okay. Whether you have done or not, that's right. The other
yes,
one,
yes,
whether you have compared with a commercially available Pseudomonas fluorescens.
Yes, madam. FPO4 is a reference strain.
Okay. No,
FPO4.
Okay, okay. So a reference strain from where you have procured this one?
It's also earlier isolated from Kutanad, and its secondary metabolite chemical composition were early discovered. This FPO4 state can be used against the
whether it is a commercially available thing or like that. That's what I want to know.
The particular strain is isolated from my guide, Dr. C. Dilip.
So is this your PhD work?
Yes, hello, madam. Also, it is available in NIST,
Is this your
Trivandrum.
okay? Is this your PhD work?
Yes, yes, madam. It's a one part.
Okay. So it's just a suggestion. If you want to get, means it should be comparable with a similar product also. So that will be good for your work. That's all. Otherwise,
Madam, can I explain?
Means you should compare with a commercially available pseudomonas fluorescens from a reputed agency. So that will be very good for your
Okay,
work.
okay, ma'am.
That's all.
Thank you.
Otherwise, it's a complete step. Okay.
Okay, ma'am. Thank you for valuable suggestion.
Preetala ma'am, it's a nice presentation.
Thank you.
I am answered actually. My question was also on the reference train only. And I want to add one more point, like usually consortium is working more. So instead of going for a single organism, you can go for a consortium. And second is, you said that the soil is acidic already.
Yes, madam.
Is it so? And in
Yes.
Kutanadi area and all, we can see the accumulation of ferric, like reddish color scams will be there already. So I wonder why siderophore again is acting on ion accumulation and enhancing PGPR, since it is acidic environment. And you have studied only siderophore. You are explained, you might have studied, but explained only about the siderophore production and the PGPR activity or increase in plant growth. Is it so? So
Yes.
you can add more points to be considered.
Okay.
Is it clear? My suggestion
Also,
like
yes, madam. Is a part of my PhD work. I do the pesticide, check the pesticide resistance of these strains, and also root colonization ability of these strains, and multiple pesticide resistance ability of these strains.
Okay, that's good. In your study, what you have explained here, you said about siderophore. Siderophore is ion accumulating proteinaceous compound, and in acidic pH, already ion accumulation is there in paddy fields. So, what is the relevance of this pseudomonas again adding? But you have scientifically proved it, so it's a good work anyway. Consider all these things.
Thank
Nice
you, ma'am.
work.
Thank you. Thank you for valuable suggestion.
Thank you, Dr. Sumi and Dr. Sudha, for your valuable suggestions to the participant. There is no questions in the chat box, so we will move to the next participant. Sherin, shall we?
Sharon has connectivity problems.
Yeah, Edna, can you carry? So next
Okay, ma'am.
participant is OPF 17, Edna, Beanie Francis.
Okay, OPF 17, Binny Francis. Binny, are you here? Binny Francis.
Hello. Hello.
Yes, you are audible.
Yes, ma'am, I am here.
Okay, can you share your screen?
Yes, ma'am, I now share the screen.
Okay.
Can you?
No, yeah,
See
it's
it?
come. It's come. It's come. Can you put it in the
Can I
slideshow?
start? Okay, ma'am.
Can you put? Yeah.
Okay, ma'am. Okay, ma'am. Okay, ma'am. Can you see?
Yes, it is visible. Yes.
Ma'am?
Yes. Now
Can
you
I
can
start?
start.
Can I start?
Yes, now you can start.
Okay. Good evening to all. Good evening to all. I am going to present on the topic antimicrobial activity of spices and herbal extracts against Vibrio parahemolyticus and Escherichia coli. This work was done under the supervision of Dr. A. Mohammad Hatta, Professor, Department of Marine Biology, Microbiology and Biochemistry. First, introduction. Food antimicrobials, including food preservatives, have been widely used to control foodborne pathogens. And in the recent years, due to an increased concern about the synthetic nature of many antimicrobial agents on the customer's health, there is a rising demand for the natural food additives. Therefore, we prefer for herbs and spices. They've been widely used for centuries as traditional medicines, flavoring agents, and preservatives. In the present study, we are focusing on two organisms, E. coli and Parahaemolyticus. They are naturally present in the aquatic environments. Among these organisms, Bio-Parahaemolyticus is a halophilic gram-negative seafood-borne pathogens, and they are the leading cause of seafood-associated gastroenteritis or the consumption of raw or undercooked seafood. And E. coli, we are now the normal microflora of digestive tract of warm-blooded animals, but the strains with acute virulent factors, they have the ability to cause diarrheal disease in healthy humans. These organisms, they entering into the food processing environments contaminate raw materials. In food processing environments, the bacteria, they expose levels of nutrients. And in the case of seafood industry, seafood is a highly perishable commodity. They provide a suitable medium for microorganisms. After attaching these microorganisms onto the food contact surfaces, they form biofilm. In the food industry, the biofilm formation is a serious problem. And in literature, it states that Vibrio parahemolyticus and E. coli, they are capable of producing distinct type of adherence factors. They enable them to adhere to the surface and initiate the biofilm formation. And the biofilm associated Vibrio parahemolyticus and E. coli, they not only causes the food outbreak, they also causes the economic loss to the seafood industry. Therefore, my objective of my study is to investigate the antimicrobial activity of spices and herbal extracts against E. coli and Vibrio parahemolyticus. They are isolated from food contact surface in processing plants and attempt to identify the most active of the plant extracts. Next one, materials and methods. A total of 34 spices and herbs was used for the present study. There are two kinds of spices, fresh spices and dried spices. If the fresh spices, they clean, washed, crushed with mortar and pestle, and these extracts were sealed and filtered, sterilized. In the case of dry spices,
Hello, you're not audible.
hello.
Now your voice. Yeah. Hello.
Hello, ma'am.
Yes, yes.
Am I audible?
Yes, you are.
Next, antibacterial activity testing was done using agar-well diffusion method. For this method, we are making a lawn culture on the Mullen Hinnon agar place. Then agar wells were prepared using a micropipette. 100 microliter of the spices, herbal extracts were added to the wells, and these plates were incubated at 37 degrees Celsius for 24 hours. And the diameter of sono inhibitions was measured and represented in millimeter. And this sono inhibition greater than 60 millimeter were considered as the extracts with highly activity, and between 12 and 16 millimeter they are considered as moderately activity, and less than 12 millimeter they have no activity. These test microorganisms were also tested against commonly used antibiotics, and these comparisons were also done. And the antimicrobial activity of selected spices, that is, the extracts with prominent activity, were done at various concentrations against E. coli and Vibrio parahemolyticus. Next one, results. The table 1 shows the antibacterial effect of active extracts of spices and herbs in vitro. From this table, we can see that Garcinia gamiguta, both fruits and seeds were used for the study. They both shows antimicrobial activity against E. coli and Vibrio parahemolyticus. And then pomegranate also shows the antimicrobial activity. And also the allium set.
PPT and audio no. PPT is not visible and it's not audible.
Hello, Beanie. Are you there? It's not audible.
It is not been seen.
Presenting, ma'am. Am I
Yeah.
audible?
Yeah, now you are audible. Please share your screen. Can't see your screen.
Ma'am, I'm still presenting up to results. Is it audible?
Yes, but the
Hello, ma'am?
slides are not visible. The slides are not visible.
Now. Ma'am, is it?
Yes.
Is it safe? Can I start?
No. Yeah,
Bini
yeah,
ma'am,
you can
can you
start, but
please
you have to
share your
share your
screen?
screen.
I have already shared, ma'am. Yes. Are you see my?
Can't view your slides. Yeah, now it's come.
Hello.
Yeah, now it's come.
May I start from results section?
Yes, yes.
Yes,
Please
Bini,
make it
you can
fast.
start from results section itself.
Okay, ma'am. The table 1 shows antibacterial effect of the accused extract of spices and herbs in vitro. Among these 34 spices and herbs tested, Yersinia gummi, that is Kodampuli, and then Punica granate.
Bini, 1 second. Would you put it in slideshow mode?
Okay, ma'am. Okay, ma'am. Is it see? Can you see it, ma'am?
Yes,
Hello?
it is visible, but not in the slideshow
Hello?
mode. Yeah, now it's visible.
Now it's visible.
Okay. Among these extracts, cloves, pomegranate, then garlic and garcinia, they shows antimicrobial activity against both the E. coli and Vibrio parahemolyticus. Next, the table 2 shows the comparison of diametrosomal inhibition of spice and herbal extracts with antibiotics. From this table, we can see that the antimicrobial activity or diametrosomal inhibitions of these extracts were more or less similar to the antibiotics. Next one, table 3, antimicrobial activity of selected spice against E. coli at various concentrations. The extracts with most prominent activity were tested at different concentrations against E. coli and Vibrio parahemolyticus. From this table, we can see that in the case of Garcinia extract, at low concentration, they do not show any antimicrobial activity. The activity will be lost. And in the case of pomegranate and cloves, they shows antimicrobial activity at low concentration also. Next, table 4 shows the antimicrobial activity of selected spices against Vibrio parahemolyticus at various concentration. In the same, in the Vibrio parahemolyticus also, the Garcinia shows in the low concentration they have no antimicrobial activity. But in the case of pomegranate and cloves, at the lower concentration also they shows antimicrobial activity against Vibrio parahemolyticus. These are the plates showing the diameter zone of inhibitions against both E. coli and Vibrio parahemolyticus. And from this work, we can conclude that these cloves and pomegranate can be used for further study for the antimicrobial control of E. coli and Vibrio parahemolyticus in food industry. Thank you. Hello, ma'am. Hello.
Thank you, Bini. Over to Dr. Sumi and Dr. Sudha for any questions and giving valuable suggestions.
Yeah, Bini, it's good work. Like it's very clear that you have used
thank you.
Spices for preventing effect of Vibrio parahemolyticus and E. coli. So I have no questions. Actually, it is very explanatory, self-explanatory. It is very simple presentation. If Sumi have any questions, she can ask. It's done. Good presentation.
Thank you, ma'am.
Sumi ma'am, over to you.
Are you hearing me? Getting me?
Yeah, it's very feeble voice actually.
Hello.
Hello.
Hello, ma'am. Hello, ma'am.
Am I audible?
Yes, ma'am, audible.
So, what type of extract did you take?
Ma'am, this is only a preliminary work. I have, for preliminary work, I have used aqueous extracts. For further work, I have to use solvent extracts.
Okay. So, why did you choose these two organisms?
Sorry, participant.
Hello.
Sumi, sorry to interrupt.
Ma'am.
So,
Hello,
sorry, Sumi.
Yeah, tell me.
Hello,
Disha,
ma'am.
Tell me.
We have to break up here for the keynote speaker. Our keynote speaker is ready.
Okay, ma'am.
We will wind up this session here, and after the keynote speak, we can continue it.
Okay,
We
ma'am.
have one more talk, I think. We have one more.
One or two. I think two more papers are there.
Okay, we can continue after the.
Okay, we will do that.
No problem, ma'am. No problem.
We can start our keynote speak.
Okay.
Okay, ma'am. Thank you, ma'am. Hello.
Hello, am I audible to you?
Yes, ma'am.
Hello. A warm good evening to one and all. I welcome you all to the Day 2 keynote session of Bio-Inventiyon 2020, Today International Web Conference on Recent Advances in Biosciences. Myself, Dr. Shaini N.K., Coordinator, Internal Quality Assurance Cell, and Assistant Professor in Microbiology Department of Safi Institute of Advanced Studies. Today, we have an eminent scientist, Dr. Vijay K. Juneja, here to share his ideas and knowledge on the topic microbial modeling of pathogen in foods and its application in assuring food safety. Now, I would like to welcome and invite Dr. Jisha MS, Conference Convener and Honorary Director, National Institute of Plant Science Technology and Professor, School of Biosciences, to introduce our keynote speaker. Ma'am, please, ma'am. Jisha ma'am, are you there? Hello? Hello? Jisha ma'am?
Ma'am, Disha ma'am is there. I think she's having some network problem. She's trying to talk. Hello.
Okay, ma'am. Vijay sir, we are having heavy rainfall in this area, so kindly cooperate with us, sir. Maybe that is why we are facing technical issues, sir.
No problem. Can you hear me?
Yeah, sure, sir. Very clear and loud.
Okay, Shabana, can you hear me?
Yes, ma'am. There is an echo. Please mute one device, please.
Yes,
Okay.
ma'am. Please continue.
Okay. Today we are fortunate to have an eminent scientist, Dr. Vijay Juneja from USDA Agriculture Research Service. Dr. Vijay Juneja is established microbiologist with extensive experience with aerobic and anaerobic
Can you please,
pathogens.
ma'am, can you please mute any one device? No, now you're not audible. Ma'am, you can switch off the speaker of the laptop and speak through the mobile.
Ma'am, please unmute yourself, please. Jisha ma'am. Are you able to unmute?
Unmute. Ma'am, can you unmute from your phone?
Ma'am, if not, please rejoin once, ma'am. It's okay, ma'am. The climate conditions are getting harsh. It's okay. But if you are not able to unmute, please rejoin. Just try, please, or just unmute. I'm sending the request to unmute.
Okay, can you hear me, Shabana? Hello?
Yes, ma'am. Yes, ma'am. Clear and loud. Please continue, ma'am.
Voice is clear?
Yes, ma'am. It is clear.
Sorry, sir. Sorry for the interruption.
That's okay.
Today we are fortunate to have an eminent scientist, Dr. Vijay Juneja from USDA Agriculture Research Service, Winmore, USA. Dr. Vijay Juneja is an established microbiologist with extensive experience with aerobic and anaerobic foodborne pathogens, broad interest in basic and applied research in food science and technology, including food microbiology, molecular biology, biochemistry, predictive microbiology, and risk assessment. Currently, he is a lead scientist of a research project on predictive microbiology at the Eastern Regional Research Center, ARS, Winmore. He received a degree in veterinary medicine from JB University of Agriculture and Technology, Panagar, in animal science, and PhD in food technology and science, both from University of Tennessee, Knoxville. Dr. Juneja is among the world's leading authorities in food safety research. He has developed a nationally and internationally recognized research program on foodborne pathogens with emphasis on microbiological safety of minimally processed foods and predictive microbiology. His research program has been highly productive, generating over 300 publications, including over 175 peer-reviewed journal articles, 47 book chapters, including 10 in the Encyclopedia of Food Microbiology, and is a co-editor of 8 books and one for food microbiology special issue entitled Predictive Modeling in Food. Currently, he serves as an editor of Food Science and Technology and is a member of the editorial boards of Journal of Food Protection, Foodborne Pathogens and Disease, International Journal of Food Microbiology, and Frontiers in Microbiology. He has served in leadership positions in several professional societies and is a recipient of several awards. It's very difficult to list out all his achievements. With immense pleasure and honor, I would like to inform you all that I have also achieved a golden opportunity to collaborate with Dr. Juneja for the Global Initiative of Academic Network Program initiated by MHRD, Government of India. We are looking for the further orders. We are very fortunate to have you here as our lead speaker. We are eagerly waiting to hear from you, sir. He'll be talking on microbial modeling of pathogens in food and its application in assuring food safety. On behalf of organizers, I am welcome you to this international webinar, Bio-Inventiyon 2020, Recent Advances in Biosciences, and inviting you, sir, to deliver the talk. Thank you, sir.
Thank you very much. Thank you. Can you hear me?
Okay, sir.
Yeah, so I'll go ahead and share the slides.
Okay, sir. You're audible, sir.
Is it okay?
Okay,
Can you
okay,
see the
sir.
slides?
Yes, sir. Yes, sir. Yes, sir. We can see.
Okay, I can go ahead and start now.
Okay, sir.
Okay, good morning from here. It's 8 o'clock and I know it is evening over there. So good evening to all of you. And first of all, I would like to thank Dr. Jisha for inviting me to give this presentation. It is my great pleasure to interact with you and to talk about microbial modeling. I believe most of the professionals who joined are from Kerala, and I was visiting Kerala 3 years ago on site for sightseeing, and it's one of the most beautiful states in India, and I plan to visit Kerala once again sometime. And I was born and brought up in New Delhi, and this was only twice when I grew up. People never used to travel in India, and so this was only twice I visited South India. In 2008, I was invited by CFTRI Mysore when Dr. Prakash was the director of CFTRI, and this so 3 years ago I visited Kerala. So what I would like to talk today is microbial modeling of pathogens in foods and its applications in assuring food safety. Of course, I'll be talking basically about what do you have to consider to develop a predictive model, and what are the various steps in developing the models, what are the different kinds of model and the applications of the models. And before I forget, I need to mention to you, I listened to the previous presentations with great interest, and I have published series of papers on natural antimicrobials, including 1 or 2 papers on spices, and I have several review articles on natural antimicrobials. And most of the time I use these natural antimicrobials in developing the predictive models as one of the food formulation factors. So certainly there is a great opportunity to collaborate and interact and strengthen the working relationship with the researchers at your university. And one of the goals of this presentation is to initiate and ultimately strengthen the working relationships between the US Department of Agriculture and your university, or strengthen the working relationship between the two countries. So this is the Eastern Regional Research Center where I work. It is located 150 miles from Washington, DC, and 110 miles from New York City. If ever you are in the eastern part of the United States, you are most welcome to visit us and see our state-of-the-art resources. This building was originally built in 1940, but it was completely renovated about 15 years ago. So we have all state-of-the-art facilities inside, and we have about 80 to 90 PhD level category 1 scientists who are working on various projects, and 60 to 70% of those researchers are working in food safety. So as you know, I work for the US Department of Agriculture. I work for the ARS of the US Department of Agriculture Research Service, and researchers conduct research on meat, poultry, and egg products. And the way we ARS scientists in the USDA work is that we already have the funding from the Congress, but we continue to maintain dialogue with the regulatory agencies as well as with the industry to discuss technical matters and emerging issues, as well as to understand and define the specific problems and high priority research needs. And so our research project, which we develop, or all the research we do, it addresses critical food safety issues, challenges, and concerns of the food industry and regulatory agencies, which in other words, we certainly have to show an impact of our research. You know, the Congress will not give us money to do research if we are not able to show the impact or be able to enhance the safety of our food supply. So this is FSIS of the USDA, Food Safety Inspection Services. They are basically regulatory agencies. FDA, you must have heard, Food and Drug Administration, and they also are, this is also regulatory agencies and deal with products with less than 3% meat or less than 2% poultry, and food additives and packaging and labeling, etc. Then Centers for Disease Control, you must have heard a lot about the CDC because of the ongoing COVID crisis. It's basically they deal with the outbreak investigations, for example. And then EPA that is concerned with the drinking water. So here I have collaboration in the last more than 3 decades. I have collaboration with researchers for and all over the world. And this is a good opportunity to strengthen or initiate and ultimately strengthen the working relations or collaborations with your university. So feel free to contact me if you have any question with regard to food safety or food safety microbiology. And if I don't have the answer, certainly I will dig out the answer for you and provide you with that, you know. So how safe is the food? We take it for granted that our food is safe for human consumption. However, foodborne illness can affect anyone, and certain categories of certain groups of individuals are more at risk of getting foodborne illness, and these include the pregnant woman, young children, older adults, all those individuals with suppressed immune system, which means those that are undergoing chemotherapy, for example. So if you fall into one of these categories, or if you care at home for someone who falls into this category, you need to take make sure that they take special precautions while handling food. So foodborne illness is certainly a significant public health problem and accounts for 2.2 million lives annually, and 1.9 million of those are children. In most of the cases, the illnesses occur due to unintentional errors during production, processing, or handling of the food product. And in the United States, there are about 48 million cases, 130,000 hospitalization, and 3,000 deaths. And of course, in the developing countries, there's much higher prevalence and not well documented, which we all know. So food contamination creates enormous social and economic burden on communities and their health system. And the economic cost has been estimated to be $152 billion. And this includes the cost due to reduced productivity, lost wages, pains and sufferings, medical costs, food recalls, and as well as the lawsuits, which means anybody can file a lawsuit if they get sick after consuming a food that was contaminated with pathogenic microorganism. So number of the fruits and vegetables have also been the causative agent and then foodborne illness associated with fruits and vegetables have been on the increase and result in 1.2 million cases and 7,000 hospitalization and about 130 deaths. So one of the major concerns is the contamination of food is to control the traditional as well as emerging pathogens. And this includes the pathogens that are of increased virulence at low infectious doses, or those pathogens that are resistant to antibiotics or food-related stresses. All the food formulation factors certainly constitute a stress to the foodborne pathogens and renders the pathogens more resistant to the subsequent homologous or heterologous intervention treatment. And also there's a concern with regard to the contamination of the food and water with the enteric pathogens of animal origin. So underlying forces that may make foodborne illness more of a problem to years in years to come are the new generation food, refrigerated, ready-to-eat food products with extended shelf life. Emerging pathogens continue to emerge, and sometimes they increase their virulence. Improper food preparation, storage, and distribution practices, we all know that sufficient evidence exists that most of the time the food poisoning occurs because of those who are handling the food are not properly trained, insufficient training of retail employees, for example, and increasingly global food supply and increase in the number of people at risk because of the increased lifespan of human beings. So globalization means the governments of all countries have the first and foremost responsibility to ensure the safety of our food supply, and the rest is, of course, to facilitate the trade. This is a slide I borrowed from somewhere, and it says that this is bread, and some of the ingredients for this are obtained from all these different countries. And for example, honey from India and guar gum from India, and there are several other countries where the ingredients are obtained for any particular product. So globalization and international trade has significantly impacted the food is produced, processed, and handled and distributed, and presents us as new challenges as far as the food safety or the quality of the food is concerned. Governments all over the world are taking series of initiatives to protect our food supply, and food supply throughout the entire food chain from the production to consumption. Series of new initiatives are being taken. We don't want to take any chance that the food we consume should make anyone sick. Another problem is because increase import and export of different raw materials for foods or for or the fruits and vegetables, for example, there's certainly an increased concern with regard to cross-border translocation of pathogens. And another concern that appears to be is that because in some of the developing countries, not much is known about the harvesting practices, for example, or the quality of the irrigation water that is used. So governments have been focusing more on specification of the raw materials, which we know, reducing the maximum residue levels, or interacting more with the third-party providers so as to achieve control throughout the entire food supply chain. Also, there is a need for uniformity in food safety standards around the globe. So here I've listed some of the most common foodborne pathogens, E. coli, and the previous speakers talked about the efficacy of the spices and herbal extracts in controlling E. coli. Listeria E. coli is especially concerned with ground beef, and then we have Listeria monocytogenes concerned with ready-to-eat foods, Salmonella, again, a pathogen associated with poultry products, along with Campylobacter jejuni, Staph aureus, which produces is certainly a concern because of the heat-stable enterotoxin that is elaborated by this particular pathogen, and Yersinia enterocolitica. There are some of these pathogens, they can grow even at the refrigeration temperatures. We all know that, like Listeria monocytogenes and probably Yersinia. So these are the spore-forming pathogens, Bacillus and Clostridium. Those who are aware of the scientific literature in food safety, they must have known that I have published series of papers on the spore-forming pathogens, particularly Clostridium botulinum, C. perfringens, B. cereus. So this is just a slide I wanted to show about the pathogen, what models can do and how can you use the models, and then I will get into depth with regard to the predictive microbiology. So the user can certainly use our user-friendly program to find out what would be the microbial levels at different storage temperatures. What level of salt would be required to restrict microbial growth to a certain predetermined level during storage for 1 week at 10 degrees. You can input your criteria and you can estimate or predict the behavior of the pathogens under your conditions, for example, of storage or salt level. And what would be the microbial levels during 1 week of storage? 7 degrees if the pH is increased from 5.5 to 5.8. And this is with regard to if you're developing a new product, for example, or if you're trying to extend the shelf life of your existing product. So what is predictive microbiology? The science of predictive microbiology is based on the assumption that the microbial behavior is reproducible and can be quantified by characterizing the factors that affect the growth, survival, and inactivation of the pathogen in the food product. In other words, this is a way to describe the microbial behavior by mathematical equations, but we don't want to scare anybody with the mathematical equations, so we convert those equations into easy to use, user-friendly software that can be used by anyone, even with working in the food processing industry with a high school background. So the models, why use models? To describe the experimental data aimed at predicting the microbial behavior within the range of experimental values tested. We interpolation, which we call, to predict outcome of a process, number of log reductions, to predict for untested conditions. For example, if you have collected the data at in a particular food product with a salt level 1, 2, and 3%, you can certainly predict the behavior of the pathogen at a salt level, for example, 1.28%. Or same thing holds true with regard to the pH. If you have collected if you have developed a model and after collecting the data at a pH level of 5.5 to 6.5, you can certainly predict at any of the levels of pH within this lower and the extreme levels of the particular parameter you have used in the lab to collect the data. So these models which we use are extensively used as a building blocks in microbial risk assessment. And this is the area I have been working for a long time. And I know this is an area that is of great interest to researchers as well as to the Food Safety Authority in India. So there's certainly an opportunity to interact and strengthen the working relations with researchers who have been working in this area for a long time. So this is a good example. I want to show you how these are used as building blocks in risk assessment. For example, in the processing industry, there are a number of unit operations. So you can use a model for each particular unit operations to determine the final levels of pathogens at the end of the, for example, process. So initial contamination, you know that based on your knowledge of the initial prevalence levels, you know as a result of pasteurization, the levels will decrease. Then during when the handling and preparation of the cooked after pasteurization in the food processing plant, for example, when you are cutting a ham, for example, on a cutting board, there's certainly a transfer models which are used to determine if the slicing blade is contaminated with pathogens, how many slices of that particular product, cooked product, would be contaminated. Then distribution and retail, you know, there would be chances of growth, particularly if the product is mild temperature abused, and of course, the consumption. So first of all, you identify a research problem. And then once the research problem is identified, which means you keep yourself abreast of the industry needs and regulatory agencies needs, so that the data that you collect and develop the models can be used by regulatory agencies for developing their regulations for the safety of the processed foods, for example. And the risk assessment that is done by the risk assessors and food safety managers, you can certainly identify those data gaps and then conduct the research and develop the models so as to address those data gaps, so that the risk assessors and food safety managers can determine the impact of food processing and handling conditions on the risk of foodborne illness. So once you have know that, because each research which you do should have an impact. Without an impact, I believe we are wasting all the resources, all the resources and the funds. And if that happens in my agency, that if the research is not having an impact, that project is abolished even. So then you develop an experimental design, which I can talk for hours, but because of the limited time, we will not be talking. So there are different kinds of experimental design: incomplete factorial or complete factorial or central composite design. And then you develop you work with your students and with the technicians to collect extensive data in the lab. And then of course you develop the primary model, you develop the secondary model, and then once the model is developed, you certainly look at the predictions and you validate the models, which is, I think, internal validation, external validation. You can get the commercial products to validate, or you can use the data collected in the lab that was not used in developing the models to validate the prediction that is obtained from the secondary modeling. And certainly you develop then the user interface, which is a user-friendly program. So you select a target organism. Which means you certainly want a target organism or the pathogen that was isolated from foodborne illness, or you use the clinical isolates you have to use, or you have to use the isolate that are representative of a particular meat species. You don't want to use an isolate for developing a predictive model for poultry product if that particular strain was isolated from a produce or from mutton or from beef or from pork. So certainly you have to select the target organism, inoculate into growth medium at the levels that is prevalent in the real world, and then you can store under controlled conditions. How do you, and then you sample at relevant periodic intervals. How do you decide that? Based on our modeling program, you can get a good idea of what should be the sampling time of your experiment so that you don't waste time in the lab and come up with no results, or the pathogen has grown to such a high level that you are not able to catch sufficient data points during the lag phase or the exponential growth phase. Then you certainly construct a model, you validate the model, and then you refine the model. If you need help in developing the model, certainly I'll be more than happy to analyze the data and develop the model, provided you have collected sufficient data in the lab. And I can certainly help your young researchers to understand the modeling, how to collect the data and develop a model so that they lead the research once people like me retire and turn over a new leaf of their life, So there are a number of factors you have to consider. You have to microbial strains, we just talked, factors, what levels. You don't want too much salt levels in the product because that would have an adverse effect on the organoleptic attributes, on the sensory attributes, and also with regard to the concerns with regard to hypertension. You can also look at the indigenous microflora, inoculum level. We already talked. You certainly, growth phase or the stationary phase is very, very important. For example, it is well known that pathogens should be in the stationary phase when you do the thermal inactivation, because they are considered to be highly heat resistant. Prior history of the microbe, that is very, very important area of science, and number of young researchers do their PhD thesis on this particular area, where you can, it is sufficient evidence that how the microorganism is grown, or the prior history of the microorganism before the contaminates the food certainly has a role, as well as the behavior of the pathogens in your food product of interest. Dependent variables, you certainly have to have an idea, which is very simple, whether you're looking for D values or generation time or lag time of the pathogen, or growth, no growth in case of probability-based models. Substrate, certainly it's very simple. You have to decide that which pathogen is a concern in which product so that you conduct your studies in that particular product. So this is a very simple slide where you see the dependent variable we talked earlier, that is the D values or lag time, for example, or stationary or the generation time, for example, and then you have independent variables, what is your salt levels or and the pH levels or the natural antimicrobial like spices, for example. If you want to develop a model assessing the effects and interaction of various spices or herbal extract or essential oils with the interaction of salt and salt, for example, so as to quantify a reduced level of salt that would secure microbial stability of a food product. And I have series of papers where we have assessed the effects and interactions of oregano, cinnamaldehyde, thyme, and pomegranate, and different green tea or different kinds of teas, and several other natural antimicrobials and develop models accordingly. And most of these models are available in our pathogen modeling program that anyone can use and very, very user-friendly. So primary models, the first you develop for growth, survival, inactivation, and there are some of the examples I have given, and the simple is of course the D values, and then the secondary models, which is the Z values, for example, which you all know is Z values, describing changes in parameters values of the primary model with changes in environmental conditions, which means how a pH or water activity or level of spice will affect the growth of the bacteria. You can develop that, see that, and then tertiary models, which I'll be talking, the user-friendly pathogen modeling program. So these are the different kinds of models, which I will not go in details, and because of the limited time, otherwise empirical models where polynomials, for example, is a good example of an empirical model. I'll talk in a minute. And most of the models in the past in our pathogen modeling program are based on response surface modeling, mechanistic model, and then we have deterministic model that always gives the same output when provided with the same input. Probabilistic model, a mathematical model that gives a variable output when provided with the same input. And here in this case, we have variability model. So empirical models, these are the first generation models. They are very, very easy to use, but they don't have any biological meaning. So whether you use a compass or logistic, in all cases, you get a Can you hear me? Can you hear me?
Yes. Yes, sir. Yes, sir.
I got a note here that the host has muted you. Okay,
it's mistake,
anyhow.
sir. It will continue, sir. Please continue.
I understand it happens, okay, so there's a lag phase. These are, so regardless of what empirical model you use, you always get a sigmoidal growth curve. And you see, this is a good example that a researcher or a technician who is working on the computer can use a compass or logistic. You always find a sort of sigmoidal growth curve, and we mu max it, and then a n max, which is a stationary phase, or lambda, the lag phase. So this is I think I missed a slide. This is the square root. This is a square root model that was, we have used it extensively in our dynamic models. If somebody is aware of my publications, where the square root of the growth rate is a function of temperature below the optimum temperature, and Rutkowski in 1982 found that square root of the growth rate gives a linear relationship with temperature at suboptimal conditions. And then later on, Rutkowski, after a year, expanded that model to cover the entire growth temperature range. And I have been using the square root model for decades in developing the models for the fate of the surviving spore formers during the cooling of the cooked products. For B. cereus, Clostridium botulinum, and Clostridium perfringens, I have developed series of models, and because the USDA regulations Appendix B, you can see that regulations require that it's the processor responsibility that the surviving spore former should not grow by more than 1 log. So researchers have been, so the processors in the food processing industry and the retail environment have been using the models which we have developed to demonstrate to the food safety inspections that during cooling deviations due to electrical outage or equipment, refrigeration equipment failure, or in case of customized chilling of the product, there was less than 1 growth increase. And it's very, very simple and easy to use those models. And we have the models for all cured and uncured meat products. So this is a 3-step process to develop a dynamic model. First of all, you generate the primary growth parameters from a series of experiments conducted under isothermal conditions. Then parameters of the primary models are used to develop the secondary models within a temperature range. As a function of temperatures, the parameters of the secondary model with a set of differential equations that describe growth under small increments of time is used to develop a model applicable to the entire growth temperature range of the pathogen. This is just a 3-step process, the way we develop the dynamic models. So this is a differential equation. I'll not go into details. So this is another cardinal parameter model. It is the key feature of this model is that secondary parameters have a biological meaning, and it is based on the assumption that the cumulative inhibitory effect of the secondary parameters is additive and ranges from 0 to 1 if the growth conditions are optimum. Each term has a value equal to 1, and so in that case the growth rate or the mu max is equal to optimum growth rate. And this shows the models for the combined effect of water activity, pH, and temperature. This is another response surface model I talked earlier, which is a purely empirical model, and to assess the effects and interactions of multiple food formulation factors like temperature, pH, water activity. It's very, very easy and simple to use, but and you can fit extensive experimental data to the multiple linear regressions. And then the disadvantage is that the terms do not have any biological meaning, and sometimes the extensive number of interactions gives a false estimate. This is, I was talking earlier, the models, transfer models that are developed. We used to develop that in the past, not anymore. For example, if the blade is contaminated, how many slices of a particular cooked product would be contaminated with that contaminated blade. There are some models we have in our pathogen modeling program, and there's a probability-based models that can also be developed based on what particular level of sodium pyrophosphate, for example, and sodium lactate, or what level of salt would lead to no growth of the pathogen. So, and also we have time to inactivation models too, which I have not shown here, which means under the unfavorable or the harsh condition of the food product, how long will it take for the pathogen during storage to get to below the detection limits. D values, Z values, and F values, I'll not talk in details because of the limited time. So D values is the time in minutes at a particular temperature required to destroy 90% of the viable cells or spores of a particular organism. It's the measure of the heat sensitivity of the pathogen and is obtained by plotting log number of survivors on the y-axis and time at an x-axis. And this is known as the survivor curve. And D value is numerically equal to the number of minutes required for the survivor curve to traverse one log cycle, and mathematically it is equal to the reciprocal of the slope of the survivor curve. This is a thermal test time curve. Z values or Z values, what we call in India, is the change in heating temperature required to change the D values by 90%, where you have the log D values here and the temperature on the x-axis. When a microbial population is heated at a particular temperature, cells die at a constant rate. And this traditional first-order kinetics model has served the food industry and regulatory agencies for decades. However, deviations in the log number of survivors with time are frequently observed, which means there's a shoulder followed by a linear decline in the log number of survivors with time, or this is the convex survivor curve, or concave survivor curve, and this is the shoulders. TL, when the population remains equal to the initial population as a result of heating at a constant temperature, followed by a linear decline in the log number of survivors, and then the subpopulation of more resistant bacteria, that is the tailing of the survivor curves. So there are a number of theories that have been put forward to explain the deviation in the log number of survivors with time, and first one is the experimental errors and artifacts of the heating and plating technique, which means clumping of small number of cells or spores may protect those spores, rendering them more heat resistant. Variability of heat sensitivities in the heated population, which means there may be a distribution of heat resistances within a population of microbial cells. Heat adaptation during heating, this is also an area that has been researched a lot and is an area that is of great interest, which means acquisition of heat resistances as a result of heating, and as a result of the low temperature, long time cooking of the food products, there is sufficient evidence that the pathogens becomes more hardy or more heat resistant, similar to the concept about what we learned in 9th grade, survival of the fittest in our biology class. So this is a linear model that is used, which the two discontinuous equations are used to model the shoulder and the first decline, and this model does not take into account the subpopulation, the more resistant bacteria. And there's a TL where the population remains equal to the initial population, followed by linear decline in the log number of survivors with time. This is a logistic model, and in brief, I would like to mention that F1 is the fraction of initial population in the major population, and TL is the shoulder or the lag period, which means if there is no subpopulation, then F1 becomes equal to 1, and the right side of the equation becomes 0, and then you get a shoulder or the first decline. And if there is no shoulder, you see in the experimental data you have collected in the lab, then the equation essentially becomes a straight line. It's very, very easy and simple to use, and there are a number of, anybody can understand in a few hours and use these models to generate the parameters of the primary model. This is a Weibull model. This is used in addition to the linear regression, which you all know. It takes into account both the convex and the concave survivor curves. However, it overestimates under the dynamic conditions. This is a good example of the effects of interactions of various food formulation factors that can be used to collect the data, and certainly if you use the various levels of each of these parameters in your formulated product in the lab, you certainly have to develop a fractional factorial or design or a central composite design so as to limit the number of experimental experiments you have to do in the lab. If you want a complete factorial design, then you will get probably more than 1,000 experiments, and it's not practically feasible for anybody to conduct 1,000 experiments and repeat it 2 or 3 times. And so the D values that are obtained are converted to natural logarithm form and analyzed by second order response surface regression to develop this kind of multiple regression equations. And so this is a good example where we have at pH 6.5, you see the observed D values are 57.7 of Clostridium botulinum non-proteolytic Clostridium botulinum spores in ground turkey. And the difference between proteolytic and non-proteolytic Clostridium botulinum spores are the Clostridium botulinum type A and B spores are highly heat resistant. and they are resistant to environmental stresses, but they do not grow at temperature less than 10 degrees centigrade, whereas non-proteolytics type B and type E, which is primarily found in seafood, can grow at temperatures as low as 3.3 degrees centigrade. So, but they are relatively less heat resistant, so it is practically feasible to destroy those spores by heat and utilizing and rendering the spores sensitive to the lethal effect of heat by the use of all these additives in the food product. And here I want to show is that the D values can be decreased from 57 to 40 and 39 to 32 by altering all these parameters in the food. And of course, we expect the expected D values to be higher in all cases so as to be more conservative. And this is a good three-dimensional graph where I'm trying to show the effect of the pH, salt, and sodium pyrophosphate and how the D values can be decreased from 49 minutes to 20 minutes. So these models we develop are extensively used in developing HACCP plans, developing critical control points, establishing critical control limits, and validating HACCP plans, and determining the effect of process deviation, evaluating the relative severity of the problems caused by process deviation, and finally, they are used to estimate the expected effectiveness of the corrective action as a consequence of deviation from a critical limit. And so they are extensively used in the food processing industry. Why process deviation occurs? Because of the electrical outage, or because of the refrigeration system failure, or because of the thermal processing equipment failure. So the processors can use our model to determine if the product is safe for human consumption or not, and to determine compliance with the regulatory performance standards so that the product is approved by the inspectors for transportation, distribution, storage, or handling in the supermarkets or by consumers. So like I mentioned, they are used in developing HACCP plans, in building blocks in microbial risk assessment, in shelf life and safety predictions or estimation under a variety of different storage conditions. They are used as a scientific basis for developing regulatory performance standards. And like I just mentioned about less than 1 increase of Clostridium perfringens. So food processors are used, and for the LM in ready-to-eat foods, controlling growth, they can be, their models for all these things and can be used for determining compliance with the regulatory performance standards, as well as these models are used as data gaps in microbial risk assessments and building a risk assessment for a particular food product. Time function integration, which is an area that is of great interest in India, I haven't heard, and certainly these models can be used to estimate the behavior of the pathogens. And the research that is being conducted is to correlate the fate of the bacteria with the kind temperature sensors on the packaging material of the food product. Education, the professors or the faculty in the universities can use our very, very easy, user-friendly program to demonstrate to the growth how bacteria grows and dies in a food product in their food microbiology class. And they can be used by the students in the lab to determine what should be the frequency of sampling of their experiments in their food product, because they can get some idea and some estimation under the similar conditions from our pathogen modeling program. So for product development, usually small processors are interested in developing a new product that can be marketed easily because of the consumer's demand, so they can estimate how long this product will last at 8 degrees, for example. How is the formulation stable for the desired microbial shelf life? Because suddenly we want absolute no growth of the pathogen in the food product. And certainly if the spoilage of the food product occurs first, that is not a concern, because nobody is going to consume that product. Whereas if the pathogen grows first, or if the toxin, deadly toxin, neurotoxin is elaborated in the food product, that is a serious concern. So we do have the models for the deadly neurotoxin producing pathogens, the spore formings in the food product. What storage temperature should be used, what microbial specifications are realistic. So these are the different links of our different user-friendly pathogen modeling program. And this is a predictive microbiology information portal. This is a comprehensive website that brings together predictive models for foodborne pathogens, research data, relevant regulatory policies and guidelines, and other useful food safety related links to diverse and numerous resources associated with the models. I have 15 minutes, so I will try to conclude my presentation in 15 minutes. So this model provides information on the underlying principles of predictive microbiology, as well as the use and interpretation of the predictive models, so that the processors can alter the process parameters and achieve the microbiological safety of the food products. It's very, very easy and simple to operate, and you can, anyone, any student or those with a high school background in the food industry can understand how to use it. You just click start, it gets you to this particular slide. And this is COM-based, for example. This is tutorial. This is COM-based is, I'll get into a tutorial where you get step to step. If you click this, you can get step-by-step instructions on how to navigate the portal and obtain the necessary information. This is a very, it gives you a very simple, basic concepts on food safety microbiology regarding the pH of the food, how bacteria grows, how bacteria dies, and what are the different kinds of models. You can get all that information if you click this. This is a pathogen modeling program, which I'll talk in a minute. You can access the online version or the standalone version through this link also. This is a resource locator, which is very, very important. So this is the COM-base. It's a relational database on food microbiology. It contains thousands of data sets on growth, survival, and inactivation of foodborne pathogens under environmental conditions that are relevant to food processing operations. And so you can just input your search criteria and you can retrieve all the raw data that matches your search criteria. We encourage the researchers who publish the papers to deposit their raw data in COM-base, and we host this COM-base at our center. And if you submit any query in the COM-base, there's a link. It would be directed to my email address, and I would be glad to respond to you if you have any problem in retrieving any raw data. So the use of COM-base certainly eliminates unnecessary repetition of experiments. It reduces redundancy in conducting biological studies. It certainly increases the efficiency of the research efforts, and of course, it increases the safety of our food supply. So if you click this, resource locator, you can select the organism, you can select the food type, then it gives you what are the resources that are associated with your food and the pathogen of concern, and then these are the sources of the various resources. So here we have selected ready to eat, LM in ready to eat meat products. There are 60 resources, and here we have selected 7 models. They are listed over here. If you click this, you can get little more information about that particular model from here. So here we have the 4 sources of those resources listed over here. It's very, very easy and simple to operate. So here you can access either the online version or the download version of the pathogen modeling program. This lists all the models that are available in the online version of the pathogen modeling program, and I will not read all these models for you. You can just read them later. These are the heat inactivation models we have, and for all the different pathogens, we have the models. We have survival models, like I mentioned earlier, time to inactivation, and we have the transfer models we discussed earlier. So this is a good example that where you can certainly, with this drop-down menu, you can alter the pH, sodium chloride level, sodium nitrite level, and then temperature you can also alter. And then you can see the lag phase, which is 1.3 hours under these conditions of pH and salt levels. And you can get and this is the upper and lower confidence limits. You can also get the data in a tabular form if you click this. So it's you just have to click calculate growth data and it gives you. So this is a lag period, which is 3, and the generation time is 0.3. And here with no lag, you can also get the predictions too. Time for increase from 3 to 6 logs, and you can also look at the table. We certainly recommend processors to use the upper confidence limit so as to be more conservative. We don't want anyone to get sick. This is again the heat inactivation model under a variety of pH and sodium chloride conditions, where you can see the minutes are 59.8, and with the increase in temperature, the minutes are decreased to 16 under the same and food formulation conditions. So spore formers, these are my few last slides, and I will have certainly more than 7 minutes for question answers. So these are the spore formers, and we have to take it for granted that the spores are present in the food product, and we have to take it for granted that the time and temperature is not adequate these spores, because if you cook for a sufficiently high temperature for a long time, that would destroy the texture of the meat, for example, or the flavor or the nutrient value of the food product. So these spores will survive the time and temperature, because the aim of the cook in the retail food environment is to make the food palatable and not to destroy the spores, to destroy the vegetative cells of the foodborne pathogens. So you have to control the growth of the spore formers in your ready-to-eat food, because the presence of spores, they will not make you sick, provided unless the spores germinate and produce into very sufficiently high levels, or they produce deadly neurotoxin. The difference between Clostridium botulinum and Clostridium perfringens is that the Clostridium botulinum produces deadly neurotoxin, which is heat labile during the multiplication of the vegetative cells, whereas in case of Clostridium perfringens, you ingest a very high level of vegetative cells of Clostridium perfringens. That cells, once they survive the acidity of the stomach, they sporulate in the intestine, and during the process of sporulation, they produce enterotoxin that leads to the symptoms of food poisoning. So USDA requires that the product should be cooled in a total period of 6 hours. So this is what I'm talking about, the applications, how these models are used. It's just one example. When sometime if I give workshops on the modeling, then I give a number of different scenarios, and those who are attending the workshop, they have their own laptops, and they use their laptops to predict the behavior of the pathogens under a variety of different conditions, and that is a good sort of exercises for them to learn to the use of the modeling program and to understand the behavior of the pathogen in the food product. So like I mentioned earlier, this is a USDA regulation to cool the product in a period of 6 hours. However, the product cooled in a period of 15 hours because the cooling deviation occurred. There was a power electrical outage, which commonly happens in India. And the processors are then in big problem how to ensure how to make sure that the product is safe for human consumption. So you can use our models. You can go to our cooling model for C. perfringens. Why C. perfringens is used? Because this is the most rapidly growing foodborne pathogens. And if you control Clostridium perfringens, you don't have to worry for the deadly neurotoxin producing Clostridium botulinum, whereas the regulation clearly states that there should be less than 1 log growth of Clostridium perfringens and absolute no growth of Clostridium botulinum surviving spores, because it is not even known until today what is the minimum level number of vegetative cells of Clostridium botulinum that can elaborate deadly neurotoxin. So you go to your Excel spreadsheet where you have collected the cooling curve. You just click that and the data comes over here. You don't have to type the data in the model, and then you can look at the calculate growth data. If you click that, that gives you to the next screen, which is the net growth, which is in this particular case is 6.54, and upper confidence limit is 7.1. What does it show is it's more than 1 log. So the product is not fit for human consumption, which means that the product should be destroyed. There's another scenario where the product cooled in a period of 12.5 hours, and it entirely depends how long for this cooling of the product in 12.5 hours the product stayed within these temperature range, because of these are the ideal growth temperatures of the pathogen, and the removal of heat from any cooked product is exponential in nature, which means as it gets close to the refrigeration or the room temperature, the rate of decrease of the temperature suddenly decreases. So this is again the go-to modeling program. You can retrieve the data, and then it says retrieve the cooling curve you have recorded in your processing plant, and then you can see the net growth is 0.59, which means the product can be released if the product would be safe for human consumption. And here is my contact information. I have another scientist who works on with me, Tom Oscar. He works on various, also develops the models, particularly more of the risk assessment sort of models, and I develop the models that are used as a building blocks for microbial risk assessments. And if you have any questions, feel free to contact me. And I think I have concluded my presentation 7 minutes before an hour that was scheduled for my presentation. So if you have any questions, feel free to contact me now, or you can either send me a text message, a message on my cell phone, or you can call me on my cell phone, or you can send me an email. And sometimes, since I get a number of queries, emails, so sometime if you don't hear from me, you can always send me a reminder. This concludes my presentation, and thank you very much.
Thank you very much, sir. So you have given us a very valuable talk, sir. It was a very wonderful session, very informative and enlightening. So thank you very much, sir, for sharing your valuable ideas and also your precious time with us, sir. So we have some queries in the chat box. Can I read out, sir?
Sure, go ahead and read those.
So the question is from Najma. She has asked, the processed foods are made through so much scientific technique and testing and trials. Still, in a long run, it has found to be cause of various diseases like cancer. Why such wound become a bane? That is for query, sir.
Because the product is temperature abuse. Number one thing, the product is not supposed to be sitting in your car after you buy from the refrigerated cabinets in the grocery stores for several hours, for example. And you have to take the food and refrigerate as soon as you take it, and you have to use the product within the designated shelf life of the food product. So if the product is temperature abused, then certainly that is a major cause for food poisoning. And sometimes what happens is that consumers are unaware. They take the ready-to-eat food, for example, from the refrigerator and start slicing them on a cutting board that was previously used for raw food. So they contaminated the ready-to-eat food. So, or their hands were contaminated. So you have to take it. What I think is most of the time, the processors or the food is processed is safe for human consumption because they have followed all the guidelines and the protocols and what you call the standard operating procedures or standard processing procedures, and the food is inspected. And this is all I talked. And certainly it's a temperature abuse of the product or mishandling of the food product by the consumers that leads to food poisoning, in my personal opinion. And certainly the models which we have shown can be used by processors, by the regulators, or by the small processors, or by the food industry who do not have much resources to do microbiological studies in the lab. And moreover, it is not easy for them to introduce the pathogen in the lab. And also, so these models can be used to determine if the product is safe or not. I hope this answers the question.
Yes, sir. Thank you. Thank you, sir. Now, another query, sir. It is from Jyoti Otageri. She is asking, what are the ways of controlling spores in food items other than heat?
That is very good question she asked. First thing is, spores are present in the food product. You have to take it for granted the spores are present. Each and every soil or mud sample that was ever examined was found to contain the spores. And practically, it is practically feasible for us as a food microbiologist to destroy the non-proteolytic Clostridium botulinum spores by food formulation factors and rendering the pathogens more sensitive to the lethal effect of heat. However, if these non-proteolytic Clostridium botulinum spores, if they cannot be destroyed because of the adverse or negative impact on the product quality as a result of the harsh intervention technique that is used, you have to build hurdles in the food product so that the pathogen would not grow. And you all, the one who asked me this question must be aware of the golden rule of pH 4.6. So if the pH of the product with regard is less than 4.6, certainly the proteolytic Clostridium botulinum type A and B spores will not grow. So those products are, they can guard against those spore formers. So either you have to destroy the pathogens in the food by building hurdles, or you have to use the heat. And certainly sometime different combination of technologies are also used. For example, the high pressure can be used to induce germination of the spores. And those germinated spores can easily be destroyed by less time and temperature employed during heating. And I have several models on spore formers in our pathogen modeling program, and at least more than 100 papers on spore formers as far as their growth or inactivation in the food is concerned, and also their fate in sous vide processed foods, which is cook and bag food products. So certainly, if you have any questions with regard to the modeling program, or if anyone has any questions with regard to the research on spore formers, or they can certainly feel free to contact me. Any other questions?
I have a question, Dr. Junaid. Oh, sorry.
Go ahead. Who is this?
Hi, Dr. Junaid. Nitin here. How are you? Good morning.
Nitin is a young researcher from Hyderabad. He finished his PhD over here, and currently Nitin is a postdoc in Florida, in Texas A&M. Texas A&M. He's a very bright, intelligent young researcher. All the young researchers from India who have come to the US to conduct research and get PhD in food safety are highly successful. There is absolutely no doubt in it. Go ahead, Nitin. What is your question?
So I have two questions. One is very generalized because what are your opinions on the modeling on these low moisture foods, because the bacteria doesn't grow, but they are able to survive for longer duration. But do you think the modeling can be applied? And my second question is, it is based on the publications, like most of the papers what I've saw. Do you develop these models, or you just you use the survival or reduction data of a processing technique to predict the data?
No, we have all these models which we have published in the last decades. They are all developed by us, and those are the models that are available in our USDA pathogen modeling program. And to answer your questions about low water activity products, certainly the concern, if we did develop a model for beef jerky, we got sufficient money from an industry, and they wanted us to develop a model for beef jerky where we looked at the reduction in the water activity during the process of cooking in the oven, and we developed a model for that. And very soon, in 2 months, we will have that model available in our pathogen modeling program. It was published 2 years ago. And you certainly, like I said, we do have the time to inactivation models, which means under the harsh conditions, the water activity is too low. I assume that the pathogen survive. If there's any pathogen surviving, that would be inactivated during the process of storage. And spore formers, of course, they are not a concern under low water activity if they are not, if they don't grow. And with the pathogens, what is ideally needed is there's absolutely no growth of the pathogen. And in low water activity product, most of the pathogens would not grow, I would assume, because of the effect of the water activity in combination with spices, for example, that the previous researcher talked. And so my question to you is, if anyone in the audience, if they're interested in my review articles on natural antimicrobials, feel free to contact me. My excellent review paper on natural antimicrobials appeared in annual reviews of food science and technology, maybe 5-6 years ago. Any other questions?
Hello.
Okay.
Hello.
Yes.
Sir, Gaurav here, sir.
Hi, Gaurav, how are you? Gaurav is again a good researcher. People like me, being the editor of the journal, I find out who are the excellent researchers in the world. And Gaurav is a young researcher from CFTRI, and 6 years ago, he reviewed a paper for my journal and the review was excellent. When I saw the excellent review, I decided to contact him by sending a personal message to him. And I asked him, who are you? Tell me about you. I have never seen such an excellent review coming to me from India. Then I got to know him. So I know people from all over the world, very well known to me, those who are ambitious, hardworking, and really have a curiosity to or have an interest in research and science, you know. Gaurav, go ahead.
Welcoming Dr. Gaurav to this webinar.
Yes,
Dr.
sir.
Gaurav.
Sir. Yes, sir. Sir, actually, thank you, ma'am. Sir, actually, I have one question. Like I have one question from the one soya industry. They are purchasing the seed from the farmers, and because of in the seed they are containing the lot of variation in the microbial load. And this company is making the soya flour. So what is happening actually? They are telling we are making the 20 tons at a time. They are running the process of 20 tons in a one tank itself for making the flour, and they are getting the so much contamination of E. coli. And the reason they are telling why they don't want to increase the heat and why they don't want to go for the any other process, like they are telling we are the small industry, we cannot purchase the higher technology. Another thing, they are preparing the soya flour, they are getting the microbial contaminations. And if they increase the heat to reduce the microbial contaminations, their nitrogen solubility index will be reduced. So in that case, what could be a best practice? Can we use these predictive models to solve these type of the issue in the industrial scale?
I think what probably needs to be done is to specify the microbial levels in the seeds, you know, because you have to use some non-thermal technologies to reduce the microflora, indigenous microflora in the raw materials. Okay. What I would think that needs to be done, and there are several non-thermal technologies, but it's not easy to use those because of the limited resources by the small processors, you know.
Yes, sir.
So I think specification of the raw materials is one thing that can be done to limit the number of microflora.
Yes, sir. Because they are getting a lot of variation in the microflora, whatever raw material they are getting, because different farmers, they will harvest in the different conditions. So much microbial, sometimes they will get the less microbial load, sometimes it is too high.
This is what I mentioned initially. One of the concern is the kind of farming practices in the developing countries, and there are probably no uniformity in the laws that what kind of irrigation water they should use or what kind of harvesting practices they should have and what kind of temperature control they should have. And certainly one of the ways to do is to use some non-thermal technologies to decrease the indigenous microflora like UV light or something like that, you know.
Yes, okay. Thank you.
But we don't have any models on this particular product at this moment because I work for the US Department of Agriculture, and we deal basically with meat and poultry products and with produce safety also.
Yes, sir. Thank you, sir.
Thank you. Any other questions?
Yes, sir. So we have some few more questions, sir. Thank you, Dr. Gaurav and Nitin, for joining us today.
May I
I
ask a
think
question?
Dwivedi, Dr. Dwivedi is asking question. Sir, please continue.
Sir, good evening. Myself, Abhishek Dwivedi from Banaras Hindu University, and I am working on foodborne fungi and mycotoxins. So you have discussed a lot about bacterial system where modeling you have applied. So I wish to apply this model in my research also. Can we apply all those model in fungus, which are foodborne?
Well, that's a very good question you have asked. So before I answer your question, you work with Dr. Dubey?
Yes, sir. He is my PhD guide. I share lab with him, actually.
He is also one of the most intelligent professors I have known in India who does an excellent reviews for my paper. And when I handle his paper, they are just accepted without any changes because he sends only perfect papers to my journal. Give my regards to him. I know him very well, and I'm very much impressed with the kind of research he does in Banaras Hindu University. He's one of the researchers I'm very much impressed in India who really is very dedicated and does an outstanding job. And also he's a very good writer too. So to answer your questions, no, we do not have any models for fungi in our pathogen modeling program, but if you Google or if you review the literature, there are several models that have been developed for fungi. Okay. And I work for USA, like I said, we do on research that is based on what kind of food poisoning outbreaks we are getting in the US. Yeah. So, but there are several models for the fungi, and I would encourage you to look at those models. And there are other softwares developed from Europe. And I'm sure some of these softwares would have the models for fungi. I would hope so, but I don't know if, like the USA models, which we have, they are free of cost, and some of the European models, you have to subscribe to get an access to those models. You may want to look into that.
I will see, sir. Thank you so much.
Good. So you are a PhD student?
No, sir. I am assistant professor here, but I did PhD with Professor Dubey. I share lab with him.
Okay, wonderful.
Work together.
Wonderful.
Yes, sir.
Okay, good.
Okay, thank you, sir. Now we have few more queries in the chat box, sir. So one is from Visha Harini. She's asking, these days the manufacturers add preservatives so that the packed food doesn't get spoiled. What is your thought on this?
The manufacturers add preservatives, and what did you say after that?
Preservatives so that the packed food doesn't get spoiled. So what is your thought on this?
What doesn't get spoiled? What did you say?
So she wants to know what is your thought on this, adding preservatives to the products.
Certainly, it's a good question. Certainly, there has been a lot of concern with regard to the adverse effect of chemical synthetic preservatives. Even in the US, the green labeling and people want the foods without any preservatives. And some of these chemical preservatives used in the food certainly have been shown to have some negative adverse health effects. So I would think that one should go for the natural preservatives in the food products, and so as to have no concerns with regard to the adverse effects of the food. And that is the main reason the models which we have developed, we have not developed any model. Our goals of these models is to reduce the levels of salt and substitute the reduction of the salt with natural preservatives and develop a model. And certainly we optimize the conditions so that one of the limiting factors with the use of the essential oils is that the flavor. So we don't want too much of these natural preservatives also in the food product. And now you must be aware the use of nitrates is also being limited or not being used because the natural source of nitrates that is being used to replace sodium nitrite in the food product, in cured products to control Clostridium botulinum. So certainly chemical preservatives, in my personal opinion, should not be used in the food products. Does this answer my question?
Thank you very much, sir. Sir, the chat box is filled with appreciation note for your talk, sir. We are really blessed that we could get you today with us, sir. Thank you
very
so much for
much.
sharing your views, sir.
Okay, thank you. And then certainly I encourage the attendees of this session to look at my pathogen modeling program, both online version and standalone version, and if they have any suggestions so that we can make it more user-friendly, more easy to use, they can certainly contact me. And we have an excellent programmer in Canada who continues to update and this our modeling program. And in the coming 2-3 months, we'll have we provided funding to this programmer to incorporate 11 of our newly published models. And likewise, if anyone has any questions with COM-base, they can always send a query in the COM-base, and we would be glad to respond to that. Okay.
Sure, sir. Sure, sir. We are having food technology department in our college, sir. Surely we do that.
Okay.
Sir, any other queries? Any other queries?
I have something to talk about. Dr. Juneja, as you also aware that recently we formed the IAFP. I just want to talk about IAFP, and I encourage all the participants for the upcoming year, please join the IAFP. It's the best platform to get to know about scientists and young researchers to get, if they want to pursue their higher education. This is the best platform, and we have a Indian Association of Food Protection in North America. And please do get a membership as well. It's not that expensive, so that you can get more information about the food safety, because this is one of the best organization in entire world that all the scientists get get communications and etc.
Kindly share the links, sir.
Information, please let me know. Thank you.
Kindly share the links of the organizations in the chat box. Thank you.
Sorry, I cannot hear you, ma'am.
Certainly, what I would do is I will send an email to you, and it's an Indian affiliate of the Association for Food Protection in North America. About 10 days ago, we had a meeting when hundreds of people from India joined, and Nitin is now the new secretary of that association. So we encourage researchers in India to become a member of that so that we can strengthen the working relationship with India. And certainly you can go to foodprotection.org and you can look all about it. And if you have any questions, you can certainly contact me. And they have there are a lot of opportunities for researchers as well as students to come to the US to attend that meeting free of cost. For example, 2 of those are that this year, November 10th is the deadline for submitting symposium proposal. And if some symposium proposal is approved, I can help in getting funding for that individual to come to the US and to attend the meeting. And like this year, one assistant professor from NIFTEM, we got the funding for her and she was supposed to come. But the meeting was held last week. It was virtual, so she could not come, but IAFP is kind enough to keep those funds for her to come next year. This year, since the deadline is November 10th, I am submitting at least 2 researchers from India have contacted me, and I'll be helping them in submitting the proposals. And if at all it is approved, then this will be an opportunity for a faculty member or a student to come to the US to attend the meeting free of cost. In addition to that, sometime in January, February, we have the the IAFP meeting has student travel funds, when the students from all over the world can apply for that. They just have to write 1-page proposals. And if that is selected, then they can come here to attend the meeting free of cost. Like I have known people from Bangalore, from Hyderabad. They have come in the past, the students who applied for this travel scholarship, and they got that scholarship and then came to attend the meeting. And it's not a question of only attending the meeting, they can do a lot of networking over here too. So I encourage you researchers to look into this symposia proposals. Usually this time they're due on November 10th, the deadline, and look at the student travel scholarship sometime in January, February. And if you want to be involved with the student affiliate, with the Indian affiliate of this association, you certainly contact Nitin and he will have your name added to the mailing list at least, so that you can get an update. Already we have about 115 people from India in that mailing list. Thank you.
Thank
Any
you
other
very much, sir.
questions?
Thank you. Thank you very much for the information, sir. And Nitin, sir, we will look into it, sir, surely. Sir, we don't have any other queries in the chat box. So I think we are coming to the end of the session, sir. Once more, I would like to thank you profusely, sir. The chat box is filled with appreciation for your talk, sir. Thank you so much, sir.
Okay, thank you very much and have a nice evening.
Okay, thank you, sir. So this
Okay,
is the
bye.
end of the session.
Okay,
So this
bye.
is the end of the session.
Once again, sorry for the inconvenience, sir. And thank you very much for helping us for writing the GAN proposal. And actually, we were looking forward to be here. In October, we proposed the date, but due to the COVID pandemic, we couldn't. I think in the near future, I think we can associate, sir.
So the government of India will give you the money later on after the COVID crisis is over?
Yes, sir. Like that only they are telling now. We are
Okay.
looking for that.
Okay.
If it is positive, then we can again meet in Kerala, sir. At that time.
Yeah, certainly after the crisis is over and we are allowed to travel to India. At this moment, it's not allowed even to travel to India officially.
We are looking for that, sir.
Sure, sure. Thank you.
Thank you, sir. Thank you very much.
Okay, have a nice day.
Thank you, sir.
Jisha, ma'am.
Okay.
Are we entering into the validatory function, ma'am, now? Or should we give a break now?
Time now, it's we'll start at 8 o'clock.
We'll start at 8 o'clock. Okay. Then we'll take a 5 minutes break, ma'am.
Hello.
Sir, you will
Hello.
be here, no, sir? Vijay,
Hello.
sir?
Yes.
You will
Certainly.
be here in the validatory. Okay, thank you, sir.
Certainly, I can attend that. So this is going to be award ceremony, you said?
Yes,
Hello.
sir. Yes, sir.
Sure,
Hello.
I will attend that.
Hello. Hello.
Hello.
Hello, ma'am. This is Dr. Gauri from Odisha. Hello.
Tell Gauri.
Hello. Ma'am, when the feedback link will be shared?
Immediately after the validatory function.
Ma'am, in the email or in the chat box?
It's in the chat box. It will be given.
Chat box.
Chat box, okay.
Okay, thank you, ma'am.
Is
thank
Gauri
you.
from Bhubaneswar?
Yes, from Bhubaneswar.
Okay, I was invited to the KIT University a few years ago. KIT University. And are you from that university?
Yes, KIT University.
Are you from that
I
university?
am from Utkal University.
Okay, the
Sir,
KIT
I am from
University.
Utkal University.
The faculty in the KIT
Hello.
University, they know me very well.
Okay, ma'am. Sir, I saw you because I attended a conference at KIT University.
Okay, so you must have met
Hello.
me there.
Hello, Vijay. Hello, Vijay, sir. Hello.
Yes, did you meet me there? I was there
Yes,
a few years
I met
ago.
you there long time ago.
Yeah, maybe 5 years
Sir,
ago.
I am Dr. Gauri Shankar.
Okay,
Okay,
good.
I am Dr. Gauri Shankar. Okay, thank you, sir. Thank
Yeah,
you,
I
sir.
was
Sir, I am
talking
actually
about
plant
the...
taxonomist.
Okay, good.
Yes, yes. Okay, sir, sir,
Okay.
sir. Sir, happy to see you after a long time. Okay.
Okay.
Okay, okay, sir. Okay, thank you. Pranam, sir. Pranam. Okay. Okay, sir. Thank you.
Good evening, everyone.
Smita, we can start, I think.
Hello, Shaini. CP sir has joined.
Good evening.
Good evening.
Good evening.
Disha ma'am, what about Jayendra sir?
Jayendra sir is here.
Sir is here
Can you
too.
check?
Yeah, sir is here. So everybody is set.
Okay.
Sir, here itself. Thank you so much for keeping the right time. Dear delegates, please. I am handing over the mic to Mrs. Parabarami, the master of the ceremony. Paramam, are you there?
Hello, am I audible?
Okay, ma'am. Yes.
Okay, let's move on to the valedictory session. Okay. What you vividly imagine, ardently desire, sincerely believe, and enthusiastically act upon must inevitably come to pass. A very good evening to all the esteemed dignitaries, credited academicians, and delegates from all over the world. It has been a momentous day, and here we have reached to the valedictory session and the award distribution of the two-day International Web Conference, Bio-Inventiyon 2020. I'm honored to invite Professor Dr. Jisha MS, Conference Convener and Honorary Director, National Institute of Plant Science and Technology, Professor School of Bioscience, MG University, for the welcome speech.
A very good evening to respected dignitaries, faculty members, research scholars, and my dear students. Now we have launched the last session of the webinar Bio-Inventiyon 2020, jointly organized by National Institute of Plant Science Technology, School of Biosciences, and SAFI Institute of Advanced Technology. Last 2 days, we have witnessed many great research works being presented by talented faculties, researchers, and students. All works are really commendable and have added great shine to the conference. I would like to congratulate the winners. Actually, it was very tough to choose. The two very innovative and informative keynote addresses from the eminent personalities have added an icing on the cake to make this conference a grand success. I would also like to thank the organizers of this event who have been constantly working hard to make this web conference a grand success. A special congrats to Asha for designing the beautiful brochure, certificate, and timely publication of proceedings. Shabana Mol, the research director of SAFI, is the iron woman behind this webinar. Throughout the 2 days, from morning 8 a.m. to night 9 p.m., she was in the Zoom room for the uninterrupted sharing of the Zoom connection and also for sharing the slides without even having proper food. Both of them are alumni of School of Bioscience, MG University. Proud of you. Since it's a teamwork, on behalf of MG University, I would like to express our gratitude to all of these work behind it, my research scholars and colleagues, faculty and staff of SIAS for their help and support. And a special thanks to the principal Imbichi Koya on behalf of MG University for his wholehearted support and help. Would also like to thank my colleagues for chairing the session. Let me take this opportunity to welcome you all to the valedictory function of Bio-Inventiyon 2020. Now I am ending my duty. I extend a very warm welcome to Professor Dr. K. Jayachandran, Director, School of Biosciences, MG University, who is very supportive to all of our academic activities in spite of his busy schedule. I feel privileged to welcome our guest of honor, Mr. C.P. Kunju Muhammad, Secretary, SIAS, for this auspicious occasion. I also heartily welcome Professor Imbichi Koya, Principal, Safi Institute of Advanced Studies, to this valedictory function. We are also fortunate to have our both the keynote speakers, Dr. Keith Davis and Dr. Vijay Juneja, with us. Welcome you, sir, to this function. Finally, I would also like to welcome our dear participants to this valedictory session. Because of your kind cooperation, we could make this even a grand success. Once again, I welcome each and every one of you to this valedictory function. Thank you all.
Thank you, ma'am. Now I would like to invite the inspiration behind this event, Professor Dr. K. Jayachandran, Director, School of Biosciences, MG University.
Good evening to all. Respected Kunju Muhammad, our dear scientists Keith Davis and Vijay Juneja, Professor Jisha, Professor E.P. Imbichi Koya, Shabana Mol, Asha, and all participants. We have come to the end of this program. Recent trends in biosciences, excellently organized program. I'm really happy that School of Bioscience was also a part of that. So I use this occasion to congratulate our dear Professor Jisha teacher, for acting as a tool for making all this linking Safi Institute, linking School of Biosciences. Congratulations, dear Jisha teacher. And as far as the program is concerned, needless to mention, it was inclusive of all branches of life science. Everything was there. All applied subjects were there. Maybe either as a talk in the at the competition level, maybe as a talk from faculty, maybe as a talk from business scholar. So it included everything. I'm sure students might have enjoyed it. The most important advantage of program like this, it will add aptitude in research as far as the students are concerned. So nothing more to say about that, but still I think I have to mention specifically the two important keynote addresses given by Keith Davis and Vijay K. Juneja. One was about the concentrating on plant molecular biology, and another was predictive microbiology. We are really fortunate to get such kind of an excellent talk. And I also remember, as Dr. Professor Jisha rightly pointed out, the presentations during the competition, excellent presentations were there. I still remember Hari Shankar presenting a very good topic, and then about microRNA, psoriasis related gene functioning, and along with that there were talks on lectins, there were talks on fruit preservation. I'm sure the students might have benefited much from that. So we're really happy to be here during this function. Once again, I congratulate the organizers for making this event as a grand success. So thank you all. Thank you very much.
Thank you, sir. Next, I would like to invite guest of honor, Mr. C.P. Muhammad, sir, who is the Secretary and Manager of Safi Institute of Advanced Study, the Executive Director of KRS Group of Companies, and he is also associated with a lot of higher educational institutions across Kerala. I am honored to invite you, sir, to share a few words.
Good evening. Very good evening to all dignitaries and the participants of this webinar. I'm very proud being the secretary of SAFI Institute of Advanced Study. We could join with hands with MG University to conduct this wonderful 2-day webinar. And this is a valedictory function. I congratulate all the people behind this for making this function a grand success. Thank you very much.
Thank you, sir. A dream doesn't become a reality through magic. It takes sweat, determination, and hard work. Yes, it's a belief endorsed by Professor E.P. Imbichikoya, Principal of SAFI Institute of Advanced Study. May I introduce her for the valedictory address and also to announce categories, that is the oral and poster presentation.
Good afternoon. Good evening, one and all. The chairman of this function, Dr. Jayachandran, the director of School of Bio-Inventiyon, MG University. Dr. Jisha MS, the conference convener. Mr. C.P. Kunya Muhammad, the secretary and manager of SAFI Institute of Advanced Study. Dr. Shabana Mol, Dr. Asha, Dr. Shaini. And I'm very much happy to see Dr. Kate Davis and Vijay Juneja, the two important resource persons who presented their papers last 2 days. And my dear colleagues and attendees from the different part of the world and researchers, students, and my dear friends. I would like to congratulate both the team from the MG University and SAFI Institute of Advanced Studies for organizing this program in the most befitting manner. As this is a concluding session, just we can evaluate the sessions which was held 2 days. So I'm very much happy to understand that all the sessions were highly beneficial to the participants, especially the keynote address by Dr. Kate Davis and Vijay Juneja. Good number of papers were presented. About 59 out of 75 registered papers were presented in this particular web conference. And in addition, 14 poster presentations were also made by the faculties and also the researchers. We know that is the syllabus or the curriculum of the universities are not competent or it is not up to date to discuss the advances in the field of biosciences or any field of academics. This type of conferences and workshops will definitely enrich the knowledge and also the spirit of research by the faculties and the researchers, and it will inspire those who are actually doing their PGs and after their PG. It will inspire, these sessions will definitely inspire those who have completed their post-graduation to do the researches in the different area of biosciences. And both the sessions and also the papers, they were highly qualified or competent sessions. And I hope that this judges or the juries might have faced some difficulties to identify the best papers presented in this particular conference. So let me conclude by congratulating the participants and also this team behind this success of this particular function, especially the team led by Dr. Jisha of the Jayachandran sir of the MG University, and Dr. Shabana Mol and Dr. Asha of the SAFI Institute of Advanced Study. This collaboration between these two institutions will definitely give some more and more activities like the conferences and workshops and also the research oriented programs in future. So I am very much happy to announce the result of this paper presentation, and let me present or let me announce the best paper presentation in faculty category. It is shared by two. One is OPFC4 Dr. Harish Chandra Anandaram. The paper is Computational Analysis of MicroRNA Based Gene Regulation by Top-Down Approach. Sri Krishna Arts and Science College, Coimbatore, Tamil Nadu. And it is actually bracketed by OPFC7, Dr. Deepthi Madai. The paper is Microscopic and Fluorescence Spectroscopic Analysis of a Lectin Purified from Leucaena leucophala, D. with seeds. The faculty, Department of Biotechnology, University of Calicut. Then it is the best paper presentation researcher category is OPRC7, Dr. Smita Vijaya. Paper is Bioactive Potential of Taxol extracted from the endophytic fungi Collectrichum, Glosporides isolated from Withania somnifera. School of Biosciences, Mahatma Gandhi University, Kottayam. The best paper presentation in student category is OPSC 8 Runal Vanjale. Paper Design and Synthesis of MS2 virus-like particles, carrier of Sirna for glioma therapy, Rajiv Gandhi Center for Biotechnology, Tirunelveli. The best poster presentation in student category, PPS 7 Ajay P. Dev, paper contemplating the antifungal activity of different solvent extracts of Engardium oxidate, the student Department of Biotechnology, KVM College of Science and Technology, Chertala. Then finally, the best poster presentation in faculty bar researcher category, PPFR2, Dr. Sheikh Adil Muhammad. The paper is Effect of Water Acidification and Sanitation on Gut Morphometry and Intestinal Microflora of Broiler Chicken. FVSC and AH, Suhama, SKUAST, Kashmir. Let me take this opportunity to congratulate the winners. And once again, I congratulate the organizers, and also I wish the participants have enriched much by attending this program and by listening to the papers presented by the faculties, researchers, and students. Let me conclude. Thank you. Thank you very much. Jai Hind.
Thank you, sir. Here we present the appreciation certificate to the keynote speakers, Dr. Keith R. Davis and Dr. Vijay K. Juneja.
Keith sir, can you please on your video? Keith sir. Oh. Okay, continue.
Okay, I think there is a technical issue there. So kindly accept it as a token of love and appreciation from us, from the team Bio-Inventiyon 2020. Gratitude unlocks the fullness of life. We have Dr. Shabana Mol S, Research Director. Yes? Hello? Okay, okay.
Yes. Here, Dr. C.
Yes, so here we present the appreciation certificate to Dr. Keith R. Davis.
Thank you very much.
You brought up a good point, and
okay, sir, thank you.
With my co-founder.
Okay, here we present the appreciation certificate for Dr. Vijay K. Juneja from team of Bio-Invention 2020, and we are truly indebted for your presence and efforts.
Knowing my co-founder, and in general, it would be much better to approach it from the perspective of instead of, hey, I feel like you're only doing 20% of the work, I'd like to take 20% of your equity and take it for myself, would be a much different conversation than I did a whole bunch of research. It looks like a typical CEO would be recruited at a 25% equity stake, I think it's reasonable when the CEO is recruited that I wouldn't dilute. We would take the equity from you.
Okay, as we move on. So as I've said before, gratitude unlocks the fullness of life. And we have Dr. Shabana Mol S., the Research Director of Safi Institute of Advanced Study. And I know that she needs no introduction, and the efforts and the toil that she has put into Bio-Inventiyon 2020 cannot be weighed. I invite you, ma'am, for the vote of thanks.
Thank you, Farah ma'am. Am I audible?
Yes, ma'am.
Yes, yes.
Good evening, ladies and gentlemen. As our principal sir mentioned in his address, we tried our level best to provide the best platform to draw knowledge from diverse areas of bioscience. And with proud, I must say, this was happened as a result of signing an MOU between National Institute of Plant Science and Technology, MG University, and Safi Institute of Advanced Study. I consider this as a privilege to open a link between one of the top-rated university in the country with our second home, our college. In fact, it was our second home because we were the alumni of School of Biosciences. And all this was happened because of our talent, highly talented teachers, Dr. Jisha, my supervising guide, my mentor, Dr. Jayachandran, Dr. Jyotish, Dr. Linu, Dr. Kirti, and many more role model teachers. And we were fortunate to have such role model teachers. During the poster presentation, I was much satisfied because at that time itself, I could recognize, we could attain what we have intended by organizing this such a way. Before that, I must congratulate all the winners, and I must say all the participants are winners too, because all presentations were excellent. All participants were excellently delivered their presentations, their ideas, their work, and they were very, very successfully defended also. As Principal Sir said, it was a tough work for all the chairs. I am sure about that. See, during the poster presentation session, one of the students, that is none other than the Ajay, the best, who got the best poster award in the student category. The chairs from Czech Republic, Dr. Jasim and Dr. Mansoor Ali, the vice principal of MES Maramballi College. Both of the persons were the chair, and they asked, what was the intention behind your study? And his answer was amazing. That he replied that his grandmother is the reason, because in his childhood he was about to view that, or about to observe that his grandmother was covering the pickles with washed leaves of washed cashew leaves. And when he grown up and ended to do in his biotechnology studies, during his project he started, if such things, or covering the leaves, covering the pickles with the leaves is able to prevent the contamination, there might be something within that leaves also. So such sparks, ideas, knowledge that everyone getting during the scientific studies is the stepping stones towards major invention, I must say like that. So we were fortunate to have all such beautiful and gracious delegates here. So let me move on to my duty. First, I would like to thank the guest of honor of today, a great philanthropist, Mr. C.P. Kunji Muhammad, Secretary and Manager of Safi Institute of Advanced Study, for his all-time efforts to keep our institute always uplifted. I would like to thank Dr. Keith from the bottom of my heart. I am greatly acknowledging him because his presentation time was on the morning, like 10 a.m. in India. It was about 1 a.m. midnight in US. He was much patient. He was such a beautifully minded person that he admitted our request and he was ready to deliver his beautiful, highly informative talk at such a time. Dr. Keith, please accept the gratitude from the bottom of our heart. And now we have just witnessed Dr. Vijay. He is conquering all the hearts of all the participants in the session. It was a wonderful talk. And sir, I extend a heart full of thankfulness to you on behalf of the team Bio-Inventiyon. And now I would like to thank Dr. Jayendran, sir, a wonderful teacher, an exceptional teacher in the molecular biology field, I must say like that, for all his encouragement and support throughout this webinar. And I'm proud that he is the director of School of Biosciences now. So, sir, please accept our gratitude on behalf of team Bio-Inventiyon. Now, I would like to thank our respected principal, sir, the leading light, the visionary leader of SIAS. We are fortunate to have such a great personality with us, and he extended all his support from the beginning of this webinar. So I thank you, sir. Thank you so much for all time support. Now it's the time to thank Dr. Jisha MS, the most updated lady scientist I have ever seen. Such an enthusiastic and encouraged mentor, guide, supervisor, teacher, research, such an updated person, very active, energetic, and we all, we the team Bio-Inventiyon comprises her research team, Sharin, Etna, Meenu, Dhanraj, and the two alumni of us, that is Asha ma'am and myself from School of Biosciences, along with other team members of Bio-Inventiyon in SIAS. So I don't know how should I the way to extend my thankfulness to Jisha ma'am, and this webinar, this web conference, all this effort, please accept as a token of gratitude from our side. Thank you, ma'am. Now, I would like to thank Dr. Shaini NK, Accuracy Coordinator, Staff Institute of Advanced Study, a person who is all time with us, a person who is actively engaging in the uplifting process of our college. Shaini ma'am, we are very grateful to you, and please accept our thankfulness on behalf of Team Bio-Inventiyon. Now, I would like to thank Dr. Asha for compiling the e-abstract during the inaugural session. As Jisha ma'am said, it was a wonderful process during the inauguration time. And our Honorable Vice Chancellor inaugurated our session, and sir was very much impressed with the proceedings. And as we promised, we have started the work for ISBN proceedings, ISBN books too. Asha ma'am, thank you for being there all time in the virtual platform as a support, as we did all time during our studies and now as colleagues. Thank you, ma'am. Now, I would like to extend a great thankfulness to Ms. Sherin, Ms. Etna, Ms. Meenu, Ms. Dhanraj, the research team members of Dr. Jisha, for the all-time help, all the help they rendered throughout this, from the beginning till now. And now we are moving on is going on. Thank you, dears. Now, I would like to thank all the eminent personalities who have chaired all the session. Dr. Jayachandran, Dr. Kirti TR from School of Biosciences, Dr. Lino Matthew, again from School of Biosciences, Dr. Jaykumaran Nair from the head of the Department of Biotechnology, Kerala University, Dr. Sumi, Dr. Sudha, Dr. Jasim B from Olomouc University, Czech Republic, Dr. Mansoor Ali, Vice Principal, MES College, Maramballi, Dr. Sharal Rabalo, Assistant Professor from Christ College, Dr. Vinod Kumar from Shastra deemed to be University. Everyone was very much patient because this is a virtual platform, not a physical one. Even though we were conducted it smoothly with little errors of network problems from different sides, it was a great chance to interact with all of these chairs. And I thank all the chairs. Now, I would like to thank the Master of the Ceremonies, again Sharon, Edna, Dr. Smita from Safi, Dr. Shaini NK again from SIAS, and the Master of Ceremony of this session, Mrs. Farah Barami, our HOD of Department of Microbiology, and all the staff, teaching and non-teaching staff of SIAS, especially the administration technical support staff, Salman, Junaid, especially Salman and Junaid, because they were here for all the technical support. And one person is there, that is Mr. Safir Muhammad from Saudi Arabia. He's the divisional manager of Toastmasters Al Jubail, Saudi Arabia, here for his love and technical support rendered for our conference. And the soul of this conference, the participants, the present is everyone. They deserves a great acknowledgement from our side. Thank you all for your kind participation, and I must thank the family who were being patient with us all the time when we were in the virtual platform. So I hope I remembered with thankfulness, with gratitude, almost everyone. If I missed anyone by chance, because I myself felt feeling very, what to say, nervous or like emptiness, something like that I am feeling. I don't know why. Again, I must especially thank the husband of Dr. Shaini NK, Unnishetan, who is still here with us for helping us in this night, all the time for us, for guarding us here. And if I missed anyone, please excuse me. And please, so I think let us wind up. Thank you all. Good night. Stay safe. Thank you.
Thank you. Thank you.
And that's fine. That's
Thank you very much.
but we haven't had very many men.
Thank you, Shabana.
Thank you. Thank you.
Delegates, all the academicians who have joined us, and have a nice day ahead.
Once again, thank you, Vijay sir, and thank you, Keith sir, for giving wonderful lecture and being with us in this valedictory function.
Thank you very much.
Thank you, Sir Gauri. Hello. Hello. Hello.
Hello, Dr. Gaudi Shankar, you want to tell something?
Okay, sir. Thank you. Thank you, ma'am. Thank you, ma'am, for the nice session. For the 3 days nice session. And again, we will meet if any conference will be there.
Okay, thank you, Gaudi Shankar.
Okay. Okay. Okay, ma'am. Thank you. Thank you, ma'am.
Any of the participants want to share your experience?
Sir, I want to share, madam.
Okay.
Okay,
You can
ma'am.
tell. You can tell now.
Okay. Actually, the 3 days conference was quite excellent and was unique because the one presentation by Davis sir and this keynote session by Vijay sir. Actually, these are from, I am from a different stream. I am from plant taxonomy, but actually I was very much excited to hear, listen again the talk of Vijay sir. His presentations were very nice. Actually, these things which are needed for our daily life, that is needed for the society, and if it can reach to the common people, this information, if it can reach to the common people, I think people must be saved from different types of diseases and disorders.
Thank you, Dr.
Thank
Gauri,
you.
for your comments.
Sir, your information is quite excellent. No comments
Thank
to
you.
say, because if such information is needed now for the people who are living in the grassroots level of the society. And
So, Gauri, when you
okay.
go ahead, go ahead.
Okay,
Go
sir.
ahead.
Actually, your information, it can reach.
മീറ്റിംഗ് അവിടെ എൻഡ് ചെയ്യട്ടെ അവരോട് പറഞ്ഞിട്ട് എൻഡ് ചെയ്യാം
Your information and your all the deliberation, Vijay sir, your information and all the deliberation, it can reach to the, if can society, the common people. Then I think it will be a revolution for the discovery of new concepts, so that some diseases cannot attend, some diseases, or as well as the organisms which are responsible for causing disease, those cannot reach to the people.
Thank you. Thank you very much.
We are very excellent.
Okay.
We are very excellent. For in future days, I will contact you, sir, over your mail.
Sure. And give my
Sir, I will
regards to
contact.
When you go back, give my regards to Dr. Gargi Day. You know her?
I know her, but from the last years, I am unable to contact with them because as you know that I'm working in a college. So
Yes. Okay.
During that
Excuse me,
time,
sir.
you come to that, and
Excuse
I was
me,
presenting my paper,
Dr.
and you also.
Gauri Shankar. Am I audible?
Okay, madam. Okay, madam. Okay, madam. Audible.
Sir,
Also, I received my
trying to
certificate, madam.
leave this
Okay.
soon. Shall I?
Okay, madam.
Shall I end the meeting?
Okay, ma'am. Thank you.
So
Thank you,
thank
madam.
you
Thank you.
so much for watching in the Zoom as well as in the YouTube live. Thank you all. You will get the certificate.
Ma'am, I have received certificate, but it's recording. May I get the recording of the 3 days seminar?
Yeah, sure, Sarah, but give us 5 or 7 days because we have to settle down with all these things, and we are taking
Okay.
classes also. After this, we are directly entering to our academics, so it will take time in between. So thank you so much. Thank
It will
you
be
so
sent to our
much
email,
for
na?
being with us,
Ma'am,
everyone. Yes,
ma'am,
sir.
it will be sent to our email.
Sir, have you filled
Recording.
the feedback form?
Okay, I have filled the feedback form, received the certificate, madam.
Yeah, please. You got the certificate. Okay.
Already received the certificate.
Certificate. Oh, very good. So,
Okay.
PS, we are winding up the session. We are concluding it. We are going to end the meeting once again.
Okay, madam.
Thank you so much for being with us
Thank you.
all the
Thank you.
time. For great
Okay,
encouragement,
thank
for great participation, for great registration. Thank you so much.
Okay, ma'am. Thank you. Thank you. Thank you,
So
Vijay sir.
let
Namaste.
me
Namaste.
conclude the
Namaste, madam.
meeting. Jisha ma'am.
Okay, Shabana. Okay.
Okay, Jayendra sir, can we stop? Dr. Jayendran sir.
Gone, I think.
Okay.
We can wind up, no
Thank you,
problem.
Paramam. You are still here. Thank you so much. Thank you, Shaini ma'am.
Thank you, ma'am.
You all the participants, and once again, congratulations to all the winners. And the participants are truly winners. I already mentioned that. And I must say, thankfulness to those two people from Philippines and Ethiopia who were here all the time during the 2 days. Oh, I am sorry, I forgot to mention it. Thank you so much.
Thank you, sir. Thank you, ma'am.
Okay, thank you, Shabana and team. We are looking
Ma'am,
forward for the
no,
next
no need to
association.
thank, ma'am. We should thank you because all these because of you only.
We are looking forward for the next association.
Sure. It's a lifelong association. Indeed.
Yeah. Okay, thank you.
Okay, ma'am. So, participants, goodbye.
Okay,
Thank you. Meet
bye.
again in the next
Goodbye.
venue. I am
Thank you.
closing the Zoom room. Thank
Okay,
you.
ma'am. Thank you. Thank you. Thank you. Thank you.
Shut down zero system.